of on behalf of all the committee members and PGR training team, I welcome all participants and our today's speaker, Dr. M.P. Rana sir. So it is very pleasing to have you today with us on this online platform for PGR training. So let me introduce sir with his brief introduction. So I would like to share my screen. I hope my screen is visible. Okay. So this is a brief biodata of our today's speaker, Dr. Mukesh Kumar Rana sir. He is currently principal scientist at the Division of Genomic Resources and with his specialization in plant breeding molecular genetics. So his nature of work and achievements include active involvement in the DNA fingerprinting of Indian crop varieties for their unique identification, DNA fingerprinting protocol development for molecular techniques, and he has developed many SSR, SRAP, ISSR, AFMP, RPD markers, and they are frequently being used for hybridity testing and seed purity testing. He has rendered DNA fingerprinting services to over 2 dozens of public and private sector organizations. So these facilities are being provided on regular basis to the organizations also. And there are many awards, only a few I could mention here. So he is the owner of DBT's Associateship of Young Scientists in Niche Areas of Biotechnology, NAIP ICAR International Fellowship in USA under the field of Plant Genomic Resources Conservation. He is also Fellow of Indian Society of Genetics and Plant Breeding and Indian Society of Plant Genetic Resources. He is also the owner of gold medal, citation, and certificate of honor by Himotkarsh Sahitya Sanskriti एवं Jan Kalyan Parishad HP. So he also has many number of publications in national and international renowned journals. So, sir, with this brief introduction, I would like to welcome you and I request you to please provide your insights on the topic of today's presentation, which is DNA fingerprinting. And I hope our participants will be benefited with this your sharing of knowledge. So with this, this session is over to you, sir. Please, you can start sharing your screen and start your lecture for today. Thank you.
Thank you, Dr. Mamta. Let me first share my screen. Is it visible?
Yes, sir.
Okay.
Yeah, it's visible.
Thank you for the introduction. First of all, I'm thankful to the organizing committee for giving me this opportunity to express my views in the area. I'm not an expert in the area, but would like to share my little knowledge and expertise with you people. So if you have any questions, you can flag your concerns in the chat box. I would like to revert to you in case I'm not able to answer your queries after the short discussion time that has been allotted to us. So I will be talking in next 45 minutes about DNA fingerprinting of crop varieties. This is how I've structured my presentation. First of all, I'll be talking about plant genetic resources in relation to varieties, crop varieties. Why varieties as a genetic resource is an important component of the whole gamut of biodiversity. Then we'll be talking about DNA markers and DNA fingerprinting, the technique per se, and the techniques, basic DNA marker techniques that are being used in DNA fingerprinting. I'll be touching upon. I'll not be going into details of the science part. I'll be just trying to give you just an overview of the technology and how it is being used. So then we'll be touching upon the global and national scenarios in the use of DNA markers and varietal identification. DNA fingerprinting, there are many different views about it, but different applications, but will be restricting strictly to varietal identification. DNA fingerprinting as such is used for intraspecific identification of varieties, be it the croplands or be it the microorganisms or be it the human beings. So, and then we'll be touching upon the constraints and what could be the way forward under Indian scenario. You've been hearing probably for the last couple of days about plant genetic resources. This slide would be redundant, but just to make a link between the rest of the presentation, plant genetic resources are the basic raw material for crop improvement, as they are reservoirs of genes for resistance or tolerance to various biotic and abiotic stresses. And these plant genetic resources, they're very useful in developing designer crops. Nowadays, we're talking about transgenic crops, genome edited crops like that. So until unless we have these plant genetic resources, we can't embark upon those steps for developing designer crops. And in the light of this poor public acceptance of transgenic crops, plant genetic resources, they are very useful resource in case of varietal development program. And further, when we talk about conventional biological diversity nowadays and various IPR related issues that are restricting the accessibility of the germplasm globally, the importance of PGR, their conservation, their utilization has further increased. So these plant genetic resources, before the signing of conventional biological diversity, they were considered as the heritage of mankind. But the things changed post CBD, when these plant genetic resources had their sovereign rights of the states upon them. And when we say that states have, or the nations have sovereign rights on the plant genetic resources, it is very important on the part of the states to protect their invaluable wealth. So in India, we have to conform to these international obligations. We have this Protection of Plant Varieties and Farmers' Rights Act 2001, which was enacted upon and came into force. And consequent upon the enactment of this legislation, Protection of Plant Varieties and Farmers' Rights Authority was established, who is looking after the protection part of these crop varieties. So plant genetic resources, as you know, that it is a just component of big gamut of biodiversity, under which we have agrobiodiversity, then PGR. PGR is a small component of biodiversity, which includes land races, farmer varieties, parental lines of hybrids, release varieties, wild and weedy species, genetic stocks, and of course nowadays I put here in this list also genetically modified organisms, transgenic crops, gene edited crops, etc. So what could be a variety? Different definitions are there, and varietal definition varies from the perspective of a farmer and then from the botanical definition of a variety. Subsection 16 of Section 2 of Seeds Act 1966 defines it as a subdivision of a kind identifiable by growth, yield, plant fruit, seed, or other characteristics. International Code of Nomenclature of Cultivable Plants, which says that variety is an assemblage of cultivable plants, which is clearly distinguished by any characters, be that morphological or physiological or cytological, chemical or otherwise, and which reproduced when this assemblage of cultivated plants is reproduced, retains its distinguishing characters. So whatever its distinguishing characters are, that they are passed on from one generation to the next. UPOV, the Convention of the International Union for Protection of Plant Variety, New Varieties, defines variety as a grouping within a single botanical taxon of the lowest known rank, which grouping, irrespective of whether the conditions of the grant of a breeder's right are fully met, that grouping can be defined by the expression of characteristics resulting from a given genotype or a combination of genotypes. It is both. And which grouping can be distinguished from any other plant grouping by the expression of at least one of the set characteristics. So it should be distinguishable by at least one characteristic, and considering as a unit with regard to its suitability for being propagated unchanged. So this hasn't, if any distinguishing character is there that is retained unchanged. When we are talking about DNA fingerprinting, then fingerprint of the varieties, essential derived varieties. This is another concept, very important, which needs to be understood properly. For example, when you have a variety, if you make small cosmetic changes in that, 99.9% of the genome is same. But what is then essential derived variety? A variety predominantly derived from another initial variety, which retains the expression of the essential characteristics from the genotype or combination of genotypes of the initial variety. That is essential derived variety. So how do we protect our crop varieties? There are different ways. Registration of the varieties, germplasm. For example, PPVFRA is undertaking the task of registering of the varieties or the germplasm. We at NBPGR has also very soft kind of protection wherein trait-specific germplasms are being registered. And it is very important. Rather, it's a statutory requirement that a plant variety must be novel or distinct from the existing varieties. It has to be uniform in the essential characteristics, and those characteristics should be stable over the generations. So, and this protection of crop varieties, enactment of different acts is very useful in granting plant breeders their rights. We have these legal instruments for variety protection in India. For example, the Patents Act 1970, the Biological Diversity Act 2002, which are not directly related for the protection, but we have the Protection of Plant Varieties and Farmers' Rights Act 2001, which is directly related to the protection of plant varieties. Coming to the marker, DNA markers, I'm just making foundation to make you understand. Marker, what is a marker? Any genetic element, and that genetic element can be a locus, an allele, or a DNA sequence, or any chromosomal feature, which can be readily detected by phenotypic, cytological, or molecular techniques. If you remember, we have these different cytological features like knobs in maize, chromosomal knobs. So it can be anything that is any genetic element that can be a marker. But very importantly, that whatever genetic element we consider as a marker, it has to have sound inheritance. That means it should be heritable, one generation to the next generation, or it should follow Mendelian inheritance, Mendelian behavior. Only then we will call it marker. Only then it can be traced back. Only then it can be localized. Only then whatever manipulations we want to do with it, we can do. So any ideal marker needs to have these characteristics: highly polymorphic, reproducible, codominant, even distribution in the genome. It has to have sufficient discriminating ability. It shouldn't be subject to environmental influences. It has to be neutral, inexpensive, easy to measure. For example, DNA has a lot of scope to act as to provide you the markers. For example, in this example, we can see that one sequence we are taking a 10 nucleotide long sequence, and at one position in the nucleotide here, there can be four possible. There are four possibilities because there are four bases A, T, G, C. So in this first sequence, you can see instead of A here it is T. At the second position here instead of T it is C. In the third position here instead of G it is A. So we have large number of possibilities for this small segment of 10 nucleotides. Four possibilities are there at one position. So 4 raised to power 10 different the different ways in this the different ways in which this sequence can be rearranged is 10 raised to power 4 raised to power 10. That is 1 billion. 1 billion. Sorry, not 1 billion. 4 raised to power 10. I don't know what exactly that number would be. And human genome is 3 billions. And likewise, our crop genomes are varying in their nucleotide content. So 3 billion means 4 raised to power 3 billion. That is the possibility of variation in DNA. So coming to the different methodologies for DNA fingerprinting, DNA sequencing is the ultimate. Okay, so it is ultimate to detect the variation, but it is not possible intraspecifically to genotype all the varieties, DNA fingerprint all the varieties at this moment. Of course, that the technology is becoming cheaper, technology is advancing, and this is becoming feasible. Maybe it will be possible in coming couple of years. So, and we see that a large number of genomes in plants, at least more than 1,000 genomes, plant genomes have been sequenced, and that information is available with us, and we can use that information for varied applications, including DNA fingerprinting, which we'll be discussing in the subsequent talk. Why DNA markers only? When we have a lot of morphological markers, this is very basic slide. Everybody knows this because, as I told, there's no dearth of DNA markers, and we can generate a huge amount of markers, abundance of markers, which is virtually speaking unlimited number of markers. And when we talk about DNA, we are talking about the expressed part of the DNA, or the expression, because DNA is totally, it is not influenced by the plant development. So you take DNA even from the root or the shoot or the flower, it will be the same. Whereas once the gene or the DNA part that is expressed, it can have a lot of variations. So you are looking at the physical entity, physical thing directly. So it is very important. So and these markers have no pleiotropic effects, no influence, because we are looking directly at the DNA, physical entity. There's no pleiotropism, no influence of the environment, no epistatic effects, so and so forth. So we have numerous applications of these DNA markers, including in genetic studies, where you can we are aware that we are using for genotyping, genome mapping, phylogenetics, and many other applications as well. They are used as breeding tools also. For example, in QTL analysis, marker assisted selection, marker assisted backcrossing, marker assisted recurrent selection, association mapping, gene pyramiding, so and so forth. So these markers are used as tools nowadays, and many varieties, new varieties, and different crop plants have been released. So in production of quality, in controlling quality controls, genetic purity testing, trait purity testing, hybridity testing, these are being used. We're talking here about these legal applications, plant variety protection, patenting, essential derivation. So these DNA markers have numerous applications. So we're in this talk, we'll be focusing on just one component. So coming to what DNA fingerprinting is, this technology is around us since 1985, when it was first put forth by its inventor, Sir Alex Jeffreys. So it's nothing but a way of identifying a specific individual rather than simply identifying a species or some particular trait. So as I told you in the beginning, that DNA fingerprinting, when we're talking about it, is intraspecific. When it comes, when we talk about species identification, then it is DNA barcoding. So I'm not touching that area at all, which is entirely very interesting and new area. DNA barcoding and a tree of life has been generated using the markers, and that applications are entirely different. DNA fingerprinting is also known as DNA typing, genetic fingerprinting, or DNA profiling. Many people do not know exactly the small differences between these. When we say that DNA fingerprinting, it is totally different than what typing or profiling is. So, given the time, we'll see what those differences are. And in humans, it is currently used for identifying criminals, victims in the hospitals, for children swapping. Different applications are there. And likewise, it has numerous applications in plants as well. So just fingerprints versus DNA fingerprints. Earlier, for the identification, we used to have the fingerprints, human fingerprints. And it is said that each individual has unique fingerprints. Likewise, we can generate unique DNA profiles or DNA fingerprints for individuals. And the probability of two unrelated individuals, you sample two individuals randomly from any population, and what is the probability of having their identical DNA fingerprints is much, much lower than that of their fingerprints being identical. So the probability of identity or matching is very, very low when we use DNA fingerprints rather than fingerprints. So it's just a small comparison in case of humans and plants, cultivars. I would just say that in plants, DNA fingerprinting is much, much complicated. It is very broad as compared to the humans. In humans, we're talking about only one species, that is the Homo sapiens. Of course, we have races. But when we are talking about the plants or the cultivars, we have multitude of species, subspecies, races, so on and so forth. For example, I had worked in cotton, and DNA fingerprinting protocol was to be developed. So in cotton, there are around 24, 25 different species. But 4 species, Gossypium barbadens, Arboreum, Hirsutum, Herbaceum, these are cultivated ones. And the protocol or the DNA fingerprinting technology developed in one species is not applicable to the other one. It is entirely different. But in humans, we have only one species, Homo sapiens. So it is very straightforward. In humans, similarity in requirements, because there isn't much variation. Whereas in plants, we have diverse requirements. All outbreeding, humans all are outbreeding. Whereas we know that pollination behavior in plants, we have from vegetatively propagated to the cross, entirely 100% cross pollinated crops. So a lot of variation is there. In humans, we talk about one individual, one profile. But in plants, we have different genetic compositions. We have open pollinator varieties. We have hybrids. We have composites. We have synthetics. We have pure lines, inbreeds, varieties like that. So we have different populations of different genetic compositions. In humans, there are very weak substructures, whereas when we compare these things in the plants, from strong to very strong to weak substructures in the populations. Allelic specificity in races, whereas common alleles across groups. Applications are identity, paternity. These are the applications. Whereas we have here identity testing, purity testing, relatedness, genetic makeup, many other things. So in nutshell, what I want to say here is that It is not a straightforward process in plants as compared to the humans. Some of the basic molecular biology techniques, as I told you, my talk is very basic. I'm not going into very details, keeping in view the uh, background of the participants here. Many of you might be knowing, many of you are not even aware. So I thought of for discussing this. PCR, nowadays everybody knows PCR, polymerase chain reaction, which was introduced in 1985 by Kary Mullis, and he got even Nobel Prize for this discovery. And there's nowadays hardly any field in biology. You talk about any field which is not touched by this PCR. And in this PCR, it is nothing but for a layman, xeroxing of DNA. PCR allows a short stretch of DNA, usually which is fewer than 3,000 base pairs, to be amplified to about 1 million fold for further investigation. So, from one, as we all know that DNA is a double-stranded structure. After first cycle you get 2 copies. Then after 2 cycles you get 4 copies. So in PCR machine, which is a typical PCR machine, which is shown here, after 35 cycles, you will be having 2 raised to power 35. That is 34 billion copies of that stretch of DNA in just 2 hours, and which can be used for further downstream applications. And this is how PCR works. First of all, in PCR, we denature the DNA at a very high temperature, 95 degrees centigrade. DNA becomes single-stranded, and we anneal primers, which is the next step, at 50 to 65 degrees centigrade, varying upon the annealing temperature of the primers. And then we carry out extension. So this whole completes 1 cycle, and this cycle is then made to work 30 cycles or 35 cycles or 40 cycles in the lab conditions. Then very important, another technique is gel electrophoresis. Once you have carried out PCR in, say, for example, 10, 20 varieties, different varieties, you want to electrophorese that DNA. You want to separate DNA and see where are the difference, how we are going to identify your varieties or distinguish your varieties. So electrophoresis is a technique for separation and analysis of macromolecules, be it DNA or RNA or proteins, based on their size and charge. As we know that DNA is negatively charged, so it will move towards positive end, anode. So we load our DNA on one side, and after 2 to 3 hours, you carry out that electric charge, and after 2 to 3 hours, you can see those PCR products, they are separated from one another, and you can see the differences, if any. And we can, and this is just means how do we loading dye is added, we stain that DNA, and ultimately the figures like this you generate. Some popular DNA fingerprinting techniques that are being used. As I told, that DNA sequencing is the ultimate, and as of November 2020, 1,139 genomes have been sequenced. And Arabidopsis in plants was the first genome to be sequenced, followed by rice, popular, medicago, so and so forth. If you dive into the literature, you'll find n number of tools for fingerprinting. For example, restriction fragment, length polymorphism, VNTRs, allele-specific oligonucleotides. I'm not going to go into the details, but you can see that this list continues. SSR, CAPS, MAPS, scar markers, SNPs, samples, ISSR, so and so forth. So this is endless list. But as I told you earlier, that DNA sequencing is the ultimate. Once you sequence, everything is known to you. And in case of these DNA markers or these techniques, we are targeting specific regions in the genome. We're targeting specific regions in the genome, and those regions are compared across a set of cultivars. Another technique that came into being in 1980s was the restriction fragment length polymorphism. Nowadays, nobody's using this method, but this is very, very robust method wherein we make use of the restriction endonucleases enzymes. Okay, so restriction endonucleases, we have different types of restriction enzymes which produce either staggered ends or the flanking ends, sticky ends or the staggered ends. So you can see here how it works. For example, you have two varieties, variety 1, variety 2. We just extract DNA, cleave that DNA with the restriction enzymes. Say, for example, this is the DNA from this genotype first, variety first, second variety. And as I told that these endonuclease enzymes are very, very specific. And say in the genotype 1 variety from this restriction site is present here at 3 positions. In genotype 2 or variety 2, it is present here. Here, what will happen if there is any change? So, for example, this A is changed to G, adenine is changed to guanine. So enzyme won't be able to recognize this sequence. So it won't make a cut here. Rather, cut would be made here and here. So it will be a longer fragment and it will be a shorter fragment. So when you carry out electrophoresis, separate the DNA of these two varieties, you will find that genotype 1 has a longer fragment, which will move shorter distance here compared to the variety 2 or the genotype 2, which will move farther in the electrophoresis gel. So you can see the differences. So this marker is useful for differentiating genotype 1 and 2. So southern hybridization is this how we carry out the preparation of this visualizing the fragments. In 1980s, Welch and McLeod, they gave another technique, random amplified polymorphic DNA, which is a very simple PCR based. The earlier one, RFLP method, that is restriction based. Here, then many PCR based RAPD and onwards, many PCR based methods came into being, and nowadays PCR based methods are very common. It is a very basic technique which uses not 2 primers, but only 1 primer. And the same primer that binds, that acts as a forward primer as well as a reverse primer. And after PCR, the amplified products are separated on the agarose gels and visualized using ethidium bromide staining. And this is a typical gel image that has been produced in a lab in cotton. So these all are around 50 samples, and you can see the differences among the cultivars. And this is a dominant marker technique. So this is just another elucidation for the RAPD technique. AFLP, which is AFLP amplified fragment length polymorphism, is a combination of both these techniques: restriction-based RFLP and PCR-based RAPD. And we can generate large number of markers here, large number of markers. And this is a typical scheme. We use 2 different types of enzymes: a rare cutter and a frequent cutter. A rare cutter and frequent cutter, 2 enzymes are used. A restriction is carried out. Okay, and we ligate the adapter molecules to this restriction ends, and PCR is carried out then using specific pre-selective primers and selective primers in two different steps. And the PCR products are ultimately electrophoresed using polyacrylamide gels. So this is a lot of genetics behind this AFLP technique. I'm not going to describe it here. Simple sequence repeat markers. The simple sequence that repeat or microsatellites are tandemly repeated mono, di, tri, tetra, penta, and hexanucleotide motifs. So differences are in the cultivars are due to the repeat length of these motifs. Okay. So for example, mononucleotide means single nucleotide is repeated n number of times. This is an example of dinucleotide GT, for example. It can be anything GT or AT or GC or whatever it is. This is repeated 6 times here. It can be repeated 100 times, 200 times, 50 times, likewise. And how polymorphism is generated using SSR markers. You can see, for example, variety A, variety B. You just, as I told you earlier, also extract the DNA from these two varieties, carry out PCR. So in variety A, this motif is repeated 8 times. In variety B, it is repeated 10 times. So this will be a longer fragment after PCR compared to this fragment. And the beauty of these repeat motifs in the nature in plants is that their flanking sequences are conserved, and that conservation tendency has been utilized for generating for designing primers. Forward primer here, second this reverse primer. So these are conserved across genotypes in a species. So if these are conserved, so this remains constant, and you can amplify the intervening region, variable region, and it will move shorter distance because it will produce a longer fragment. So variety B and variety A can be distinguished. It will be 8 into 2, 16. This will be 20 nucleotides long. Just theoretical example. It can be anything. So you can differentiate your two different varieties. And the same methodology has been used in humans also. STRs, sequence tagged STS sequence tag sites are being used nowadays for DNA fingerprinting in humans, whereas earlier on the restriction-based VNTs, variable number of tandem repeats, those were used in the humans. Coming to the SNPs. Nowadays, we are talking a lot about the single nucleotide polymorphisms, a new class of third generation markers. SNP is defined as a single base change in a DNA sequence that occurs in significant proportion, more than 1% of a large population. So, for example, you take individual 1 and 2, you just sequence it, and after sequencing, you find the differences. So, this in individual 1 here, there is a change with respect to these two individuals. So, this is SNP, single nucleotide polymorphism. Or there can be small stretches which are either additions or deletions, so which we call them as indels. So there are large number of SNPs available nowadays. Not many genomes have been sequenced, and we have not many SNP polymorphisms within a short stretch also, and which can be utilized for DNA fingerprinting. And we have large number of different DNA sequencing genotyping SNP genotyping methods. As the technology is advancing, more and more detection methods are being unraveled. That has disadvantage, I would say, as far as fingerprinting is concerned. We will talk about that. This is the list of genotyping assays of SNPs continues. So for fingerprinting, SNPs have advantages like a higher recovery of information from degraded DNA samples. And this is many. For example, when you have herbarium specimens for very obsolete varieties or genotypes, when you want to use SNPs are okay, because you can extract, you can find polymorphisms with respect to the degraded DNA as well. And SNPs have lower mutation rates. But for fingerprinting, globally, there have been a lot of debate going on, and in humans, they haven't been used for identification or DNA fingerprinting. Likewise, in plants also, Many research publications are coming out, but they have disadvantages which are listed here. More number of SNPs needed to compare to SSR for equal power of discrimination. And it is said that 40 to 60 SNPs would be required for a set of 10 to 12 SSR markers. With respect to SNPs, difficult data interpretation with more loci and amplification products. Whereas SSRs or STS interpretation is very easy. SNPs are very sensitive to the quantity and integrity of the input template DNA with more number of loci. So likewise, many drawbacks are there. That is why they are not being preferred for fingerprinting purposes. Otherwise, for genome, for genotype, germplasm characterization, diversity analysis, these are very, very useful. So this is just a classification of different markers techniques. I'm not going to dwell on it. You can say that's just first generation, second generation, third generation, restriction based, then PCR came, second generation, then sequencing came. So we have many third generation markers or sequencing based markers. And all these markers can be classified into RDMs or GTMs or FMs. Random DNA markers whose location can't be traced on the genome, and gene targeted markers which are derived from the polymorphic regions from the genes, and functional markers which are derived from the polymorphic sequences within the gene, but they're associated with a certain trait, which nowadays these are becoming very, very useful functional markers as functional annotations for the sequence genomes are being carried out. So use of these markers depends upon different many things. And in this table, there is this comparison. And these markers, whatever I listed, those are some 3-4 markers I described. They are the basic ones. And there are many other markers which are modifications of these markers. So you can see some markers are co-dominant, some are dominant markers based on their reproducibility, polymorphism level, required DNA quality, marker index, genome abundance, cost, sequencing, status, PCR requirement, visualization, and how much DNA we require for carrying out the assays. So you can see here their plus points and negative points. Coming to the use of molecular markers in varietal production, International Union for Protection of Plant Varieties, UPO, now uses SSR-based techniques for variety protection. And UPO, they have identified a certain set of these markers in only a handful of crops right at the moment. And in the court of law, when there is any dispute or something like that, SSR or DNA-based markers, they have the supportive role. They're not the exclusive ones to decide or make a decree, but they have the supportive role. So DNA markers, let me say at this moment that they're not used per se to decide the fate in the law of court, but instead they are being used for strengthening with other evidences. Egypt was the first to make to accept this technology for DNA fingerprints of a new hybrid variety, and its parents they have made it a requisite now in 2-3 crops. In India, Genetic Engineering Approval Committee has been insisting on DNA evidence to prove background genotype of the transgenics for more than 10 years now. And at ICR also in India, we have made this DNA fingerprinting essential for the release of varieties from 2010 onwards. But there are many ifs and buts related to that also. We'll be just discussing those things also. In India, at an ICAR and NBPGR, we have this NRC on DNA fingerprinting erstwhile, which was established in 1996 with the primary mandate of developing and standardizing protocols for DNA fingerprinting for agricultural crops. And this NRC on DNA fingerprinting functioned for 2 plan periods with dedicated funding and manpower to put in place the scientific basis for DNA fingerprinting. And then in 2013, It was our division was renamed as Division of Genomic Resources, with a primary mandate for developing genomic resources and molecular profiling. So primary mandate was shifted towards genomic resources, but DNA fingerprinting also continues. And in our RAC's also research advisory committee meetings, also emphasis have laid that DNA fingerprinting or profiling needs to be undertaken. And we are routinely meeting the requirement of DNA profiling of pre-released varietal proposals, which are submitted both by the public sector as well as the private sector. So when Central Subcommittee on Crop Standards Notification and Release of Varieties requires under the item number 5 that DNA profile of variety, hybrid, inbred, or A and A, B are a line of the hybrid vis-a-vis check variety. This is the requirement nowadays. So under this provision, we are undertaking DNA profiling of crop varieties at ICR and BPGR. And further, it says that the detailed information on the molecular discrimination should be provided. Such information can be developed at crop-based institutes, NBPGR, or other labs. And the information should include details of amplicons, name, sequence, number, primer sequence, with respect to the polymorphic markers. And the relevant photographs should also be attached. So there's a lot of discrepancy in the methodology being followed for DNA fingerprinting, and a lot many things are to be straightened up at the moment. So you can see for the last 5 years, we have carried out DNA fingerprinting a number of crops. Here is the list. And in the last 5 years, from 16 to 22, in 48 crops, we have carried out DNA profiling work. In the last 5 years, 553 varietal proposals have been DNA profiled, and this is the year-wise split. Maximum number in case of the cereals, followed by the fibers, particularly in cotton, because a lot of seed samples, these samples keep coming to us. And we are generating income also at ICR and BPGR by rendering these services. On an average 5 to 6 lakhs per year, we are creating the resources. We are charging for this service rupees 5,000 from the public sector organizations and rupees 10,000 from private sector organizations per sample, in addition to 18% GST, and these rates are going to be revised very soon. Some of the concerns and what would the way forward. As I said, that number of institutes, not only NBPGR, but many other institutes are also undertaking this work. There has to be harmony. We need to have a standardized DNA fingerprinting protocol in each crop, and which technique is to be used. Okay, whether we want to use multi-locus DNA fingerprinting technique or the single locus method, then how many loci. What could be the sample size? For example, we have a lot many variation as far as the pollination behavior of the crops is concerned. Self-pollinated, cross-pollinated, vegetatively propagated. What should be the appropriate size, sample size? Acceptable range of types. What are the statistical parameters? What are the statistical parameters? As I was telling you in the beginning, that DNA profiling needs to be understood vis-a-vis DNA fingerprinting. Fingerprinting much very robust as profiling. You can generate using anonymous markers, anything, but you need to strengthen those profiles using certain statistical methods. Okay, only then fingerprints are generated. You need to have the probability of identity match. You need to have a database beforehand. Finding those whatever alleles or whatever markers you are using, their allele frequencies in the population are also to be known. Only then they can be called as fingerprints. When we are whether we have clones or the populations, what are essential derived varieties, what is the threshold level for calling a variety essentially derived variety or delimiting a particular variety. So these are some technical, beyond this also, there are many technical issues also, which needs to be fixed up. So way forward, I would say identification and the use of robust and mapped markers. So this is very essential. For example, across laboratories, we need to have harmony. So if lab one is using particular marker, in other lab also, if they are generating DNA profiles or the fingerprints, same marker has to be used, and these needs to be validated first. We need to development of quality standard protocols. After thorough validation across 3 to 4 independent labs, we need to quality standard protocols, DNA isolation protocol that has to be fixed across labs. PCR conditions have to be fixed. Electrophoresis conditions have to be fixed. Sometimes it is people are using gel-based or maybe page-based or maybe capillary-based. Which method? There has to be harmony. Establishment of central referral lab in case of disputes. If there is any dispute, do we have any such structure available? No. We need to have a central referral lab where those disputed cases can be referred to. Development of efficient monitoring system for protocols. I'm observing because DNA fingerprinting is generated and in the varietal release committee, it is a requirement. So they're generated like anything just to fulfill that requirement. No, there's no proper monitoring system. So that has to be put in place. DNA fingerprinting database development. You can't call your profiles fingerprints until you have database first. Allelic frequency is calculated, mismatches in the existing varieties that is done. Only then you can say that yes, your DNA fingerprints are valid. Adoption of standard reporting format for DNA fingerprint results. So everybody is reporting in their own way. We need to have a standard reporting format, and we are at NBPGR, we are working towards that also. We have prepared certain, for example, here you can see in this where from the we got the sample crop, which technique was used, which are the primers used, how many number of alleles, or the markers, and what was your criteria for selecting those markers. Okay, so besides that, many fingerprints also needs to be put in place. So we need to have a standard reporting format for DNA fingerprints across different labs of the country. And establishment of an advisory body for implementation of quality control and network, which is not existing there. We need an advisory board, advisory body. Establishment of a network of DNA fingerprinting labs and updating that network skills by HRD. That is very essentially needed. And creation of scientific and legal awareness regarding the authenticity and reliability of DNA fingerprints. So these are some of the ideas that I put forward. So in humans in India, this DNA fingerprinting bill, a proposed legislation, which is called DNA Technology Use and Application Regulation Bill 2019. This was put in the parliament. And the bill was originally proposed in 2007. And in 2012, drafting the bill started in humans, I'm talking. And this bill consists of a molecule, this proposed DNA profiling board, and under this bill, there's a proposed DNA profiling board, but that is one thing. And second is creation of the database. Those are very essential things required. And this bill aims to regulate the use of DNA technology, including DNA analysis, databases, and sample collection. It further seeks to establish national and regional DNA databases for forensics and other lawful purposes and provide for the collection, analysis, and storage of DNA samples. But what I meant to say is it's just proposed legislation. It was put into 2019 again in the Lok Sabha, and the Parliamentary Standing Committee on Science and Technology, Environment, Forests and Climate Change in 2021 raised certain concerns. The certain concerns were flagged regarding the provisions under this bill. So this is still undergoing. So until unless we think on those lines, do we have a proposed plan to build? No, we haven't even contemplating that. We haven't even drafted. We haven't even started. First step we haven't taken yet. So we are far behind. In many other countries, for humans identification, they have this acts in place now. There are worldwide, in UK, USA, Australia, many other countries. So this technology is being used, and in plants, it is still at its infancy, and many things are needed to be accomplished before we go in that direction. So this is all what I want to share. Thank you very much, and you're welcome for any queries, questions.
Thank you so much, sir. Participants, if you have any questions, you can either directly ask to sir or drop in the chat box also. Any questions?
I have one question from Vinod Kumar, JNKVB. What is the minimum requirement to establish lab for molecular characterization? Minimum requirement, you need to have a lab space. You need to have basic instruments like PCR machines and DNA extraction setup, electrophoresis setup, and it all depends upon which kind of marker you want to use. Okay, so if you're more interested, we can discuss it separately. I'm just talking about the very layman kind of things. Maybe you have to start from the agarose gel based system. But if you have to start using SNPs or some other system, your requirement would be totally different.
Any other questions? Anyone, please? So there's another question in the chat box.
What are the minimum requirements of seed for DUS testing? So this question I can't answer because DUS using molecular markers or morphological markers. And as far as DUS using molecular markers is concerned, that is not in place yet. Mohammad Raza. Mohammad Raza, does that answer my question? Okay, any other question?
Sir, Dr. Muhammad Raza replied that
Yeah, morphological, sir. Morphological.
Morphological.
Minimum requirements of seed. I don't know. Understand requirement of seeds for what? You will be planting a material in the field or using the standard whatever DOS requirements are there, and taking those observations. And it will vary from crop to crop. There are standard. Yeah, markers are best for UTL mapping. Yes, but whatever I talked about, that is entirely different. That was for fingerprinting, for identity testing only. My talk was entirely focused on cultivar identification. In one of my slides, I made a mention of different applications: UTL mapping, genome mapping, quality control, like that. There are n number of applications of the markers, but how those markers are useful for identity testing, uh, for IPR protection particularly. I think, Mamita, there's no other question.
Yes,
So
sir,
we
I think
can,
no more questions are coming
yeah.
up. Okay, so we will stop this discussion here. And I would like to thank Dr. Rana sir. Sir, you have certainly polished our knowledge on this topic again. And you explained some of the very good topics and techniques which are in their refined formats and are being used for DNA fingerprinting. And also you explained the specific requirements and parameters that are much needed for to fingerprint any variety or germplasm. So I hope our participants must have got some elated knowledge and also upgradation in their knowledge. So with all this, sir, I would like to thank you. And if anyone wish to get any of the varieties or any germplasm, any query regarding the profiling of the varieties and fingerprinting, so they can contact, I think, Dr. Rana sir, and he will be happy to help you in this regard. So thank you so much, sir.
Definitely. Okay, thank you so much, and I thank the audience, all the participants for a very patient listening. Hopefully, they might be listening.
Yes, sir. Uh, okay, participants. So now this was the lecture scheduled before the tea time. So because the lecture is over, we will break for the tea, and after 15-20 minutes we will be back. So I think next lecture is scheduled from 11. So we will meet at 11. So please make yourself available by the 11 a.m. Okay. Sir, Ravi sir, you are there?
Yes, I am there. Yes.
Yeah, okay. So is it okay that we'll be back by 11, sir?
11 o'clock. Yes.
Yeah, yeah.
Yes.
Okay, okay.
Okay. Thank you.
I am starting with the introduction of our next speaker who is with us, Dr. Rakesh Singh sir. So I have prepared a brief introduction of him. So let me share my screen so that you all can view also. So is it visible? Hope my screen is visible to everyone.
Yeah, his screen is
Yes,
visible.
ma'am, it is visible.
Okay. So we have with us Dr. Rakesh Singh sir from Division of Genomic Resources with his specialization in biotechnology in the area of plant science. And with great pleasure, I would like to inform that this sir has done a vast work in the field of rice. He has genotyped more than 729 rice varieties and more than 9,000 rice accessions with the SSR markers. And in-house, he has developed 36 plex SNP primer panel, also a core of northeastern Indian rice collections from 7,000 accessions and East Coast region rice collection of more than 2,000 accessions using the SNP markers. Sir also has quite a vast experience for the genetic enhancement of the crops like chickpea, brinjal, bear, grapes, etc. And his laboratory also provides DNA fingerprinting services, especially in the rice and chickpea crop. And sir has also developed genomic resources and database in minor and as well as various major crops. He is currently handling one of the mega projects that pertains to the functional genomics in rice and also includes the aspects of genetic stability. Sir is also providing editorial assistance in most of the renowned journals like Indian Journal of Plant Genetic Resources, then associate editorship in the Indian Journal of Genetics and Plant Breeding. He is the consulting editor, also a member of the advisory committee board. So many posts he is taking care of. And apart from this, sir also has bagged many awards in his name. Like in 2020, sir got Dr. B.R. Barwale Award, and in 2017, R.K. Arora Best Research Paper Award. In 2015, he got the Fellow of Indian Society of Genetics and Plant Breeding. So there are so many, like Jawaharlal Nehru Award in 2008. So many awards are there, sir. How many to name? And besides this, you have so many publications, more than 106 research papers in international and national journals, and more than 2,000 citations. So, sir, with this introduction, I feel very much grateful to introduce such a great personality like you, and we are very much pleased to have you with us on this platform. Thank you so much for being with us. Now, I would like to request you to kindly share your knowledge with us so that our audience as we also get benefited from, with this. Now, I will stop sharing my screen, and now the session is over to you, sir.
Thank you, Mamta, for nice introduction. So
Sure.
I would like to thank, uh, first of all, I would thank our organizers for giving this opportunity to present. Uh, my slides are visible?
Yes, sir, visible.
It's visible? Okay.
Yeah, just
Visible,
put them
sir?
in slide mode, sir.
Okay. Okay. Yeah, now it's in slide mode.
Yeah.
Yes, sir.
So thanks to organizer for giving me this opportunity to speak on this important topic, that is genomic resource generation and trait validation. So genomic resource, I would like to explain all participants is that whatever DNA, RNA related information you are generating by studying any crop plant is actually genomic resource. And if you are using those genomic resources for certain trait discovery or identification, that is trait validation. Okay, so I will start with my lecture is in two parts. First part is genomic resource generation, how to generate genomic resources in different crops. And second part of my lecture is that how to use those genomic resources in trait discovery or trait validation. So I have deliberately not taken rice wheat as an example for genomic resource generation, because I know in the audience there may be some persons who are working in case of horticultural crops, medicinal plants, or underutilized crops where there is no genomic information. No genome sequencing has been done, like rice and wheat, where there is lot of information available already. So if you are working in area of rice and wheat, you have lot of genomic information already available. But for those who are working in such crops where there is no genome sequencing project or no genomic information has been generated, for them this lecture will be more useful, because I'm going to tell you that if you are working in any crop where there is no genomic information, then this is good opportunity for you to generate those genomic information and get good credit. So first I would like to tell what are the genomic resources. So if you are generating molecular marker, probes, and primers, particularly if you are generating genomic SSR, EST SSR, or SNP markers, these are the good genomic resource you are generating. Second kind of genomic resource can be transcriptome resources, if you are studying expressed region of your plant. Third kind will be QTL resources. If you are doing biparental mapping and if you are identifying certain QTL regions, you are generating genomic resources. Fourth can be the QTN resources. If you are doing GWAS, you are generating quantitative trait nucleotide, and that can be good resource. Genome sequencing project, yes, definitely they are best genomic resource. If you are generating genomic libraries, yes, you are contributing towards genomic resource. If you are cloning candidate genes, this can be another genomic resource. If you are doing comparative genomics, yes, you are generating genomic resource. If you are using marker-based, if you are generating marker-based code, yes, you are doing genotyping and you are generating genomic resource in form of cores, which are essential for discovery of several traits. So these are the some of the list which I am presenting, which comes under category of genomic resources. There may be others also which I might have not enlisted here. I am enlisting only those genomic resources which generally breeders and agriculture scientists they come across using in these resources. So in my subsequent lecture, my first focus will be on generation of genomic resources in the form of molecular markers. So how genomic SSR, EST SSR, and SNPs can be generated in medicinal or horticultural crops or underutilized crops, that I will take up first. So just to remind you about molecular markers, so you must be knowing that RAPD, ISSR, AFLP, CAPS, SCAR, these were the most popular markers in the beginning of when the molecular biology started. And these were PCR-based marker and were very popular till 2000, initial stages. Then a phase come when people started generating some sequence-based markers. So Scott and CBDP markers, they comes under category of these markers. At present, we are generating maximum genomic resources in the form of microsatellite marker, EST-SSR marker, and SNP markers. Yes, non-PCR based techniques, RFLP was the oldest marker, but for present breeding program and molecular biology activities, we are generating maximum markers related to microsatellite, EST, SSR, and SNP. So am I audible to you all? Hello?
Yes, sir. Yes, sir.
Yes, sir. Yes, sir. Audible,
Okay.
audible.
Okay.
Clearly.
So I am not expanding these abbreviations RAPD, ISS, RAF, LP because you might be knowing or some other speaker might have explained you about these markers. So to save time, I am not going to explain these markers. So my subsequent presentation will be focused only on how SSR marker can be generated, how EST SSR marker can be generated, and how SNP marker can be generated and utilized in different crops. So for developing this microsatellite markers, there can be two strategies. One strategy is if sequence is already available. Suppose some genome project has already been completed and their data has been already uploaded on NCBI or some other server. So you can exploit that genome sequence for development of microsatellite markers. So this is one strategy which you can follow for microsatellite marker development. Second strategy will be where there is no sequence information available. So under this category, horticultural crop, medicinal plants, underutilized crops, mainly these crops comes under this category where there is no sequence information available. Now question comes how to generate genomic resources or microsatellite markers in these crops. For that, there are still two strategies. One is that you use microsatellite sequence as a probe. Means you are using artificial repeat sequences, probe in the form of probe labeled with or some radioactive substance or with some enzymes, so that you get profile on the gel or on your nylon membranes. So this is one strategy for getting microsatellite markers in your crop. But these are very restricted kind of study because this study cannot be used by others. So the third alternate strategy is to use microsatellite by developing microsatellite libraries. So for this kind of study, what we do, we generate microsatellite libraries, we do enrichment and isolate sequence positive clones, and then after sequencing that sequence positive clone, we design a specific primer, and then this primer are immortal. It can be used by anyone for doing diversity study or biparental mapping or some other studies. So in our laboratories, what we are following this third strategy, that we are generating microsatellite libraries and we are generating primers and we are utilizing them for different crop studies. So this is one example which I am quoting here, that in case of Tinospora cardifolia, it's a medicinal plant where there is no genomic information available. So we started generating genomic information in this crop by developing genomic SSR markers. So here I have enlisted how to proceed with for development of this SSR marker, genomic SSR marker in case of Tinospora cardifolia. So this kind of marker generation can be done with moderate molecular biology facilities. It doesn't require very high-fi machines. Simply moderate level of instruments, electrophoresis unit, and some shaker and laminar flow will be sufficient to generate this kind of information in those crops where there is no genomic information. So in case of Tinospora cardifolia, we have generated genomic SSR markers using these steps. That DNA was isolated, then it was fractionated, then microsatellite containing fragments were captured. For capturing this microsatellite enriched fragment, kits are available. You can take those kits and you can do enrichment of your this microsatellite library. You have to do transformation because before sequencing, you have to clone your this microsatellite regions. After cloning this microsatellite region, you have to do positive clone selection, and then you do sequencing. If you are after doing sequencing, you have to generate primers. First, you have to identify these SSR regions in that the clones, and if you are getting positive regions, using SSR Finder is one of the software which we have used. There are so many softwares available for discovery of SSR, MISA and so many. So SSR Finder here we have used for identifying these SSR regions, and then this from this SSR regions we identified primers which can amplify this SSR region in your plants. So primer 3 software was used for generating primer, and then after generating this primer, the last step is to prove that whatever microsatellite you have isolated from unknown library is working in your system. So for this, you will have to do validation of your markers. So here you can see that this this marker has been, whatever marker we have generated, we have validated on 28 accessions of Tinospora cardifolia, and you can see that based on 107 genomic SSR marker we have generated, and this has been used for diversity study. But this primer can be used for if you are generating population in case of Giloy or Tinospora cardifolia, you can use them for molecular mapping purpose also. So this work has been published in this journal. Here I have given for your reference. So detailed study, detailed steps you can learn by going through this paper. So whatever steps we have followed for generating genomic resource in case of Tinospora cardifolia for genomic SSR marker, you will get here. Next step strategy which I had shown you was that where genomic sequence is already available. So in case of Amaranthus, we were very fortunate that the genomic sequences was already available in database. So what we did, that instead of generating sequences, we took those sequences, we downloaded those sequences, and then we did two kinds of study. First, SSR identification and marker development, and second, SNP identification and chip development. So for SSR identification and marker development, what we did, we used MISA instead of SSR Finder. Here we have used MISA software, microsatellite identification tool. It's a tool because the genome was large, and for faster discovery of SSR regions, this software is very, very good software. So if you are using, within one day, you will get a whole genome SSR identified. Once SSR identification was done, then primer 3 was used for primer designing. And then SSR primers were first validated in silico, because we were generating thousands and in lakhs this software, this SSR marker. So first what we did before doing wet lab experimentation, we got them validated through ePCR. When ePCR showed good result, then we went for final validation. Similarly, in case of Amaranth, this SNP discovery, we also use this genome sequence, which was published in 2017. And then what we do, we use SAM tool and BAM tool to for SNP calling. And then after getting high quality SNP, we filtered assembly, and then this SNP were used for probe development. And finally, this probe was used for chip development. So by using this strategy, we have developed in our lab a 64K SNP chip. Also, by using this method, in case of Amaranth, where this kind of genomic resource was not available earlier. So whatever SSR markers we have developed is being published in agriculture, you can go through this reference, and you can get information that how in silico strategy can help you in generating SSR marker in those crop where this information is lacking. Here, so after ePCR, what we did wet lab validation. Here you can see that we have used, I'm presenting here this profile of 94 accessions of this amaranth using one genomic SSR marker. You can see pattern, and then these markers, more than 57 markers were used for this diversity study analysis. So by using in silico study, we generated GSSR marker in case of amaranthus crop, an underutilized crop where there is no genomic information available. In case of SNP marker, which we derived from those in silico information, we developed a chip of 64K. I'm not presenting 64K data here because that chip is under patenting process. So that will be violation of that patent. So I'm not presenting. I'm giving you brief outline that how these 64K were used for developing chip and how it got validated on this amaranthus sample. So here you can see that all the SNP marker which we use for generating that chip were covering all 16 chromosomes of amaranthus. And here you can see that this red, green, and black color pattern. So the red color indicates that these SNPs are located in genic regions. It means they are present on gene. And green region is indicating that they are present on intergenic region. Okay, it means between two genes. So here you can see that how what is the density of those SNP marker which we have used for this diversity study. And black regions are those regions where we failed to develop SNP markers, due to one or other regions. So you can see that black regions are very few. So this 64K SNP chip which we developed was sufficient, had sufficient coverage of this amaranth genome. And this chip was used for characterizing 445 genotypes of this amaranth. And you can see the distribution of phylogenetic distribution of those 445 samples which we have studied. Here you can see that almost 16-17 state sample we have taken. We have taken some exotic sample also, and they were showing this different distribution pattern. Here color indicates that the sample from which state they have taken. So here you can see that how intermixing of these samples are occurring from samples collected from different states. Next strategy is development of EST SSR, express sequence tag. It means these markers are developed from expressed region of genome. So again, I am taking example of Giloy, Tinospora cardifolia. Here we have done, besides genomic SSR marker, we have also generated EST SSR markers also. For EST SSR marker development, the primary requirement is that you have to isolate total RNA. First, you will have to isolate total RNA, and from total RNA, you will have to separate out your mRNA. Once mRNA comes in your hand, you go for reverse transcription and you develop your cDNA. And these cDNA are then put in machine for doing sequencing. So this is the strategy how these transcripts were assembled, and then they were utilized for primer development, SSR mining, and validation. So for detailed study, you can go through this paper, which I have enlisted here. You will get idea that how this EST SSR marker can be generated as a genomic resource by using a transcriptome approach. So whatever EST SSR marker we generated, we got them validated, because whatever marker you are generating, resource from unknown sources you are generating, their validation is very important. Otherwise, no one will believe that you have generated some good genomic information. So after every generation, therefore, I am showing you one slide for how it has been validated. Without validation, your publication may not be possible. So whatever genomic resource you are generating, try to validate them on some samples. Okay, so here you can see that how this primers which were EST SSR markers which were generated got validated on Tynospora cardifolia accessions through genetic diversity study. So after generating and validating those genomic resources in different crops, this is one of the important activity that you will have to upload them in the form of databases, because whatever genomic information you have generated, you have used for your purpose. Now this should be available to others for their study or for their utilization. So whatever genomic resources we have generated in our lab, we have uploaded them in the form of databases. So on the NBPGR website, you can go on this medicinal plant genomic resource database page, and here you will get two these databases. First is Tynospora database, and second is Endographis database. So in case of Tynospora and Endographis, we have generated genomic SSR as well as EST SSR markers, and their information is available on those databases. So once you will click on these databases, you will read this link, you will reach to this database. So this is the front page of that database of Tynospora cardifolia. Okay, so whatever genomic resources we have generated is available here. So in this database, we have also kept one section of tutorial that how to use these databases for generating markers or downloading genes or transcription factors, because in these databases, we have given so many genomic resources which can be utilized by researcher for their purpose. So you can go to these tabs, there are 6-7 tabs, and you can read them. And then you will get idea that what this database actually contains beside SSR markers. In this database, we have given ready-made 3 primer pair for each gene. But if you feel that you need to add some more, you need to design some additional primers, you can go in this database inside, and you can design your own primer and get them downloaded. So this much flexibility has been given in this database for primer generation and their utilization. We have given blast facility also. You can blast these EST sequences or gene sequences to study, to do comparative study with other crop species. So this facility is also available. So how this database works, or what information and features are available in this database, you can further read by reading this paper, the reference which we have given here. So if you go through this paper and read about this database, and then utilize this database if you are working on Tynospora cardifolia or related species to Tynospora cardifolia. The other thing what we have done, that we have protected our database by copyright. Though this database is freely available, but we have taken copyright of this database. So if you are developing database, I will suggest that yes, you protect your database by applying copyright, so that your database remains protected. Similarly, in case of Endographis, we have developed this database. If you are working on Endographis or related species to Endographis, you can utilize whatever information, genomic resource we have stored here, genomic SSR, EST SSR, gene sequences, transcription factor. If you are working on these aspects, you can open this database and you can get information about genomic resources available in this database. And you can read this paper also, where details have been given. And similarly, similar to Giloy, this endographis database has been also protected by taking copyright. Similarly, in case of Amaranthus, whatever SSR and SNP resources we have generated, we have stored here. So you can see here in tabs that SSR, SNPs, transcription factor, microRNAs, transporters, all those genomic resources which we could derive from public database, we have stored here for researcher. They can utilize these genomic resources for their study if you are working in case of amaranth or related crop. And detail you can read read in this reference which I have given. This paper is accepted, but very soon it will be available. I think within 15 days this paper will be available online for your because this is freely available, so you can use this paper for using this database, and this database is also protected. So after generating genomic resources, validating and preparing database, these resources are now available for utilization, that they can be used directly for trait discovery and other things. You can generate markers or genomic resources through cross-species transferability study also. This is one of the example in case of vetiver, where in our lab we have generated genomic resources for vetiver, SSR markers in case of vetiver using rice sequences. The idea of this study was that since vetiver and rice belong to same family Poaceae, so what we did that we thought that there must be some synteny between rice and vetiver grass, and we tried to transfer SSR marker of rice on vetiver, and we succeeded in transferring those SSR marker to vetiver. You can see here, I have given here the pattern that how rice SSR is getting amplified on vetiver, a grass on which there is no genomic information available, and these markers have helped in studying the genetic diversity which was available in case of vetiver. So this reference I have given, that that was published in this journal, Industrial Crop, and by going through this paper, you can get idea that how cross species or a cross genera, you can transfer markers from one species to another species and generate genomic resources for studying their diversity or their conservation strategy or whatever purpose you want to do that. So by making this presentation, I am making you to believe that if genomic resource is not available in the crop in which you are working, you can generate genomic resources by using moderate facility, molecular biology facilities. You don't need big machines for generating this kind of information. Then another strategy for generating genomic resource is through co-development. So for here I am taking rice deliberately because rice in the gene bank, rice has largest collection. I think more than 1,14,000. So how to utilize such large collection for discovery of genes for disease resistance, stress tolerance, high yield, nutrient use, etc. So how to utilize them? Because if you are using 1 lakh sample at a time, it's humanly impossible to genotype them and then use them for core development, because the basis of core collection here I have given in flowchart, and this might have been told by other speaker, because this is 8th day of my after 8th day I am presenting my work. So you might have been told by other speakers also that what is core, what is mini core. You might be knowing all these things. So the basis is that if you are here, I am presenting that you see A, B, C, D, means 4 types of alleles are existing, suppose in total collection. If you are going for a primary core, if you are reducing it, there is possibility that 100% this allele is getting represented in your primary core. If you are going for further reduction, there is possibility that you may restore 99% of original diversity. But if you are going for mini core, there is chances that you may lose some alleles. Okay, so this is the problem with core, that if you go reduce the size of your samples, there is possibility that you may lose some alleles. Okay, so to avoid this problem, we had followed different strategy in case of rice for developing molecular core. So what I have done in my lab, we have used different strategy. Instead of taking 1 lakh sample together, we have taken rice sample region wise, and we started developing core region wise, so that whatever diversity is existing in that region, at least they should come get represented 100% or 99% in the core. So using that strategy here for developing this rice core, we have used SNP markers, the most advanced marker. And probably when we published this work in 2013 in class, probably using SNP marker, probably this was the first core which we reported in case of rice. So by taking 7,000 rice sample, rice accessions, germplasm from all 7 states of Northeast, we generated a core of 701 accessions. And similarly, in case of coastal rice collection, we have generated only East Coast rice collection, because this is one of the major centers for rice diversity. And we have generated a core of 247 accessions. Both these references I have given here. So you can go through these papers and you can see that what strategy actually we have followed for developing cores in case of rice using SNP markers, the 3rd generation marker. Because when you will, I don't have sufficient time to explain everything, but I would like to say something about any core. When we were developing any core, actually the 7,000 samples were not uniformly distributed across all 7 states, because Assam sample was dominating over Tripura and Meghalaya sample. Assam sample was 3,000, 4,000, whereas in case of Tripura and Meghalaya, it was only 500. So what we did, what strategy we did, that so that we should not lose any allele, we developed core first state-wise, and then we combined them together to develop core of that region, so that whatever allele, useful allele were present in Meghalaya and Tripura sample should not get lost while making any core collection. So when you will go through this paper, you will get idea that what strategy we actually follow for developing cores, so that whatever useful alleles are existing in those regions should not get lost in your core. After developing this core was further characterized for development of mini core, because 700 number was still high. So we used 50K SNP chips, 50,000 SNP markers we have used for developing mini core. So you can see that how this mini core of 194 sample is getting distributed into 7 groups. So these 7 groups represents different types of rice which we know: indica type, japonica type, os type. These groups are getting represented in this mini core. Once we got this idea, then we thought of exploiting this mini core for trait discovery, the trait validation, the second part of my topic. So for that, what we did, before going to association mapping actual result, I would like to give brief introduction about how association mapping is done. So here you can see the flow chart where I will explain that how association mapping we do normally. So for us doing association mapping, first of all, we need population. This population may be anything: natural, breeding, germplasm, land race, maybe multi-parental breeding lines. It may be anything. So once this association panel or population is available with you, what first step would be that you go for phenotypic data generation. Okay, the trait which you want to study. Then you have to do genotyping, because in my case things were easy because I generated molecular cores. So genotypic data were already known to me. But if you are using phenotypic approach to develop core, definitely you will have to go for genotyping. Once this marker genotypic data is available, you will have to go for population structure study. So population structure you can develop by using structure software or using principal coordinate analysis. So these two methods you can use for generating population structure. Both software are freely available. You can use them and utilize them for your genome-wide association studies. And second is marker kinship. This you will get from the software Tassel or Gapit, whatever software you are using. You will get this information from based on your that software. Once this your Q value and K value are available with you, you can use generalized linear model or multiple linear model, and you can do association studies, and you can identify QTLs or QTNs. So this is in nutshell how we do GWAS. I have explained you. I hope you must have understood that how GWAS is done for trait discovery. So I'm taking example of this any core, mini core, which for which we have generated using 50,000 SNP marker, 50K SNP chip. So density you can see. Density you can see that how these SNP markers were distributed over all 12 chromosomes. So you can see that sufficient density markers were densely present on rice chromosome for trait discovery. And you can see that here I am presenting phenotypic data for grain length and grain width. Okay, so you can see that how grain length and width were varying in case of northeast germplasm samples, mini core. And after generating phenotypic data and genotypic data, this is phenotypic data, genotypic data, then we went for principal coordinate analysis. You can see that when principal coordinate analysis was done, you can see that this again this population was getting distributed in 7 groups. You can see how 7 groups are appearing. And similarly, when NJ tree was used, you can see that how 7 clusters are getting generated. So by using principal coordinate analysis, we generated Q matrix. By this marker analysis, we generated K matrix. So phenotypic data was with me, genotypic data was with me. I generated Q matrix and K matrix. So all these 4 information was generated in case of rice any core using 50K chip. And now you can see once GWAS was done for grain width, you can see how this this is the actually threshold level. Here I have used threshold level 4, log value 4. So whatever markers which are going above 4 are actually showing marker trait association for grain width. So by using this method, we could identify 80 marker trait association. But 80 marker trait association doesn't mean that 80 genes are actually involved in this for this trait. Once we do find that I will show in subsequent slide, this number drastically get reduced. Okay, suppose if I increase my lot value to 7, so number will drastically reduce. Okay, so this number changes based on the study, or we do finer analysis and we identify that okay, which this marker is associated with which gene. So when we do gene analysis, then we get actual picture that okay, this marker is associated with that gene. Or suppose if this number also gets reduced, because sometimes you get some putative genes, non-functional genes, for which this association comes. So we reject those markers for further study. Similarly, in case of grain length, we are getting 5 marker trait association, and for aroma we got 16 marker trait association for any core. And here I have given some information that how this is for grain width actually, that you can see that we are predicting how different genes which are linked to these markers. Okay, so that's why this is not called QTL. This is called quantitative trait nucleotide, QTNs, because polymorphism or the changes which we are predicting in the form of change in nucleotides, and due to change in nucleotides, phenotypic variation occurs. So which nucleotide change is associated with which phenotype is very easily gets predicted in case of GWAS studies. So GWAS study has the advantage. Besides, there are so many disadvantages of these GWAS studies, false discovery and other things. But if you are doing stringent analysis, definitely you will get directly the nucleotide variation responsible for a particular phenotype. And you can see here from this log value, how log value we have used. We have taken more than 4 and it has gone maximum up to 7. So this is one example that the genomic resource generated in the form of core has been utilized for trait discovery or trait validation for quality traits in rice. Another example I will take for medicinal plant, where trait is entirely different. For medicinal plants, secondary metabolites are more important. So I will be taking, for marker generation, I had given example of Giloetinospora cardifolia. For trait discovery, I will give example of Endographis paniculata, Kalme. Am I audible to you all? Hello?
Yes, sir.
Yes, sir. Yes, sir.
Yes, sir.
Yes, sir. Very much.
You are clearly audible, sir.
Okay. So next example for trait discovery, I am taking Endographis paniculata, where endographolite content, how that can be manipulated, or the genomic resource which we have generated in the form of database has been utilized for endographolite production. So once this genomic resource was generated and database was prepared, then we thought of utilizing that database for validation of traits. So what we did, that we took a NAC transcription factor. Actually, NAC is not a single gene. It's a combination of 3 genes. You can see NAM, ATAF, and CUC. So NAC represents these 3 genes. Okay. And this is one of the largest transcription family in case of plants. And they are known to be involved in stress regulation and secondary metabolite regulation. So we selected this NAC transcription from Endographis paniculata, which we generated through transcriptome in-house in our laboratory. Okay. So this strategy was followed for validating that NAC transcription factor and its role for endographylite production, that is trait discovery, that how this NAC is involved in endographylite production. So first, in silico identification, definitely we downloaded this NAC transcription factor from our database, which is available at NBPGR website. RNA isolation, cDNA synthesis, PCR amplification, because when we do this transcriptomes, we don't get full length genes because it's a partial gene sequences get generated. So what we did, next challenge was to generate full length gene. For that, we did race 3 and race 5, so that gene which were missing from 5' region and 3' regions, they get fully isolated. And then we did the diterpenoid analysis, profiling, and then expression analysis, and by using this abscisic acid, because where this transcription factor is actually working, that needs to be localized. For that, we did expression analysis by treating them with abscisic acid. And then once we got that, okay, this transcription factor is working on Endographis paniculata and is controlling endographylite production, the second challenge was to establish this system in heterologous system also. And for that, we took Nicotiana tobacum, because in case of endographis, when we searched nearby species, it was very difficult to get those species for validation. So since Nicotiana tobacum is very well worked out in case of in molecular biology, this thing, it's a model plant just like Arabidopsis thaliana. So we validated this transcription factor in case of tobacco. So we inserted this our transcription factor inside tobacco plant, and we extracted mRNA just to see that whether this transcription factor is manipulating the pathway which is operating and responsible for endographylite production. So once we did, now I'm presenting the result. Here I would like to request the organizers that this is my ongoing research program. So this should not, the slide should not be placed on website or somewhere else, because we have not published this result yet.
Sir, slides we are not publishing, but recording is being published. So we should not, sir. Recording, yes. Actually, it is being published on YouTube.
No, for 64K chip, I have deliberately removed everything.
Okay, sir.
Because this is under patent, and this work is under this thing, under publication. So I don't want that this should be published somewhere. Otherwise, reviewer will say that you have presented this work somewhere.
Okay, sir. Okay, sir.
Okay. So just
we'll take care, sir.
Yeah, you take care of all those things. Because I want to make these participants aware that how genomic resources are powerful tool for discovery of genes and trait. That's why I am presenting here. So once we did in silico analysis, up to this we have submitted to gene bank, no problem. Further subsequent slides are very sensitive slides. So once we did this in silico analysis, we got amplified these 3 sequences. Full length gene we got, and we submitted them to gene bank, NCBI gene bank, for generating this accession ID, gene ID. So these are the gene IDs which we have generated in case of NAC transcription factors. So these 3 NAC factors. AP represents Endographis paniculata NAC. Okay, so this number we have given to these transcription factors, which we have isolated from our database, which is loaded at NBPGR website. And further, before doing wet lab, we confirm that whether these sequences which we have generated full length, they contain this important regulatory elements are not. And we got these regulatory elements present in our full-length transcription factor. Further, we did in silico analysis just before doing wet lab, that whether these transcription factors which we have isolated full length from our database, they are getting represented in some other databases or not. When we did this BLAST analysis, we got that AP-NAC, which we had isolated from our database, is very closely related to Oryza sativa NAC22. Okay, so this gives confidence that whatever we are doing is in right direction. And similarly, this AP-NAC 2122 was getting in databases represented in database of Olia europea. So this AP-NAC 2122 is closer to Olia europea 2122. Similarly, this AP-NAC 83 was closely related to Arabidopsis thaliana transcription factor, that is NAC41. For their database, it is NAC41. For our database, this sequence is AP-NAC 83. So this gave us confidence that whatever NAC transcription factor we have studied in silico, and by using race PCR, we have generated full length, are actually getting represented in other databases, and they are actually NAC transcription factor. So we have not isolated something misleading. They are getting represented in other databases. And then we started doing our wet lab research. And we did first we did tissue expression analysis, just to see that where this transcription factor is actually getting expressed in plant. And for that, we took sample from root, stem, and leaf. And you can see that this transcription factor is getting maximum expressed in case of leaf, the actual site where endographelites expression or synthesis occurs. So this gave us confidence. Yes, whatever these three transcription factors we have full length we have isolated, two of them at least, AP-NAC 2 and 21 and 22 are getting expressed in case of leaf. The other was actually getting expressed in case of root. So it might be related to some other activity, but these two are directly linked to endographolite production. So next step, what we did, we tried to overexpress this, just to see that overexpression of these things occurs or not. Transcription factor is getting overexpressed or not, or endographolite production is also increasing simultaneously or not. For this, what we did, that we treated these plants with abscisic acid, and then we isolated sample at 0, 6 hour, 12 hour, and 14 hours. And then simultaneously, what we did diterpenoid analysis, just to see that whether accumulation of this endographolite is occurring, getting increased or not. So two things we did together, expression analysis and diterpenoid expression also. Once we did expression analysis, you can see the pattern that how this expression of NAC transcription factor is increasing after abscisic acid treatment. You can see that expression of this AP-NAC2 was increasing drastically. From 0 hour, it was almost constant. 6 hour, it reached up to twofold, and then you can see that it has reached up to 9.6-fold. So after abscisic acid treatment, this transcription factor overexpression occurred. Simultaneously, when we did this metabolite analysis, endographolite content also increased. Which data? This data is not I am showing here. So this confirmed that, okay, AP-NAC2 and AP-NAC1, NAC21 and 22 are getting overexpressed in leaf, and when abscisic acid is given, its overexpression is also occurring. So definitely these two transcription factors are controlling endographolite production. Then now question comes that where because endographite production pathway is very long and two pathways simultaneously operates. This mevalonic acid pathway and methyl erythral 4-phosphate pathway. So where this actually this transcription factor is binding and manipulating endographite production. So what we did to show that where this NAC transcription factor is binding, we took these 6 genes. These 6 genes we targeted just to see that where is that candidate gene where this NAC transcription factor is binding and manipulating the endographite production. And you see, after giving treatment, we analyze these 6 genes, and you can see that DXR, HMGR, and SDS are showing maximum change after this abscisic acid treatment. Okay, so these 3 are actually candidate genes where our this transcription factor is binding, okay, and manipulating the endographite production. So after getting this idea that these 3 genes can be the candidate gene where this NAC is binding, what we did, that we did heterologous expression analysis. Means that we try to express our this endographite transcription factor into tobacco, because whatever study till this stage we had done was done in endographis itself in vivo. So evidence for manipulation should be established in heterologous system or some third-party system. Then people will believe that whatever transcription factor we are saying is manipulating and any pathway will be established. So what we did, we took tobacco plants, and in tobacco plants, what we did, that whatever full-length cDNA of NAC transcription factor we had generated, and we showed that they are controlling pathway of the 3 candidate gene, we transferred that NAC transcription gene into tobacco by using this construct, okay? And then this construct was transformed into tobacco by using agrobacterium transformation method, okay? And then after transformation, this infiltrate were isolated from tobacco leaf, okay? And mRNA was taken out, cDNA was generated, and then transient expression analysis was done. And you can see that in case of tobacco control, we took here the infiltrate was buffer. Only buffer was induced instead of construct. And you can see that all these 3 transcription factors were getting expressed in case of tobacco also. So this shows that these transcription factors are actually getting expressed. They are working, they are binding with DNA, and they are manipulating, okay? And then we the HMGR gene, which was showing maximum expression, 9.6-fold manipulation in case of Endographis paniculata, that gene was also studied in case of tobacco. And you can see that when expression of HMGR gene was studied in infiltrate, this upregulation of HMGR was also observed. It means our transcription factor was binding with HMGR gene and was getting upregulated in case of tobacco. So whatever pattern which we noticed in case of Endographis paniculata is similar pattern is getting established in case of tobacco. So this shows that the whatever NAC transcription factor we had identified in case of Endographis paniculata, and we have established that it is binding with endographite pathway gene and manipulating endographite production, is getting expressed in case of tobacco also. So this establishes that yes, this is useful genomic resource or candidate gene which can be utilized for manipulating endographite production in case of Endographis paniculata. So this is one of the classical example that how genomic resource can be targeted for a specific trait and its manipulation. And further, one step further, we took this HMGR sequence. We have done getting from this promoter region, and we have done analysis of this promoter region, and we have identified this putative region where this our NAC transcription factor was binding. So it gives sufficient evidence that this is useful genomic resource NAC transcription factor, which we have isolated from transcriptome and being utilized for endographylite production. So this can be used as a candidate potential gene which can be utilized for manipulating endographylite production. So whenever you are doing this kind of study, you will have to collect sufficient evidences. Otherwise, people will not believe that whatever you are saying is correct. So you will have to use these steps to get a useful gene or a trait-related gene which can be utilized for future breeding program or manipulating or transgenic or genome editing. You can use these genes for that purpose. But at the last, I would like to say that we have at our Division of Genomic Resources, there is one database for National Genomic Resource Repository in the name of Genomic Resource Repository. It also contains a lot of information about genomic DNA, libraries, markers, QTLs, because this is public database. Here anyone can submit his or her genomic information. It will be curated and stored here and will be utilized by other researchers. So suppose if you fail to develop your own databases or you don't have facility to develop your own databases, you can deposit your information here at this genomic resource repository. This will be curated and will be stored in your name and will be utilized by others. So I will request that if you have any information, you can submit in these databases also. So at last, thank you. Thank you very much for giving me this opportunity to speak on this important topic.
Thank you so much, sir. And really, it was so much fascinating to know that so much of work is going on in your laboratory. And I'm sure that just like me, our audience also felt that we consistently keep listening to you. And now I request our audience, if they have any question, please ask to the sir. And I'm sure that sir will be happy to answer the questions.
Yeah, definitely.
Some of the questions are there in the chat box also, sir. Should I read it for you or?
Yeah, you can read it. You can read it.
Okay. Sir, I think Dr. Sheetal Patel, she has asked that what is ePCR?
It is the electronic PCR, in silico analysis. So what we do that once we design our primer for this thing on this is a sequence, what we do that we have several other related sequences generated from same species on different from resequencing data. So what we do that we put that sequence, different sequences and that primer, and we do electronic PCR. So that this software has facility that it amplifies that band and shows on computer that yes, this primer is working. So this helps us in preserving our resources. Suppose if you develop hundreds of markers, 50 fails to generate on your genome. So that will be total waste in wet lab. So we do ePCR first, we validate on electronically, then whatever primers are working perfectly on computer, we take those sequences only for primer synthesis and they utilize them for doing our diversity analysis or molecular mapping studies. That is ePCR is freely available on net.
Okay. So I hope that she has heard the answer. Now, the next question is from Dr. J.M. Nida Gundi. He or she, I don't know by the name, but she is asking that in principle, GWAS takes account of historic recombination in an unstructured population, thus including released varieties or advanced lines derived from structured population in association mapping panel affects precision of study.
Novel discovery sometimes may not occur because you will be landing on same things that you won't be getting new recombination and other things. But yes, people are using, still using. That's why I have quoted that people are using breeding lines because they believe that once they develop breeding line, new recombination occurs, and that new recombination may be exploited for discovery. Okay, so not release varieties. If you are using, if you are developing mapping population or you are developing breeding line, then GWAS, yes, you can go for that because artificially you develop new recombinations, and that can be studied by using GWAS. So not for varieties, not for varieties. It won't be much useful. For breeding lines, it will be useful.
Sir, another question from again Dr. Sheetal Patel. She is asking that GWAS, how we can know that there is false association between trait and marker?
That you will have to, because for every SNP, position is known. So by using that position, you can go to genome and see that in that region, whether there is any gene is existing or something false intergenic region is there. So by doing finer analysis at gene level, you can know that this thing. And second thing is that you go for Ben-Ferroni corrections. For false discovery, you go for Ben-Ferroni corrections. If you are increasing your lot more than 5, definitely whatever false association is there, it will get rejected. So that is the way, or either you apply Ben-Ferroni correction or you go for manual method, you go for at genomic position, you see that whether there is any gene present or not, and that is useful for your trait or not. Based on that, you can discard those genes which are not useful for your purpose, particularly QTLs, where there are so many genes are involved for controlling that trait.
Sir, next question is from Dr. Devraj, TNAU. He's asking for GWAS, how many locations we have to fix for multi-location testing?
3 locations.
Any other questions from participants? These are from chat box that I read for you. Anyone interested can please ask the question from sir. Participants. Sir, one or two questions I would also like to ask if our participants. Sir, that you explained that while this SNP mapping, we get some regions which are very high in density that give the high dense SNP regions. Some are low, but some do not have at all any SNP. So what is the reason? They do not have any variation.
Okay. This is actually related to FE matrix chip. This problem is related to FE matrix chip because they say that if you are identifying one SNP, there should not be another SNP at least 200 base pair left side, at least 200 base pair right side. So you are eliminating 400 base pair nearby SNPs because once they do SNP calling, if SNPs are present in between, they actually interfere with your signals and you don't get clear-cut signal. So it has nothing to do with your SNP discovery. We discover so many SNPs, but when we go for chip fabrication, due to all these chip fabrication conditions, actually those SNP gets deleted. Okay, so they give opportunity to you that you decide that which one you want actually. So suppose if you in a particular 1 centimorgan or 1 kb region, suppose 6 SNPs you are getting. They want only 2 SNP in that region. So you will have to select which 2 you actually want. And due to fabrication conditioning conditions, you will have to eliminate those additional SNPs. So this is the reason that sometimes these gaps occurs.
So does it
Otherwise,
mean,
there is no reason. If genome sequence is perfect, you will get SNP in each and every region.
okay. So when we are using chip, so does it mean that if one SNP is at a particular location, another will be considered only after 400 base pair apart? Is it like that?
Yes, yes.
Okay.
So they keep gap actually.
Okay.
So because
400.
signal interference is biggest problem when they do high density genotyping. So
Okay.
signal interference, to avoid that's the signal interference, they keep this condition. Once you will design, you will get those ideas that what FE chip has problems.
Okay, sir. Sir, another question that I want to ask is that usually when we identify some gene, so we try to validate them in the heterologous system also.
Yeah.
So can you please explain what is that and why it is necessary to check them in the heterologous?
Yes, because that is the reason is that once you say that my NAC is or my transcription factor is binding with this gene and this gene is getting overexpressed. Okay. But there might be some other transcription factors which you have not isolated from this plant system, might be binding there and maybe helping co-expression together. So just to avoid that confusion, you will have to go, because when I choose tobacco system, it was totally alien. So no transcription factor which was present in case of endographite is supposed to be there also. Okay, but pathway more or less operates in every plant. Gene is constant, so operates same way. So once you go in heterologous system and you make your construct and make it transient express, it will express for certain period and then get switch off. Okay, so when you do this kind of study and during that period, if suppose the gene which you are saying that is getting overexpressed, the simultaneously that gene is also getting overexpressed, it means you are directly establishing that okay, this gene is the gene which I am saying is controlling function of this gene is actually getting established. So once you say that this transcription factor or this gene is controlling production of this, you will have to go in some alien system. Yeast to hybrid system is best system because yeast is totally eukaryotic, but it's like prokaryote. So if you go in yeast system, that will be the best establishment of your this findings. Okay, so once you say that in our plant this is doing okay, there are so many things operating in that plant. So to get clear cut this thing idea that okay, this gene is getting governed by this factor, you will have to go in some other system where this system is not known. That's why we go for heterologous system.
So can it be done in silico also?
No one will believe in silico. Because ultimately for trait will be utilized by breeder or some other biotechnologies for genome editing or for doing marker assisted transfer. So it should be validated in working life system. In silico won't work.
Okay, sir. So these are my two questions from you, sir. I will once again check if there is any more questions in the chat box. Dr. Devraj has asked that should we go for a single trait or multiple traits in Jivas? Which one is better? I think that's what.
Both are equally good because this Jivas study is only 5-6 years old and not much has been done. So we can go. Probably he might be asking about GLM and MLM or trait. He's asking.
Okay.
MLM, GLM method he's asking, or he's asking about trait.
If he is here, he can ask, sir, what does he mean?
Otherwise,
Sir,
trait. If he's asking for trait, he can go either way. It hardly affects.
okay. So, should we take up one more question, sir?
Yes, why not?
Okay. Dr. J.M. Nijagundi, sir, for Jivas among SNP chip-based genotyping and whole genome sequencing, which one is more reliable and robust?
GPS, probably he's asking about he or she is asking about GPS. Actually, chip and GPS is always debatable. The both has some advantage and disadvantages. Okay, so if you are doing chip based and you are using germplasm, where population is germplasm, where you don't have control about recombination and you don't have history. For that purpose, chip is better because you are fixing your SNPs or point of recombinations, and you are testing all population across that point. So that is better approach for doing GWAS study. Okay, and second advantage of chip is that once chip is developed, it is immortal, fixed. So it can be utilized by other research groups, and across research groups, you can compare their results also. Consistency can be checked very easily. In GBS, every time when you generate library, there is library, every time new library gets generated, it generates different genome region information. So sometimes whatever SNP one group is reporting might be getting not reported in other research group. So this problem sometime in GBS comes when we do genotyping by sequencing. That creates some problem. So if you are working on germplasm and this kind of thing, I will suggest that you go for a chip development, because your chip will become immortal, your SNPs will be fixed, and it can be utilized by other research group, and you can compare your results with them also for any trait. That is the advantage.
Okay, other questions we can take up in the mail for sir, because our next speaker has also joined. So, thank you very much,
Thank you
sir,
so much,
for
sir.
nice, informative, and excellent lecture, sir. So it was very much informative, and certainly our participants might have enjoyed all the lecture contents. And
if they have further any question, they can mail me also. I
yes,
will
sir,
be
we will be
happy to,
circulating the mail and contact
yes,
number.
I will be
to
happy
them.
to answer their queries.
Yeah. So they can ask anytime to you. So thank you very much, sir.
Okay,
Sir.
thanks.
Thank you, sir. Thank you so much. So our next speaker is with us. Sir, are you there? I hope.
Yeah.
Yeah. So our next speaker is Dr. Anti Kumar from IARI. So I will introduce our next speaker. Yeah, let me share the screen. So we have with us Dr. Anti Kumar from IARI Division of Plant Pathology. He is principal scientist, and sir has a very vast experience of working in the field of plant pathology. So starting from his graduation, he has done graduation from TNAU Madurai, then his master's in plant pathology from TNAU Coimbatore, PhD plant pathology from IARI. Then he got the DBT fellowship in the laboratory of phytopathology and visited Washington University. And his research qualifications include over 25 years of research experience in the field of plant pathology with a special training on microbial genomes. He is quite conversant with various advanced molecular techniques, genomics, and bioinformatics tools. And his other interest areas include non-host resistance in rice against biotrops, use of genomic tools to understand the plant microbe interaction, whole genome sequencing of the plant pathogens, plant microbiome, use of microbial elicitors, etc. Then sir is presently working in exploring and exploiting the philosphere microbiome for eco-friendly management of the crop diseases with a special emphasis on rice plus rice philosphere system. And currently he is involved in many externally funded projects too. Sir has completed till now 14 projects, and also assisting as a collaborator, and editorial assignments are also being handled by him for various organizations. And sir has editorial responsibilities for Indian phytopathology and BMC microbiology. Then he has got so many fellowships, awards, and recognitions, such as ICR Junior Research Fellowship, then CSIR Research Fellowship, DBT Overseas Fellowship for visiting the Washington University, DST Travel Grant for visiting Rhodes Islands. So many are there. Then he has made a vast contribution through his research, and gene bank submissions have been done by him. Then he has also filed patents entitled Bacterial Fermentation Technology for Production of High Quality Odor-Free White Pepper from Mature Green Pepper. Then again, he has so many high impact publications in the Journal of Fungi, so many international national journals. So, sir, I'm sorry that I could not read everything, but you have such vast experience that that we will be utilizing today for our audience and enhancing our knowledge also. So with this, I request you to please share your knowledge and expertise in your area. Thank you so much, sir, for being with us.
Yeah, thank you, organizers. Am I audible to all of you?
Yes, sir.
Okay, it's pity that we have to do this program online, and we have been doing it for last 3 years. Not only this kind of training, but also the courses what we are handling for students. And one thing I really understood, only few of the online participants, for various reasons, they really pay attention to the entire content. I hope this audience is different, and I sincerely wishing for that. So my talk is entirely on how best we can utilize the PGR for making that material useful in agriculture. So from that perspective, the topic what you assigned to me is very, very important, very relevant. So ultimately it comes down to PGR, the plant genetic resource. Whatever breeders they do now, the starting material is PGR. So what I intend to convey to all of you is some of my thought process. I have been associated with the screening program during my early career, and I really understood the ins and out of the screening program, the benefits and also the limitations. So I would be sharing those knowledge, whatever I gained over the period of my experience. Here, I would be putting some of my thought process also with some of the classical examples, the nationally and internationally planned disease, how people are doing the screening program to narrow down to that material that can be pumped into the breeding program or directly into the farmer's field as a resistant material. So this is what I have in my mind. We have been always receiving a query from the breeders, the choice of pathogen. I think we can enter into some kind of discussion towards the end of my presentation. The choice, what should be the pathogen a breeder should be using for screening program? So this is question number one. And question number two, the scoring method. What makes, how can we call a resistant plant resistant? And what are all the data points that gives us confidence to call them as a resistant material? So these two are some of the pressing questions. And during my presentation, I will be highlighting that. So with that brief note, while thanking the audience, authors, the organizers of this program for inviting me, let me, with your permission, let me share my presentation. It's not a very big presentation. Just give me a minute.
Yes, sir, sure.
Okay, here we go. I hope the slides are on the screen.
Yes, sir, they are.
Okay, please confirm whether it is in the presentation mode. No.
Yet to come,
No, sir.
No, you please confirm it's yes or no, ma'am.
it's
No,
still
It is
okay.
no, sir,
sir.
no.
No. Yeah, I know that. Yeah, great. Thank you. I'll share it again. Yeah, I think it should be all right now. Is it on the screen?
Yes,
Yes,
sir.
sir, now it's perfect.
Okay, great. Thank you so much. So thanks for the introducer for generous introduction. Thank you. The title of this presentation is Screening Plant Genetic Resources for Disease Resistance. I already highlighted the importance of PGR. I think various other speakers might be talking about it. Come to disease resistance. This is one of the cheapest method of plant disease management. So it doesn't cost anything if you have the material in your hand. You have to just supply the material to the farming community. They will grow and they will reap the benefit of that. So by deploying a resistant material, you can eliminate so many other hassles of plant disease management. The most important point is the chemical methods of plant disease management. So that really a big benefit, not only to the grower, not only to the farmers, but also to the environment. So we all know that chemicals are polluting the environment. So here comes the importance of resistant germplasm in agriculture. But before we touch upon the actual flow of the lecture, these are all some of my thought process. The fact that plants do have resistance against the pathogen, that is primarily guided by several factors. The factor which is genetically coded or the one that is thoroughly exploited in breeding programs. So I think most of you might have heard about the broad methods of or the broad categories of resistance in the plant. I think the horizontal resistance and vertical resistance. And we all know that the vertical resistance is something which can be manipulatable, amenable for breeding program. But the horizontal resistance, it is not that easy to exploit in agriculture. And the breeding program in agriculture is entirely dependent on, if not entirely, primarily dependent on the vertical resistance. We'll be having some discussion on that, where the vertical resistance scores and where the horizontal resistance scores and where the horizontal resistance fails. And what are all the modern concepts of these two forms of resistance, that also we'll have some discussion. Just have a reading of the statement, what I put on the screen. Plants have genetically imprinted mechanism to resist disease caused by pathogens. This is something which is evolutionarily associated, because ever since the angiosperm started emerging on the planet, that happened around 400 million years ago, the microbes were predated those angiosperms. So that means the plants do interact with microbes ever since they started appearing on the biosphere. So during the course of evolution, if not all, certain plant material or certain germplasm, they developed or they acquired the so-called genetic skills to stop the microbial pathogens from exploiting them. And whatever we are experiencing now in the form of vertical resistance or even horizontal resistance, you can trace them to the evolution, right? So if that is true, the fundamental mechanisms that is operating in the plant that governs the resistance, that includes the physical and chemical barrier. This is something often ignored in our area, but let us not forget the plants always have certain physical barriers and chemical barriers. They may have a dual function, if not the multiple function, right? So for example, thick cuticle, the plants might have evolved to conserve the so-called the moisture. So the same thick cuticle can prevent a strong pathogen from entering into the host, and so on and so forth. There are several such things you can see in a plant. And come to the chemical defense, that is something, it is always there in the plant. The so-called preformed and induced chemicals. Induced chemical means the chemical which is induced in the plant system, mostly the secondary metabolites. They are a potential antimicrobials, and they are toxins, and they can really stop the pathogen from making further inroads in the plant system. So the physical and chemical barrier is something evolved in the plant right from the beginning of their life. So two other forms of resistance, or rather three more other forms of resistance. One is induced systemic resistance. Here I segregated the chemical defense from the induced systemic resistance, just to make the second one as a secondary metabolite, and the induced systemic resistance something connected with the induced proteins. So these induced proteins, by and large, they can activate the defense in the distant plant part so that the plants are not suffering too much from the plant pathogens and the disease. So this is what is called ISR. This ISR is primarily induced by proteins. If you ask me who is inducing those proteins, it is the microbes or microbiome that is colonizing the plant in the rhizosphere. They are the potential trigger for those proteins. And these proteins are, excuse me. Excuse me. Mamta Singh, can you sensitize some of the... Okay, let me proceed. Induced systemic resistance is something which is happening in plant, and every plant at every time. So this is something typically microbiome induced one. We have two other forms of resistance, and you can, here comes the modern definition of vertical and horizontal resistance. So let's take the first one, the pathogen associated molecular patterns. So this is nothing but the horizontal resistance. So we all know that the horizontal resistance is something polygenic in nature. There are several plant genes are involved in governing the resistance. So these genes, the plant genes, they code for a kind of a protein. Those proteins make up a receptor, what is called as pattern recognition receptors, and these receptors are anchored in the cytoplasmic membrane, and they interact with the pathogen associated molecular patterns. So this form of resistance is what is called as pattern triggered immunity, or PTI. And this is nothing but what we have been taught in our early education, that is horizontal resistance. So this form of resistance is often quite strong, but the only issue here is, since too many genes are involved, polygenic in nature, sometimes it becomes difficult for the breeders to transfer all those genes into the new genotype. So the last one in the category is resistant genes. This is nothing but the vertical resistance contributing plant factor. And this is typically a plant protein. We call them as a cytoplasmic receptor or intracellular receptor. And these receptors can see the specialized microbes or the adapted pathogens. They interact with effector molecules and trigger a very strong form of immunity in the plant system. And this form of immunity is called effector triggered immunity. And the same thing what we talked in the previous years as vertical resistance. So the take-home message from this particular slide is the horizontal resistance the modern terminology for that particular form of resistance is PTI, and the vertical resistance is actually the effector triggered immunity. So between these two, the plant breeders, they exploit the resistant genes or effector triggered immunity, or the so-called ETI, thoroughly in the plant breeding programs. So that was some of the updates. I thought I'll share it with you. Having said that, since to do all those things, we need to identify the donor, we need to identify the resistant material. If that resistant material is having all relevant agronomic traits, you can straight away use them as a kind of cultivar or variety, whatever you want to call. If it is not having certain traits, or it is having a selective traits, then it is subjected to the so-called selective breeding program, or even genetic engineering approaches, right? So here comes how to segregate the one which is having resistance from the rest of the population. Certainly, we need to have a strong screening program or strong filtering mechanisms. So this particular approach is developed in the last 30, 40 years, if not 100 years. And certainly this involves a population of germplasm and also the pathogen on which somebody is working, right? So I'll take a snapshot of different methods people use for screening crops for disease resistance. Of course, the top two people normally use, all the time they use, of course, the field trial is standing in number one. That means growing the plant in endemic place for that particular disease, or you create a kind of sick plots in your farm, and you grow the material and try to see which one is surviving and which one is not. And surviving plants are further scrutinized into different kind of mechanisms, and you come to a conclusion that this is the material which is having resistance against the so-called pathogen. So field trials are very effective, and if provided the place where you are screening is known for the so-called disease, the endemic place. And the second comes the greenhouse trials. Very often the institutes are located in a place where you don't see the disease coming in the farm, and certainly you need a different kind of mechanisms to do that. Here comes the greenhouse, controlled greenhouse trials, or polyhouse trials, or whatever name you want to call. They are integrated in many screening programs now. The plants are challenged with the pathogen of the choice. And here comes the choice of the pathogen, the race that is being used, everything matters here. So you grow the plants in the controlled environment, and you challenge it with the pathogen and see how many of them are going to survive. And you make some kind of calculations and statistical approaches and come out with a material which is going to resist, which is going to be called as a resistant material. Two other methods, somewhat popular, but not too much. The in vitro, the tissues are grown in the laboratory conditions and challenged with the pathogen to evaluate the resistance. This became very popular when the tissue culture methods of crop improvement gained popularity in 25 years back. So for some pathogens, it is still used, but for several pathogens, considering the cost consideration and the hassles involved, people automatically they go for the previous two methods, that is the field trial and greenhouse trials. The recent addition in the whole bucket list is the so-called molecular markers. So here we use different kind of technologies. Of course, people, the DNA markers are often used to identify the plants with the resistant gene. And this is something like a rapid screening of germplasm for the particular resistant genes. And thereby you can shortlist the material, and the shortlisted material again subjected to one of the three methods, what I just now told, either the field screening, greenhouse trials, and in vitro test. So I would say that not single method is good enough. There is always a combination of method, so that your conclusions are not going wrong, and it is accepted by the larger audience when it goes for the breeding program or eventually into the farmer's field. So these are all the methods what normally we use, the general methods. Some of the considerations I want to really tell here. The question is, I'm going to narrate in the coming slides how many different kind of methods are there, different kind of scoring options are there, and how to call a material a resistant. Different considerations are there. But before that, the choice of the method what you are trying to deploy in your program, it largely depends on the pathogen. The pathogen would decide what kind of screening methodology you have to adopt and what kind of scoring method you have to adopt. So entirely dependent on the pathogen. As the flow goes, I'll tell what pathogen needs what kind of screening method and what kind of scorecard that we'll discuss. So pathogen, target crop, and of course the environment. These things are very, very important, right? And the screening for disease resistance is an ongoing process. There is no end to it. I make this statement here. Why there is no end to it? Because we all know that there is something like a boom and bust is always operating in pathosystems, and breeders develop a resistant variety. A pathogen do the breeding on its own in the environment. It comes out with the different capabilities. What we call it as a new pathogen emergence, or the existing pathogen can evolve new strategies to defeat the so-called resistance. So if that is something which is reality, and certainly resistant breeding program in every crop or every pathosystem, it is an ongoing process and there is no end to it. Right. I just wanted to remind you that the UG99 story, those who are working on rust must be knowing that, and everyone believed that a particular type of wheat cultivar having a SAR31 gene is permanent resistant, but it is not. In 1999, that particular cultivar succumbed to a disease in Uganda in Africa. Then the new pathogen emerged that made a kind of news in agricultural, especially in the wheat breeding programs. So what I mean to tell here is pathogen race evolution is something which is happening automatically in nature, right? We cannot stop it. And of course, by incorporating the disease resistance in the breeding program, here comes the importance of PGR comes. Once the PGR with the resistance is identified, and if it is having some kind of lacna, then we need to use them in the breeding program so that the traits or the gene, what is governing the resistance, can be transferred into the agronomically important material through plant breeder. Of course, plant breeder alone cannot do it. They need the assistance of plant pathologist, and together you can make the crop more resilient and less reliant on the chemicals, leading to a more sustainable and productive agriculture. So this is typically a kind of a collaborative effort between the breeding as well as plant pathology team. And here comes the scoring method. So remember, for the audience who are not well versed with the plant pathology, there are two kinds of the entire pathogen group you can put under two groups. One is the foliar pathogens and another one is the soil-borne pathogens. And we all know that foliar pathogens, they rarely kill the plant, but soil-borne pathogens, they can kill the plant. So this is thoroughly exploited in the scoring methods, right? We'll come to that point. We'll have a lot of examples in my slides. The methods used to quantify the severity of the disease symptoms in the plant and to assess their response to the challenges, disease challenges. The disease scoring method is something very, very tricky, and sometimes people call it as a kind of very cumbersome method, but it is not that cumbersome. And let us, as we progress, you'll come to know why I'm making this statement. And the most popular, the scoring method, right? The visual rating. I think all of you must be knowing, seeing is believing, right? So this method involves the subjective evaluation of the material entirely based on the symptoms, right? The visual appearances, and people try to score the percentage of the leaf area which is affected, right? There are scorecards, there are matching images, and they will compare it with that and try to give a score. In other terms, the images are converted into some numerical figures, right? So the visual rating is all about that one, that you are going to give a rating based on the symptoms, right? And index scale, a method uses a numerical scale to rate disease symptoms, right? And with the higher number is always a severe disease, and number closer to 0 is approaching immunity or resistance. This is what is called index scale. And you have disease progress curve, This is very simple. A method plots the severity of the disease symptoms over time. It allows for a quantitative assessment of the rate of disease development. This is what is called as disease progress curve. The next one is very popular, AUDPC. It is nothing but the previous one only, but the only thing is over the period of entire crop growing season, if you are doing that, then it takes a different name, that is area under disease progress curve. This method calculates the area under disease progress curve, providing a quantitative measure of disease severity over the entire growing season of that particular plant, right? And the fifth one is a DNA-based method. This is a new addition in the whole scoring. Here, people use a very rapid and high throughput method, for example, qPCRs or even sequencing, to quantify the pathogen's genetic material in the plant tissue and to determine the extent of infection. This is something to do with, how much a pathogen load is there in the plant material. And if that load, a susceptible check is succumbing to the disease and the resistant material is still a resistant, then that gives you a kind of a robust answer, whether it is a disease escape or it is a genuinely disease resistant. So this is something very, very important. And in the, for example, this is one of the questions people normally ask. The resistance is really a resistant or it is a matter of escape. So how to rule out? Very simple. You just see whether the material is still having the pathogen in it. If the pathogen is there and the plant is not showing up any symptoms, then you can call it as a resistant. And there may be a scenario, the plant is not showing up any disease, but it may be the case like the pathogen is not there. So this is typically a kind of paradox when people are doing this kind of screening in the open field trials. So here comes the DNA method. It can very nicely complement the breeder's decision or pathologist's decision whether the material is really a disease resistant or disease escape. So here, once again, I must emphasize that the commonly used disease scale, again, not one method is adopted here. It is always a combination of methods. The last method can very well be integrated into other 4 programs, whatever I just highlighted on the screen, right? With that, again, some more considerations. Here comes the importance of statistical analysis. Every screening method, whatever we are doing, the results are always subjected to a statistical analysis to compare the resistant capability of the breeding line in controlling the disease. And here comes the check. The check is always called the susceptibility check or susceptible check. And if there is an option, you can still have another check, which is a well-known material, which is giving resistance, but it is not having the desired agronomic trait. So at least the susceptible check must be integrated in the disease screening program just to confirm or to gain confidence over the methodology, whatever we adopted. So susceptible check assumes an importance in this program. Of course, I already told you the choice of disease scoring method. It depends upon the pathogen, target crop, and also the resource available. I made a point already. Resource availability is also an important point, whether to integrate a couple of methods or 3-4 methods so that you make a very robust conclusion rather than a single data point and jumping into some conclusion. So it all depends upon how much resources you have. So if you have a kind of a climate controlled greenhouse where you can regulate the environmental factors for creating the disease, and you can very well, the field screening, the shortlisted material from the field screening can get into that in plant screening in the greenhouse under a pathogen inoculated trial, so that you are very sure about the material which is having the genuine resistance against the disease. So this is, again, depends upon the resources. And however, the ultimate goal of the entire program is to provide a reliable and accurate assessment of disease severity that can guide the breeding decisions, right? With that background, I'm just going to take you to the tour of some of the globally important plant diseases or plant pathogens, if you want to call, and how the scoring methods are adapted for those pathogens, right? So let's take one by one. I have to really convey to you that I have only considered 3 broader groups of plant pathogens. They are very important in agriculture. I excluded the viruses here. The reason being, the scoring methods for plant viruses are still evolving, and very often the screening program is not what do you call for many viruses, the screening methods are not robustly concluded. So certainly for fungal pathogens, oomycetes pathogens and bacterial pathogens, we have a thumping success in agriculture, not only in our country, the rest of the world as well. And some of the disease I am going to tell here, you can very easily relate maybe in your studies. So let's take the first one, fungal diseases. I'll get back to one of my previous slides. I talked about vertical resistance, right? This is one of the questions people normally ask. Why breeding program is very successful in fungal pathogens most often, right? So there is an answer. The answer goes back to the so-called gene for gene hypothesis that was proposed sometime in 1940s, 1945 period. So here, the hypothesis is that there is a gene for gene interaction between the pathogen and the host, and the pathogen in question there is a fungal pathogen, that too a biotrophic pathogens. So that was the foundation for the so-called plant breeding program leading to resistance, right? So there is a gene for gene interaction is always happening, especially with the biotrophic fungal pathogens. Biotrophic pathogen means the pathogen which needs the live host to complete their life cycle, right? You can think of rust, you can think of powdery mildews, you can think of downy mildews, all those pathogens, right? They need a live host. You cannot culture them in the dead material. They don't survive in the soil in the absence of their host. So these pathogens, they enjoy the so-called gene for gene interactions. You may ask, what is a pathogen gene and what is the host gene they interact? It's quite clear. I already talked about it. It is the pathogen gene codes for pathogen factor, pathogenic factor. Those factors are called effectors. These effectors are interacting with, or the plants have developed a mechanism to visualize, or I would say to interact with the, physically interact with the pathogen effector. And those plant proteins are called resistance proteins or cytoplasmic receptors. So this is thoroughly documented in fungal pathogens or fungal pathosystems. And let's take some of the examples. I put number one disease here is rice blast. And we all know that resistant breeding program is complemented the rice breeding program. Rather, it really made a big contributions. I think over 100 plus resistant genes have been documented in rice germplasm, and substantial part of those germplasm is the wild cultivars and wild relatives. And I think you can think of PA51, the classical one, and there are beta, there are several examples, right? I'm not going to highlight the importance of this disease. This is number one fungal pathogens in the world, can create havoc in any rice growing areas. And this is the scoring method. I just put it on the screen, right? So this scoring method was developed in 1992 by Mackil and Boneman. This is 0 to 5 scale, right? I already told the higher number indicates very serious disease, and lower number closer to 0 indicates there is no infection, right? If you are getting a score 0, and you can very well call that material as a resistant material, if not immune. And as the number goes, you can score them as a susceptible material. Ultimately, what you are doing here is the extent of lesions, what you are seeing on the leaf is converting into some kind of numerical figures. So for that, some guidelines people have developed. Those guidelines are put here. You can see that, right? So at end of the day, you are going to calculate the so-called PDI. PDI is nothing but percent disease index, right? So here there is a interpretation just to guide the breeders. If you are getting a score 0 to 3, you may call that material as a resistant. If it is above 3, that means 4 and 5, that is being rated as susceptible, right? That means it may not go to the next round of screening or next round of activities, right? So the desired number here is 0 to 3, right? And this is what the PDI, how it should be calculated. Sum of numerical ratings divided by total number of leaves per plant observed into maximum score into 100. So I am not going to further explain here how to do that. Please explore different literatures. I'm just giving a flavor of how scoring can be done for a disease like rice blast, right? I'll go to the next disease. Of course, we do have this disease in our country, but this is one of the internationally well-known disease, that is botrytis cinerea in different fruits. And the previous disease, I forgot to tell you, you have to make the plant and pathogen interacting, and the pathogen is delivered in the form of the infective probacule. Remember, the infective probacule is something which varies from pathogen to pathogen. So for some pathogen, the spore, or we may call it as a conidia, for example, in the case of Magnapurti, only the spore alone can initiate the disease, right? So please remember this point. Many times people think any part of the fungal body can be used as an inoculum. Certainly not. For example, in the case of rice blast, you cannot use the mycelium, because the mycelium or the hypha, I think, please recall your younger studies, hypha, the body of the fungus, or the mycelium, the mass, they don't infect. It is only the conidia that infects, right? The conidia germinates upon arrival on the leaf, and upon germination, it forms a germ tube. The germ tube forms a kind of aprosorium. Aprosorium is what we call in a simple language is a launching pad. From the launching pad, the infection pig comes vertically towards the epidermal layer of the plant, and it enters into the leaf, and it colonizes the mesophilic cells, and it starts working intracellularly. And of course, it can produce some haustoria to take the nourishment from the plant, and the plant react to it very badly, and the symptoms are created. So remember, probacule is something very important, and the probacule for this disease is conidia, right? For certain pathogens, you can use mycelia also. And for example, the necrotropes. I think you should remember necrotropes are something typically soil-borne. Think of a pathogen like Rhizoctonia solani, Rhizoctonia betaticola, Sclerotium ralphsi. All those pathogens, they are soil-borne. You can still supply the mycelium as an inoculum. But if a pathogen which is adapted on the foliar part, always remember the conidia is infective probacule, right? The conidia is needed, and there is a threshold level of conidia is also needed to cause the disease, even on the susceptible germplasm. People have worked out it should be at least a million conidia should be there in per ml of water what you are spraying. Remember this point. If any number less than a million, there may be a failure and infection even on the susceptible material, then the whole screening program may go wrong, right? So conidia is the material in this particular case. And for this pathogen also, again, the conidia takes the center stage here. You take the conidia, make a suspension of the conidia. Here it is 10 to the power 5 to 10 to the power 6. And you just either inoculate on the whole fruit, or you go for a spot inoculation or detached leaf inoculation, and subjected to a condition where the disease can come. And this is how the disease is scored here. And the scoring here is unlike the previous pathogen. Here it is 0 to 3 score. 0 is no infection, and 3 is a very serious infection. And the pictorial, it is depicted something like this. You don't see any kind of the browning here. That means it is completely immune to that particular pathogen. As the disease, the different kind of score, it is going to be called as resistant to susceptibility. For example, 0 tone is resistant, and 2 to 3 is susceptible, right? We'll move on to the next disease. That is rust, right? I think I'm sure some of you must be handling this pathogen here. And again, the inoculum is the iridospores, and these iridospores are inoculated by dusting on the plant part and subjected to a kind of conditions where the disease can come, and you start scoring. And the rust scoring is always very tricky. Too narrow images or the infections are scored. Let's see what it is. This is how it is scored, right? We call it as ITs. IT means infection types, and it gets the score from IT0, IT0 FLUC. Second one is IT0 FLUC. Please remember this word, IT0 FLUC. And you have IT1 to IT4, and there is one more is added, that is ITX. So every score has got its own meaning. And IT0 and IT0 FLUC is immune. IT0 is completely immune. You see a green plant leaf, and you get ITS0 is, of course, very subtle HR. HR stands for hypersensitive response. And very, very subtle. You may not be able to see through naked eye, but if you see against light or something, you'll see inside the plant leaf, and you get a score of IT 0 flag. Then 1, 2, 3, 4, it depends upon the quantum of the lesions, quantum of the iridia, what you are experiencing on the leaf surface. And ITX is something very important, that is the random distribution. There is no uniform pattern on the leaf, right? This is how the rust scoring is happened. This is an example for the leaf rust, Puccinia tritisica, right? And we'll move on to, so far what we discussed is something foliar pathogens, which is affecting leaf and fruits. Now we'll move on to another 2 examples. They are affecting the plant from the rhizosphere or the root. Fusariums. All of you know that fusariums are responsible for wilt, what is called fungal wilt. It is a soil-borne pathogen, and this is a very trickiest disease. And the scoring has been always debated, and there is 2 arguments are going on. Let us have some examples, some points to discuss here. And again, the conidia, fusarium forms a different kind of conidia. The macro, micro, sometimes chlamydospores. Sometimes they form mesoconidia also. So primarily, the easiest one to multiply in the lab is the macro and microconidia. You can very easily harvest them. Just put it in the rhizosphere of the plant and wait for the symptoms to appear and compare it with the so-called susceptible cultivar and try to score them. This is the scoring. This scoring, there is some people accept this score, and some group of people, they don't accept it. Why they don't accept? And the reason being, either the plant wilt or it doesn't wilt. There is nothing in between. So this is one school of argument. And again, but there must be some criteria to rate the germplasm. This is somewhat accepted by many people. And there is a group they are opposing also. They say either it is resistant or susceptible, right? So all in between is what is called as the half-baked infection, right? So this still can be debated. And in my opinion, you can include the pathogen load as one of the criteria here to rate them right from 0, 1, 3, 5, 7, and 9, right? In the presence of pathogen, if the host is still able to resist, it is not wilting, then you can very well call it as a resistant material, right? And in a population, if certain number of plants are still wilting, and the question is why the rest of the plants are not wilting, whether it is a case of disease escape or it is genuinely resistant or there is a pathogen load differences. So I always have a feeling like if you integrate pathogen load here, we can very well revise this particular score, right? And this is a formula. Percentage disease incidence is a sum of numerical rating. That's the same formula what I already told. And the scoring is quite easy because it is a soil-borne pathogen. In a population of 100 plants, how many of them are still not wilting? How many of them are wilting? That score can be counted in the field, and you can derive at that particular percentage wilting, right? So this is about fusarium. In the case of vegetables, the same principle is even for the so-called banana. This is called the Panama wilt of banana, one of the #1 disease. And in our country also, I think those who are listening to that, TR4, that is tropical race 4, is making an issue in banana in many places, especially in Bihar and other places. And resistant breeding program follows the typically what I already talked about in the previous slide. There is a 0 to 4 score and try to estimate how many plants are succumbing to the inoculation or the natural endemic area and try to call them as a resistant or susceptible material, right? And another disease, again in banana, that is a foliar pathogen. I think it has slipped here. This is again the same story like rice blast. You have a kind of you just estimate how much of area of the leaf is affected by this particular disease and give a score and come to a kind of rating. So far, what we discussed is foliar pathogens, which is affecting a foliar fungal pathogen, which is affecting the plant, and also soil-borne pathogen, which is affecting the plant. Now we will move to another group of pathogens, very important in agriculture, oomycetes. So those who are not too familiar, remember 25 years back, the oomycetes and fungi were put under the same group of fungi, and later the tools, the molecular tools and the new knowledge about the taxonomy has segregated these two from each other. oomycetes are completely different group of inditii, and they deserve a different classification system, and they define different status in the classification, right? So remember, Phytophthora infestans is one of the oomycetes pathogens, a very important problem in potato and tomato. And screening program has been quite successful in these two crops, especially in potatoes. And again, much like the rust disease, we have a score 0 to 9 scale. And this 0 to 9 scale, again, the 0 is somewhat called as immune, and the 9th one is what is called as extremely susceptible. And in between, what is happening is the different grades, right? And the next one is downy mildews, and it's a problem in many crops. And what I brought here is only the vegetable cooker pits. And here again, the story is same. You have a scale 0 to 3, and the 0 is again immune, and higher scaling is what is called as highly susceptible. These two pathogens, remember, they produce sporangia, and the sporangia is harvested from the plate, petty plate, and you again reintroduce in the soil and try to create the disease condition. Here, this is a Phytophthora is a kind of hemibiotroph, but the downy mildew pathogen is typically biotroph. It won't work the same way as the Phytophthora. You have to do different mechanisms, and people have developed a different strategy for downy mildew screening. The powdery lesions are collected, and the conidia is harvested, and because you cannot culture them, the harvested conidia are used as an inoculum for either by spray method and just go for high throughput screening in the laboratory, right? Now we'll move on to bacterial disease of croplands. Breeding program has been very successful, at least in rice. I think all of you might have heard about XC21, a success story in rice. And the gene has been cloned from the wild relative of rice, that is Oryza, calling that it is one of the wild relatives. If I recall, I'll tell again. So that particular XC21 gene is being thoroughly exploited in rice breeding, rice bacterial blight management. So yeah, I think that is coming next, and I'll come to that. Yeah, we come to Ralston later. And this is a leaf clip method, very successful because this pathogen is a vascular pathogen. So people have developed a very simple technique. You culture this pathogen and make an inoculum in a beaker. Use a scissors to clip the leaf tip and introduce the pathogen in the vascular part. Since it's a vascular pathogen, the leaf clipping is good enough to deliver the pathogen right in the vascular. There it can multiply and it can cause disease. This is a scoring method, what is adopted here based on the length of the so-called lesion, right? If the length is 0 to 5 centimeter, remember, it is called resistant. That means even up to 5 centimeter of a lesion on the adult plant leaf is considered as resistant, right? And if it is 5 to 10 centimeter, moderately resistant, 10 to 15 centimeter, moderately susceptible, and it is more than 15 centimeter, it is susceptible. And this score has been very successful in rating rice germplasm for bacterial blight resistant screening program. We'll move on to another important pathogen. Is a serious problem in vegetables and to some extent in spice crops like ginger, that is a bacterial wilt. It's much similar to the so-called fungal wilt. Here the symptoms are almost same, but the only thing is the pathogen is a bacteria here, and very easy to inoculate. You make a kind of bacterial suspension and release the bacterial suspension in the rhizosphere, much like what you did in the case of fusariums, and try to create a disease condition. Keep a susceptible cultivar and compare the susceptible cultivar with the rest of the plants. Again, the story is same. How to score them? Quite simple. You just count the number of plants that is expressing the wilting symptom among the total number of plants, multiply by 100. That gives you percentage disease incidence in the field. Again, it is a debatable. There is something like disease tolerance is there in the case of bacterial wilt. The two schools of thought, again, now arguments are still going on. And one is There is no such thing like a tolerance to bacterial wilt. It is resistant or no resistance, or I would call susceptible. There is nothing like in between. If something is happening in between, it may be the case of disease escapes or below threshold level of pathogen that is unable to really complete the life cycle. So this debate is an ongoing debate in wilt research worldwide, right? I already talked about this rice bacterial blight. We'll move on to another important disease, what is called as Xanthomonas campestis, Bethovar campestis in cruciferous members, and also vesicatoria in several solanaceous vegetables, right? For both the pathogen, the foliar inoculation is done. You create artificial conditions for creating the disease. Or people sometimes they do even a secluded place where they can go for releasing the pathogen right in the field and creating the conditions for the disease. Of course, that need to be done very cautiously. It should be away from the farmer's field. It should be away from the hotspots of those plants. Then you can still do that. And the most preferred method is you go for in vitro or secluded trials in greenhouses, right? And this is scoring methods, foliar 0 to 9 scale. And you convert those images into a kind of numerical figures. Again, the lowest score is called susceptible, resistant, and the highest score is always susceptible and extremely susceptible conditions. And you start scoring them. So here 1, 2, 2 is called highly resistant, 3 is moderately resistant, and above 5 is susceptible. And another important, this is kind of Pseudomonas syringae in the case of vegetables. Not a very top-end disease in our country, but it's a problem elsewhere. The scoring method, there are different kind of methods are adopted here. People go for seed soaking so that the pathogen is delivered right in the seed, and the material is screened for resistance. Stem stabbing method, it is what is called as a tiny injury is created, a non-lethal injury is created, and the pathogen is delivered and creating the condition for disease to come, and you start going for the scoring. This is a scoring method. And different people have really adopted different kind of scoring methods, right? And 1 to 4 scale. And again, it is the same principle, whatever I already told you. And this is another important bacterial pathogen, Erwinia. It's a rot pathogen. I just deliberately brought it here because rots are an important problem in agriculture, especially in the case of post-harvest succulent vegetables and fruits. And screening program has been developed for fruit like vegetables like carrots, especially for the soft rot. And again, most of the rot causing pathogens are wound parasites or opportunistic pathogens. They need a kind of wound. Certainly the wound is integrated in the disease screening program. You create a tiny wound and try to introduce a pathogen there and create rotting symptoms. And this is scored something like this. And 0 to 4 scale, and the scales descriptions are given there. If the surface is not at all rot, you can give a score 0, that is resistant or immune. And the other score like 1, 2, 3, 4, 1 is nothing but 1 to 24% of the surface area decayed, 25 to 49%, that is score 2. You may ask how to convert that images into percentage area. Image analysis system is very handy nowadays. You can very easily score just by scanning those material into a kind of image analysis system, and you can calculate just based on the differential color what is being captured by the camera. So 25 to 49 is surface decayed, 15 to 74 is surface decayed, that is getting the score 3, and the 4 susceptible, that is 75% and above, is completely surface decayed. So this is for a soft rot, and the calculation part is done here. And again, another problem, we do have this problem in our country. This is Streptomyces scabies, the scab disease in potatoes. Again, this pathogen, has created problem in the hotspots of potato growing regions. What it does is it reduces the marketability of your potatoes. So certainly a huge loss. We need certain methods. And this is how the scale, it's an interesting kind of scale. Scale A, the percentage tuber area which is covered, 1 is 10%, 2 is 25%, 10 to 25%, and the score goes like that. And the 5 is susceptible and 1 is resistant. And again, another alternative score is also there. Lesion type 1, superficial scab, medium size scab, deeper it's something like something through the vertical enlargement of the disease on the screen on the skin. So this is about a potato scab, and this disease we may have in our country, but it never highlighted as an important problem in our country because we have much better disease to deal with. For example, powdery mildews, downy mildews, and of course the viral diseases, Xylella fastidiosa. This is a problem, and of course in temperate countries and elsewhere, this is a serious problem. And this scoring, I just thought of just conveying into you that for a disease like vascular, the xylem limited bacteria too, we have disease scoring methods people have developed. This is very important because when somebody can develop for a disease which is colonizing the vascular, the xylem part, especially the xylem limited bacterium, showing up the symptom of some sort of leaf shades of color changes on the leaf. They just converted that images into a figure, 0 to 7 figure, and up to 3 it is resistant and 4 to 7 is susceptible, right? So I can go on and on like this, but I just brought an example which is having a relevance for the country as well as at international level, if not all, at least few selected one, representing fungal, oomycetes, and bacterial pathogens. Now we will move on to another important aspect. Where do we stand in terms of sustainability of this particular method? I already made a point. It is ongoing method. There is no end to it. Let us have some discussion on that. We all know that the resistant breakdown is a serious issue in agriculture, especially the resistant breeding program. So in my perspective, if you ask me, resistant breeding program, although it has got its own plus points, you can prevent the chemical sprays, you don't depend on other methods, you can very well make agriculture very remunerative. But remember, to come to that stage of releasing a resistant variety, the efforts, the cumulative efforts, right from the day one, identifying the PGR, the day you are going to launch a material for resistance, it's an enormous effort. If you calculate the salaries, if you calculate the efforts, money, it's very expensive. There is no doubt about that. So end of the day, what you are bringing out is a method which is very cheap, right? So the question here is, pathogen does the same effort on its own. And there is a paper appeared a few years back. What happens in Magnaporthi when the breeders are developing a resistant type, a resistant cultivar against Magnaporthi? You know, the story what I learned was this resistance, whatever we develop, the longevity is 3 to 5 years only, especially in the case of Magnaporthi. That is an alarming situation. Why? Because within 3 years of a continuous cultivation of the so-called elite line of rice, which is having a blast resistance integrated, in addition to all other agronomic and trade-related traits, a pathogen make a selection and come out with a different capability and make your resistance nullified. So that is a threat in agriculture. I would say this is much similar to a threat what all of us are facing in pharma industry, what is called as a resistant building up in the case of pathogens against antibiotics. This AMR and the resistant breaking down, strikingly, they follow the same mechanisms, right? There is a mutation in the effector coding genes, and that mutation makes the effector not detected by the so-called resistant proteins, and the resistant protein cannot activate the so-called vertical resistance in the host and succumb to the disease. This is what is called as a loss of efficiency of the plant resistance mechanism, resulting in enhanced susceptibility of otherwise a pathogen what we thought it is resistant, a plant what we thought is resistant. Let us see the factors that contribute towards this resistant breaking down, right? So rice blast example I told, it took only 3 to 5 years. And the question comes whether the pathogen mutates before the launch of this variety, or it mutates during the course of interaction of that resistant variety and the pathogen population which is there in the environment. This is ongoing debate. Nobody can answer this question at this moment. But what we know right now, the large scale cultivation of that particular elite type, We may boast like 1 million acre is under a particular type, which is having resistance to this bacterial blight or the fungal blast. What happens is the selection is operating there in the open field, right? See, pathogen need to survive there. It comes out with its own mechanisms. I make one point here. Everything depends upon the population, what is being released in the environment, right? We release a rice cultivar in millions of acres. You know, a single lesion of Magnopurthy blast can generate in 7 days 30,000 conidia. This is an astronomical number. I'm talking about one lesion, right? In a particular plant, you just count the number of lesions and just the sporulating lesions, the sum total of those conidia is something which is very well mathematically amenable for the mutation frequency, right? Even if you put a mutation frequency of 1 to 10 to 9, you have a substantial number of pathogen is already having the mutation in it. And the only thing happening in the open field is we are selecting the so-called resistant breaking type, what we call a pathotype, which is having the capability to break the resistance. This is the typical boom and bust cycle, what we have been witnessing in several crops, at least in the case of rice, wheat, and of course, so many other crops as well, right? In potatoes also. So the point here is the factors. Let us see some couple of factors, which is what that guides that. Pathogen adaptation is the number one, right? See, remember, the pathogens, what we are talking in agriculture, they are all domesticated microbes because we domesticated the crop domesticated the pathogens, right? So if that is true, certainly this is an evolving situation, right? So whatever variety we develop, a pathogen is going to develop on its own, right? And ultimately, we are only selecting those resistant the pathogen which is capable of defeating the resistance, right? See, the pathogen adaptation, it can evolve a strategy to overcome the plant defenses, right? For example, altered effector capability, and this new effector that can overcome the resistant proteins or resistant protein functioning. This is well documented, and we have quite a lot of molecular evidences for that. And host adaptation, that is another thing. The plants can evolve new susceptibility factors that can make more vulnerable to the pathogen attack. This may sound very different for us, but this is something which is happening. For example, we all know that the susceptibility factor is, which is something very relevant for plant to be a plant, right? Nowadays, under the genome editing era, people are looking for the susceptibility factors now, right? The gene which is coding for susceptibility factors. Why? If you can edit that, you can make the plant resistant to that particular pathogen. But the risk, what is associated here is the susceptibility factor is something which is so important for the plant life itself. So that means if you meddle with that particular gene, probably the plant may get affected some of the very important traits, right? Maybe related to its own agronomic trait or something which is very vital for them to flower. We really cannot predict that. So the host adaptation can happen just by new susceptibility factors. Without our knowledge, we bring into the agricultural system. How? We want certain traits to be integrated in the cultivar. Maybe enhanced nutritional capability, nutritional factors, or one or other thing which we got to know that it has got a better market value, and we breed for that, and we end up making the plant otherwise resistant is susceptible. This is what is called as host adaptation. And environmental change is another very important factor. People know it for the last 30-40 years. A temperature can make a wilt-resistant plant into susceptible. That means a temperature-induced induced breaking down of the resistance. This is well known. And of course, moisture, light level can alter the plant pathogen interaction and result in the resistant breaking down. And loss of resistant gene, that is another important thing. Over the time, the resistant gene can become inactive or lost due to genetic drift or mutations. So this is it's also happening. And reduced expression of resistant genes for various reasons. These are all some of the factors which might make your plant susceptible to the pathogen again. And so that makes the breeding program a non-stop or a continuous, right? So with that, I would conclude. I thank all my students who support our research in IRI, mostly students, and of course the project staff. And I thank different funding agencies and different authorities who is guiding us for supporting our programs. And thanks for your time, and let us have some interaction before we close this lecture. Yeah.
Yeah, okay,
Can I
participants,
stop
the session is open for discussion now. Sir, there are some questions in chat box, so
Yeah,
by
can read.
Sasikant Patel. How to differentiate the freezing injury and fusarium head blight in wheat at time of flowering?
I'm not sure I can really qualify to answer this question because I have not seen the freezing injury happening in the wheat myself. So, but the confusion is not between freezing injury and fusarium head blight. The real confusion now is fusarium head blight and wheat blast. Right. So that is happening now. And I think there are tiny pink color spots are coming in the case of fusarium. That's what I understood from my colleagues. So, but there are finer subtle variations are there in the symptoms, right? Freezing injury and fusarium head blight, probably on the spot, we cannot say, but by culturing method, we can certainly tell because fusarium is something somebody can culture it very easily, right? So maybe visually, probably we may not be able to tell very confidently, but certainly through a culturing mechanism, maybe if somebody is giving 3-4 days time, we can certainly tell whether it is due to fusarium or freezing injury. Shashikant Patel, is it okay?
Okay, sir.
Yeah, thank you. Yeah. Kindly give an example for perennial tree plants. So probably we have to ask the Tree Breeding Institute how they are doing. So tree is something at screening can happen at seedling level for resistance breeding, so that that material can be promoted to the next level for large scale planting. But in big plants, adult plants, absolutely I cannot answer. So it's maybe you can answer, maybe some drone based technology can come into help, or some sampling strategy we can do. Maybe you collect the leaf, for example, pomegranate, we do that, right? If you consider pomegranate as a miniaturized tree. So what we do, we collect the random sampling, right? And the random sampling we collect and we score the disease, for example, bacterial blight of a pomegranate, we do that, right? And it comes beautifully well. Yeah, but unfortunately, we don't have a resistance.
Sir, this is myself, Sir Dhanavendan, sir. Because
Yeah,
sir, in tree germplasm, we are maintaining only minimum population. In case of cereal, we are maintaining large population, 100, like that we can easy.
What disease you are talking about?
Sir, that is a mulberry. I am screening sometimes that leaf spot and powder mildew. That powder mildew is a major problem for the now evolved varieties. That's why I am asking if such a kind of any resistant variety, because I have only 4 plants. So that
no. Yeah, Dhanavendan, excellent question. For screening for disease in the trees, it has to happen in the nursery only. For obvious reasons, because you cannot create an endemic area for this disease, right? Because you may say I can release a pathogen there and go for screening. That won't work. I'll take the example of black pepper. I happen to witness in a close quarters. So what we do, we don't release the pathogen in the open environment. It is prohibited actually. Yes. Right? We should not do that. So what the best method is, you take the juvenile plants. It may be a cutting or seed generated seedling. Create a population. Don't jump into the screening program in the very first year. First, you build a population, right? The population size can be anywhere between 100 to 1,000 seedlings. 100 to 1,000 seedling, anybody can.
For each accession, sir?
Of course, if there is a possibility, at least a decent number, which is amenable for statistical analysis. In your position, what I would do, I would keep a moderate number of 25 plants per accession. Correct. And you create a population. Suppose if you are handling 100 accession, and each accession you keep 25 plants. For your information, if you want to know more about that, in black pepper, this is done beautifully well. So what they do, they maintain a population of this black pepper cutting. It's a rooted cutting, right? They maintain a population of 25 population per accession. The entire greenhouse, right? Maybe 20 meters by 5 meters or 20 meters by 10 meters. It all depends upon you. And create a population there and go for a kind of pathogen spray if it is a foliar pathogen. And keep a susceptible check alongside. and create all the conditions for disease, and you can score it.
Okay.
So keeping only 4 plants, I don't think we can make a robust conclusion. That's what I'm coming to the point. Don't jump into the screening program the very first year. You first build a population. I'm sure the casualty will be there. That's a problem in tree crops. And even if 50% success is there, there's a thumping success, right? There will be heavy casualty. So you try to maintain all agronomic input so that not too much of casualty. And you release the pathogen. For foliar pathogen, go for spray. For soil-borne pathogen, you just go for soil inoculum. Very easy to do that. They just release the uniform quantity of pathogen, whatever may be the pathogen. I believe fusarium is a problem in some of the nursery crops. Maybe rhizoctonia may be the problem. You make inoculum, make a uniform dose, and give it to all the rhizosphere and wait for the disease to come. It's doable, right? And you don't have to go to open field to do that screening program. Right? So right in this juvenile plants, you do that, and you segregate the resistant versus susceptible and take it to the next. Yeah, mulberry.
Thank you.
Yeah.
Thank you so much, sir, for explaining nicely. Any other questions are there?
If no more question, I must tell a couple of points. Yeah, anybody?
Hello, madam.
Yeah,
I have
yes,
one question.
K.N. Gupta. Yeah.
Myself, Dr. K.N. Gupta from JNK Jabalpur, sir. Hello.
Yeah, I'm
Yes,
listening,
yes.
sir. Yeah, please.
I am a pathologist, sir. So first, I listen your wonderful presentation, and I have so learning so many things, sir.
Thank
I
you.
have one question, sir.
Yes.
Sir, if you screening the germplasm line across the location, so some center have disease percentage more high, like 90%, 80%. Some state, some location percentage is very less, like 10%, 20%. So when I go to mean evaluation, then percentage reduction percentage is less, below 20. So give me some suggestion for how to identify this type of germplasm line and result. Yes, sir.
Yeah, see, I'm not sure you are a pathologist, but disease is in
my sir, my PI is Dr. B.K. Barnwal, sir. I am a biology man.
Excellent. Yeah, very nice to know. Listen, the variations in the disease outcome is always there because we know that the 33% of the disease contributing factors come from environment. Correct.
Right,
the fact that you are going for multi-location evaluation, you cannot have the same environmental condition in all the places. So that factor you have to consider. You understand? So the best way forward is, see, not all diseases are prevalent in all the places, correct? So while selecting the location for disease screening evaluation, don't introduce a pathogen which is non-existent in that place. That is number 1 point. Number 2 point, you try to create a uniform condition. I'm not sure you are talking about the greenhouse evaluation or open field evaluation. If it is an open field evaluation, it is bound to happen. That is bound to happen. You cannot control that because it's an open field evaluation. Environmental factors are completely different. What you are screening in Delhi, what you are screening in Nagpur will have a completely different set of environment. We are talking about only the scoreable environmental factors like temperature, humidity, light. What about other factors, soil factors? What about other nutritional factors? What about other cohabitation factors? So everything is a variable there, and certainly it is bound to vary. But the message what I'm trying to tell here, you keep the susceptible check in every place, and your interpretation should be based on that susceptible check you put under the same condition, rather than comparing two different places. Comparing two different places is nothing but apple to orange comparison only, right? Isn't it? So the best is it should be apple to apple comparison only. The best way forward is a susceptible check you maintain in every place, and you evaluate your germplasm against that what do you call the susceptible check and come to a kind of
BM maintain susceptible as well as restage check also, sir.
Yeah, that's what I'm telling you. Open field evaluation, this is bound to happen. So nothing you can do about it, right? But how to minimize it? You have to create a kind of, that's what I told. Combination of two methods is always important, right? So you have to create a kind of a controlled greenhouse trial where you regulate the pathogen population, regulate the environmental factors, and put all your germplasm and screen under that condition. That would give you much better a stable result than open field evaluation, right?
Right, sir. Thank you very much, sir. Thank you very much, sir.
Thank you. Thank you. There is a
Thank you.
question, viral diseases. I carefully avoided that because there is
Indeed,
nothing.
sir, this is a question for macrofauna, stream rot and phyllodes.
Okay. Yeah.
Major root and stream rot, macrofauna fazulana disease.
Yeah, I understand. Yeah, it's a dry
Soil-borne and
loving disease.
seed-borne.
Yeah, dry loving disease.
Yeah, yeah.
And, yeah, certainly the moisture, other factors would influence the outcome of the disease. You are not
Yes,
going to
right.
get the same results everywhere. Yeah, not possible.
Right, maybe 90% disease I observe also.
Yeah. How to screen viral disease? I think I wish somebody develops it so that next presentation I can make that. Yeah.
Okay, I hope I learned something from you. Okay, sir. Thank you very much, sir. Thank you.
Yeah, thank you. Somebody is asking that question, Chantana Behra. So my reply would be, I think KN Gupta will make one and he will communicate to Chandana Behra so that we will be educated from this effort. Screening viral disease has been always a challenge. The reason being, the resistant breaking down is rampant because we all know that the COVID situation, what happened, RNA viruses are extremely amenable for mutations. No virus is stable, RNA viruses, right? Today you develop one variety which is resistant, tomorrow it is going to succumb to the disease again. So maybe the stability factor is something which play a part. And number two, the gene for gene interaction. I was talking about this. In fungal disease, it is so successful in our country. The reason being there is a gene for gene interaction is operating in fungal pathogens because these two are eukaryotic systems, plant and fungus. And to some extent, plant and bacteria as well. The gene for gene interaction is happening, right? XA21, AX21 interaction, we know that. Right? In rice, several resistant genes like PA51, then PETA, several other genes, PA2, PA9, those genes have been documented. And we know that where it is interacting with the pathogen effect is also cloned. So the point is, unless you have a gene for gene interaction, there's no question of gene deployment comes into picture for this resistant breeding program. So in virus, the story would be quite different, right? So first of all, we need to ascertain whether gene for gene interaction is really happening in plant virus interaction. If that is happening, how stable that plant resistant gene, which is conferring resistant. Of course, people have already identified certain genes in the case of leaf curl virus as a TY1, TY2, TY3, it is already there. But the durability of those resistance is always under scanner, right? Because I already told a eukaryotic pathogen Macnopurdi can develop around 30,000 conidia from single lesion. You know how many viral particles are generated in the symptomatic leaf of tobacco or tomato? It runs into millions, right? And the chances of getting 1 or 2 particles mutated is always there. And the cultivar is going to be succumb to the disease sooner or later. So maybe this is one of the reasons why the resistant breeding program is not very successful in viral diseases so far. And probably in future, a better technology will come, maybe better opportunities will come. We may come out with a kind of scoring methods. Yeah. Thank you. Yeah. So I'm just reading out the comments, how to screen, and there is information, very informative talk. Thank you so much. This is an opportunity given to me. The first time I made these slides in my career. So thank you, PGR people, for giving me the opportunity. So maybe my lecture note, you can share it with the people who registered for this course. So we put a lot of effort in that. So you can read that. Yeah. Thank you. Yeah.
Thank you so much, sir. Actually, you gave us the opportunity to listen to you. It was such a nice presentation and especially useful for those who are working with the respective diseases that you
Madam, before you conclude, I want to make one statement here. Whoever is involved in resistant breeding program on whatever pathogen, please include a pathologist on board. Reason is many times people approach me for a type of race of pathogen, which I'm not working on that, right? So this choice of pathogen is very, very important, right? If you are choosing a wrong pathogen, all your efforts will go waste. So there is a guiding principle also. The guiding principle here is the ruling pathogen, right? We know about the ruling cultivar. The same way we have a ruling pathogen. So we have to use a ruling pathogen, the pathogen in action that need to be isolated and it should be preserved somewhere, right? Here comes one of the most important issue what all of us are facing in our country. We do not have a culture collection where pathogens are preserved. This is a reality. pathogens are preserved in the individual labs because I have several pathogens, but I'm looking for a place where I can house it so that the rest of the countrymen can use it. There is no culture collection. Even if culture collection is there, it goes to a general collection and it gets lost there. And nobody's paying attention to maintaining the virulence of those pathogens because somebody is having, I have thousands of collections and my culture goes there as one of the thousands. And the individual attention, because the stability of the pathogen is very important. And this stability can be maintained provided you create a specialty culture collection in our country where we keep only the pathogen collection. I'll take one example, my own experience. UK has got a pathogen collection, right? They have a national collection of plant pathogenic bacteria. What they do, they maintain only the pathogenic bacteria, global level. They have the collection from India also, right? So the point is, anybody can access to that collection. You have to pay some money. That money can be given, not an issue. But what is more important here is how authentic that collection, number one, how virulent that collection, number two, and number three, whether the collection has got the passport information. These three factors are very important, right? If these three factors are there, we can access the collection and we can use in our breeding program. Now what's happening is somebody is having one collection, somebody is asking, hearsay, culture changes the hand, and that the so-called non-scientific method of inoculations are going on. We make a wrong interpretation. So all these things we have to put an end, provided at least the government take a policy decision to create a center where we keep the plant pathogens. Right? I would always tell this point, a national collection of plant pathogens that need to be created. And it would serve the needs of all plant breeders across all institutes, be it SAUs or it is a kind of IRI, ICR institutes or NIPG, different institutes, right? Some institutes are having the technology, they don't have the pathogen and germplasm. They come to us. When the pathogen changes the hand and the cultivar changes the hand, the relationship stops there. Correct? So what I'm trying to tell here is you take a right pathologist in your team, and get the right pathogen with the pathogen material is preserved somewhere so that you don't lose track of what you are doing. Right? This is my small appeal to all of you. And those who are involved in this program, make this point and put a word to your authorities. There is a need for a pathogen collection in our country, if not state-wise, at least in the central. Thank you so much. Much like what you are doing in NPPGR, right? Yeah.
Thank you, sir.
Thank you. It is so nice to see your enthusiasm explaining and instructing everyone. So we would like to hear you in future also. With this request, I would again like to thank you on behalf of our organizing team as well as our participants. Thank you so much, sir, for being with us.
Thank you. I can leave now?
Yes, sir. Yes, sure. Sure.
Thank you so much, and
Yes,
thanks for your patience.
Thank you, sir.
Yeah, thank you.
Thank you,
Thank
sir.
you very much, sir.
And thank you, Gautam sir. I do not know whether he is logged in, but convey my regards to him also. Yeah, thank you.
Sure, sir.
Right. Thank you.
Okay, so we will reassemble at 2:30. So today's session was somewhat longer than expected, so no problem. Next session will start on time. So you all are requested to be back by 2:30. Thank you.
Presentation mode.
Okay.
Yes, working.
So, yeah. Okay, sir. So our today's speakers are first. I would like to introduce Dr. Lalitarya ma'am. She is principal scientist at NBPGR in the Division of Genomic Resources, and her specialization is in biochemistry in the area of plant sciences. Her nature of work includes fingerprinting and diversity analysis of the various crops, especially in the crop group of millets. She is involved in the establishment of the DNA fingerprinting of cereals and millets at ICAR-PGR. She has developed a large number of SSR markers also in barley, wheat, sorghum, pearl millet. Then her laboratory also provides DNA fingerprinting services to the private and public sectors in the crops of related to millets. Then she also has characterized more than 1600 accessions of the little millet recently at NVPGR. Then she also has generated various EST, SSR markers and other genomic tools for millet group. Then she generated leaf transcriptome data in little millet, finger millet, kodo millet, and their bio projects are also available at NCBI. And she is one of the faculties of plant genetic resources at ICAR and PGR, teaching enzymology. Then she has been also involved in teaching the ecology and biodiversity courses, PGR courses, and she has also guided MSc and PhD students. She also has bagged so many awards with her name, like Best Research Paper awards. Then she also has been recognized as the Fellow of Indian Society of Plant Genetic Resources. And she has very good number of publications in various journals. Then our next speaker is Dr. Manjusha Verma. She is also one of the principal scientists at the Division of Genomic Resources. Sorry, I wrongly mentioned IRI. She is at NVPGR. Then her nature of work includes various development of the fingerprints in the crop group, especially pertaining to the cucurbitaceous crops. She also has developed the first SSR-based linkage map in bottle gourd. Then she has the experience in established methods as well as their integration with emerging genomic technologies for the understanding of the genetic variation in horticultural crops. She has evaluated genetic variability based on morphological and biochemical molecular markers in several cucurbitaceous crops. She has developed thousands of SSR markers in bottle gourd, sponge gourd, watermelon, small millets, etc. And she also has developed the first ever transcriptomic signature in bottle gourd and tobacco leaf curl New Delhi virus resistance in susceptible sponge gourd. So she is also working with Dr. Lalit Madam. So both of these will be our today's speaker. So I welcome both of you, ma'am. And I will stop sharing and request both of you to please proceed with your schedule. So now the session is over to you. Thank you for being with us, ma'am.
Thank you, Mamta, for the introduction, and good afternoon to all the dear participants. Today's presentation is more towards, it's a practical session. So I'm just sharing my screen for the presentation. Am I audible, Mamta? My voice is clear?
Yes, ma'am, it's quite clear, ma'am.
Okay. So today's presentation, molecular techniques for characterization and evaluation of PGR, is a practical session where we will discuss about, different protocols for different molecular techniques that we use for characterization of plant genetic resources. And I'll start with my first slide. This slide, just I wanted to show like on what different crops we have worked in our lab. Cereals, millets, then we have medicinal plants, cucurbit crops. We have also worked on guided students on French bean and rose. So this presentation and the use of molecular markers will show how different markers, specific and random markers, they can be used in different crop groups. So when we talk about characterization is the key to utilization. So we have large number of germplasm, which is available ex situ in our gene banks, as well as in situ in natural habitats. So we need to characterize them so that we can utilize them in crop improvement programs. So how we can characterize them? We all know that we have different methods of phenotyping. We can phenotype the agromorphological traits. We can go for, measuring different primary and secondary metabolites. We can also use seed proteins or, isoenzymes for characterization, and molecular markers, which are basically DNA-based markers. So all these we can use together or individually to characterize our germplasm. So why, molecular techniques, they are important because molecular techniques have the added advantage of not being affected by environmental conditions. And secondly, they are rapid, fast, and you need not grow the plant up to a particular stage, and you can just take even small seedlings, any part of the plant, and extract DNA and start your, experiments and get the results. So we have different kind of molecular markers that I have shown in these pictures. We have hybridization-based markers like RFLP, and in nowadays we have the microarrays, which are hybridization-based. RFLP involved, radio-labeled primers. That's why they are not in use now. We are using now PCR-based markers, and in PCR-based markers also, we have the markers which are random, like ISSR, RABD, FLP, which we can apply it on any of the crops. But then we have, these are neutral markers, and we have some, gene-specific markers also, like SRAB, SCOT, that we have used in different crops for characterization. And then we have crop-specific markers like SSRs. These are the profiles for SSR that have been run on Metaphor Agarose, and this have been run on, sequencer. This is the automated system for running, your PCR products. Then we have now we are in the era of genomics. So we are using, sequencing techniques to genotype our germplasm. And it will all depend upon, what kind of information we already have. If we don't have much information, we can go for like reference genome sequence. We can go for, GBS, genotyping by sequencing, or RAD sequencing. If we have reference sequence and we have, a diverse set of germplasm that we can use for, resequencing. So we can use the sequencing technology to sequence whole genome, to resequence the lines, and then we can use that data for marker trait associations, for finding novel alleles and all that. So I'll basically discuss the protocols for the techniques like ISSR, SSR, SRAB that we have used in our lab, and these markers, we are in the era of genomics, but the information that is being generated by sequencing, that is to be validated. And that validation can be done using these techniques like SSRs. We are developing, we use, gel-based techniques, and we have a number of labs in our country which don't have these facilities. So these simple and PCR-based techniques can be applied, and they can be, if we have smaller sets, we can use these techniques. And for specific purpose only, we need these techniques to characterize our germplasm. So my next slide is to show, like, because in NBPGR, different scientists, they are working on larger sets. They have started, using this sequencing technologies to characterize the germplasm. So one, like in rice, Dr. Rakesh Singh, you have heard in the morning, he's working on SNP chip and using SNP chip to characterize the genotypes. Then, on wheat also, and this pulse crop also, oil crops also, this work is, we are using next generation sequencing technologies to characterize the germplasm. So what is the use of, like, why we are using it? We are characterizing it, we are getting, sequence-based data. So what is the use? So use is like, and for that, like, how we can use before that, if we want some marker trait associations, or we want to pinpoint novel alleles or variants, for that, we have to first purify the germplasm. Okay, so it will all depend upon, the type of crops that breeding behavior the crops have. Like if they are self-pollinated or cross-pollinated. So you have to follow that protocol to purify the material. Once the material is purified, then we multiply it so that we have enough, sample for each accession to characterize it phenotypically, as well as we want to, profile their, secondary or primary metabolites. Then we can genotype also. So same, larger sets, they could be genotyped using these sequence-based technologies, and the data generated can be analyzed through different statistical softwares to construct, molecular cores. From phenotypic data, we can generate phenotypic cores, and we can, generate reference sets also with respect to nutritional profiling data, and even if we have screened them for biotic or abiotic stresses. So ultimately we can use this data individually, or we can combine them, and we can set up a diverse panel or a composite panel that can be used for evaluation at multi-location, so that a larger set will be, all the diversity that is there in the larger set, it will be captured in a smaller set that can be, easy to handle and can be characterized through nutritional profiling, then phenotyping for agromorphological traits. We can easily, if we have sequence information, reference sequence, we can go for resequencing, we can go for GBS if we don't have any reference information. So all this data then can be used for, QTL mapping, GWAS mapping, if we have mapping populations, that also can be characterized, and we can, find out marker trait associations or different variants. So that way we can, use this particular genomic information to find out our, diverse genotypes, trait-specific genotypes that can be utilized in crop breeding programs, and they will certainly, enhance that. It will speed up the process of, breeding programs. Now, since we all know, for any molecular technique, the basic is, DNA. So for each technique, we need quality DNA. For that, you have to extract the DNA from samples. Samples, you can take, any sample, you can take seed, you can take seedlings, any part of the plant can be used for DNA extraction. In our lab, we have we use CTAB method, but sometimes we use commercial kits also. The CTAB method, I will tell you that this method is very useful. We have applied it on cereals, millets, and cucurbit bit crops. We have tried it on medicinal plant like morinda also, and French bean, rose, any type of plant, DNA can be easily extracted using CTAB method. So, and the beauty of this process is that you can scale it up, you can scale it down, depending upon, how much sample is with you. Like if you have small sample, you can just take 1 ml of the CTAB buffer initial step. And if you have large quantity, enough quantity of material is there, then you can, take 10 ml or 15 ml CTAB buffer. So first step is like what care you have to take. You need pestle mortars. They should be chilled one. And then you have certain buffers and different reagents with you. And a water bath. So we, water bath, we maintain it at 65 degrees Celsius. And say I'm taking 15 ml CTAB buffer, which consists of CTAB, NaCl, EDTA, tris-HCl, and beta-mercaptoethanol. So that particular buffer, we take centrifuge tubes, Oak Ridge tubes, and put 15 ml in that tubes. And keep it for few minutes in water bath so that it is preheated. And the next step is like we take the material, put it in a chilled this pestle mortar, grind it, and you have to grind it properly. Make a fine powder so that the proper cell lysis is there. And then if in between you see that thawing is there, it is getting moisture is there, so you just put more liquid nitrogen and immediately transfer the ground material to the tube containing CTAB. This particular step is very important, to get good quality as well as good to get good quantity DNA. So what we do after crushing that, we transfer the material into 15 ml CTAB buffer, and then we mix it very vigorously. Here at this particular stage, you can mix the samples vigorously so that the proper cell lysis process occurs, and then we keep it at water bath at 65 degree. Maybe depending upon our time, we can range it from 30 minutes to up to 1 hour we can keep. So before going to the next step, I'll tell you what is the use of CTAB. CTAB buffer is basically, it's a detergent which help in the lysis of cells, and also because we have polysaccharides also, so it will bind to polysaccharides and remove them. And secondly, like we have put in this, EDTA. EDTA act as a chelating agent because we have in the cell nucleases also. So if they are there in the active form, so they may, destroy the DNA. To keep the DNA or nucleic acid safe, we put EDTA because it binds to magnesium ion, which are necessary for the activity of, your nucleases. Then we are putting beta mercaptoethanol. Beta mercaptoethanol, it helps in the denaturation of proteins. It breaks the disulfide bond between cysteine residues, and even it helps in the removal of tannins also. So this is the and in the next step. So I'll come to the next step where we have kept, our ground sample at 65 degree in CTAP buffer. So we take it out, and then what we do, we open the caps and put equal volume. If we have taken 15 ml CTAP buffer, we put 15 ml of chloroform and isoamyl alcohol in 24:1 ratio. Okay, and if we have taken 1 ml, we'll put same 1 ml if we are doing in a smaller scale in a pint of tubes. So after that, we cap it, and here in this, from this particular step, we don't have to follow the vigorous shaking. We will mix it very gently, and you can form number 8 in the air, and you can mix your sample. And after 5 to 10 minutes, you centrifuge it at 10,000 RPM for 15 minutes, or maybe 12,000 RPM for 10 minutes. So then you centrifuge it, and you can see in this particular picture that, you get an upper cell debris here, and here is the upper aqueous phase. And in this, you have DNA, RNA, and other impurities along with that. Now, at this stage, we have to collect this supernatant into a new tube. Coming to the next step, why we are using isoamyl alcohol and chloroform. Chloroform, basically, it helps in the denaturation of protein. And isoamyl alcohol, it stabilizes it prevents the foam formation. And in the next step, we will see, like, now the supernatant that we have collected in a separate tube, here we will put chilled isopropanol. So the function of this isopropanol is to precipitate your nucleic acid. Here in this step, in the supernatant, you have nucleic acid, both you have DNA as well as RNA. So when you put chilled isopropanol, it will help in the precipitation of nucleic acid. How it is doing? Like isopropanol, it will form hydrogen bonding with the water molecules, and less water molecule will be available for the dissolution of your nucleic acid. So they will get precipitated, and it in some of the crops like wheat, sorghum, immediately after putting your isopropanol, the nucleic acid gets precipitated. But in some of the crops, it takes time. So you can keep it at -20 degree for a few hours, and even maybe overnight. But preferably, if it is getting precipitated, we should not keep it overnight or for longer time period, because it will, start precipitating impurities also. Then you have to get rid of those impurities. So it's better if you are getting your, nucleic acids are getting precipitated immediately, or after an hour, or you should follow the next step. So after that, we centrifuge it at 10,000 RPM for 15 minutes, and we will get the pellet, that is your nucleic acid. Now we have to wash it. For washing, we use 70% ethanol. Mamta, hello?
Yes, ma'am. Yes, ma'am.
Is it okay? Because speaking online, it seems like I don't know.
Yes, everything is okay.
No, ma'am.
Okay.
In between,
It's good.
if you want, you can interact also.
So, I am able to convey my message properly.
Yes, you are.
Oh, okay.
Yeah.
So, washing we do with 70% ethanol. We don't do it with 100% ethanol, because if we are using 100% ethanol, it will precipitate the DNA. So we keep, we cover it with 1 ml, or depending upon the pellet, we cover it with 70% ethanol, keep it for 10 minutes. And then again, we centrifuge it and remove that upper, aqueous phase, and the pellet is there. You can wash it 1 or 2 times. And then you can, after centrifuging, you can keep your pellet for drying. Drying you can do, if you have less time, you can go for lyophilizers also. If you don't have, if you have time, then you can, it's better to keep your, pellet to dry in air. We keep it overnight. And next day when we come, we put TE buffer to dissolve the our pellet. So in that pellet, now we have DNA as well as RNA. So if you want to check whether you got your DNA or RNA, so we just, run it on 0.8% gel and see, and this, you can see this picture, the above one is the DNA and the below one is the RNA band. So we have to now get rid of this RNA. So what we can do, like after, we dissolve the DNA, this nucleic acid gets dissolved, you keep it for, maybe half a day or 1 day, and then you can use it. Or if you want it immediately, you can keep it at, 65 degree for 10 minutes. So that it is dissolved. And then, what is the next step? We use RNase in the concentration of 10 mg per ml. And we put it in this dissolved, nucleic acids and keep it for 1 hour at 37 degrees Celsius. So it will denature all the RNA. Now, because you have to, RNA is denatured, but you have to remove all this, proteins and other kind of impurities further. So we use phenol chloroform isoamyl alcohol step. And in this, the ratio is 25:24:1. So in that ratio, we prepare the mixture and then we use it. If it is 1 ml, we put equal volume of this phenol chloroform isoamyl alcohol. Again, in this step, we mix it very gently. That is the only first step where you can mix the, your samples with the solution vigorously. That is the first step. Otherwise, in all the steps, you have to very carefully, very gently, you have to mix the samples. So here also you can form 8 in air and mix it for 5 to 10 minutes and then centrifuge it for, at 10,000 RPM for 15 minutes. And then what will you see? So you will see this kind of picture. This is the upper aqueous phase. This is the interface where you get coagulated proteins. And this lower phase is your, where lipid kind of impurities are there. That is hydrophobic phase. So you're now in the above aqueous phase, your pure DNA is there. Okay. So all the impurities, whether they are protein, lipids, and the kind of impurities, they are there in the either in the interface or in the lower phase. So very gently we have to collect this aqueous phase into another tube, and there we can mix it with again equal volume of chloroform isoamyl alcohol in 24:1 ratio. Again, that step is repeated. Mix it gently, centrifuge it, again recover the upper aqueous phase. And now to the upper aqueous phase, what we have to do, whatever volume we are getting, say we have started with 1 ml and now we are getting 700 microliter. So, accordingly, you have to put 1/10 volume of 3 molar sodium acetate and 2.5 times of chilled ethanol. So the purpose of putting sodium acetate or chilled ethanol is precipitation of DNA. Now we will get the DNA in pure form. Now, at the first stage, when you are getting nucleic acid, you will, like white colored, or, if some other, secondary metabolites are there, then something, color kind of thing is there in the precipitate. But here at this stage, you will get crystal clear, you will see precipitates of DNA. And the purpose of putting chilled ethanol is same that I told you earlier also, like it will bind to your water molecule, and less water molecule will be available for, dissolution of this DNA, and they will get precipitated. And the sodium acetate we are putting because they are enhancing this precipitation process, because sodium ions are positively charged. So they will bind to the, negatively charged phosphate ions on the DNA. And so the DNA will be getting precipitated. And now washing step, again, we have to do it with 70% ethanol. Again, the same sample, same, step is repeated that we have done it before purifying it. And we dry the pellet, centrifuge it and dry the pellet overnight or in a lyophilizer, depending upon the time you have, and dissolve it in minimum amount of TE. Now, here we dissolve it in minimum amount of TE because we want, to get your DNA in concentrated amount. Then while working for PCRs and other reactions, you can dilute it. So now this is the picture to show how, how this purified DNA look like. So we have covered DNA extraction and purification step. One thing more I would like to tell you, like if you have the problems of polyphenols, then you can add PVP at the rate of 1%. If you have more proteins, you can add proteinase K. So that way, you know, these things can be added. And like in Morinda, we have to add 1%, you know, this PVP. But in cereal and millets, we don't need it. And we just, you know, without that, we have carried out extraction steps. So now for DNA quantification, we generally use here nanodrop. That is the spectrophotometer that we are using it. It is very simple to operate. You need not, you know, take that, you know, H dye or that carcinogenic dyes for estimation, or that BSA and calibration and all that. That kind of, you know, tedious process is not required. And just, you know, 1 microliter of sample is sufficient to estimate the DNA. So what we do, like this is the, you know, nanodrop that look like, and here is the pedestal. And at this particular point, you use, you know, we use very small volume pipettes, like 0.1 to 0.10 microliter, because we have to dispense 1 microliter of the dissolved DNA. So we put it here. And before that, you have to do a blank estimation, like the buffer you are taking, and that buffer, you know, it is put as a blank, and then you take the set the blank. Here you can see this particular picture, that is the interface of that particular, you know, nanodrop readings that come in because it is attached to a computer, so that, you know, software face look like that. Here you can measure the readings. Here is the blank set. And so after setting blank, you have to, you know, it is basically, calibrating it, and then you have to take your unknown sample, that is your DNA. So then you have to measure the reading. It will give you reading in this form. You can see here it is written 249.7 nanogram. So in a microliter of this particular sample, we are getting 249 nanogram. And you can see the readings here, like ratios of 260 by 280, then 260 by 230. So generally we see, like if the DNA is pure, that reading will be 260 by 280, ratio will be 1.8, and 260 by 230 ratio will be between 1.8 to 2. So if that kind of, you know, readings you are getting, your DNA is pure. And here you can see how beautiful peak we are getting. And so this is our stock DNA solution, and depending upon the PCR requirement, we you know, dilute to working solution, like 20 nanogram per microliter we make it. And another thing, like this particular instrument nanodrop that we are using, starting from 2 nanogram up to 3700 nanogram per microliter concentrations can be measured with this. So even if your sample is concentrated, then also it will give you a good reading. And we are using this particular instrument for past many years, and without any hassle, you can quantify your DNA. Now, I will just tell you the different protocols that we are using for different marker techniques. This is the protocol for RAPD, because RAPD markers, they are just 10-mer markers. So here we need very low annealing temperature. So we are using 37 degree. Basically, first I would like to tell, like it will be good for those who have not done PCR before. So PCR step, it involves 3 steps. Like one is the denaturation, another one is annealing, and then extension. So basically denaturation step, it denatures your double stranded DNA into single ones. And then at the annealing step, your primers, they will bind at the complementary sites. And extension step, basically whatever binding is done, and you know, your GMTPs are being added, so they will be extended, and your, you know, particular stands, they will be getting amplified. So that is the extension step. And generally we do, you know, that denaturation step at 94 to 96 degree. We in our lab are following 94 degree. And then annealing steps, it will all depend upon the, you know, type of technique, or basically the type of, you know, the length of the primer that you are taking. If they are the 10-mer, 37 degree annealing temperature is sufficient. But if you are taking, you know, from 16 or 18 base pair up to 24 base pair primers, then you have to, your annealing temperature would be high, and they may range from 48 to 64 degree. And then extension step, that is performed at 72 degrees Celsius only. And the reagent that we use for PCR, they are, you should have your template DNA. You can make, you know, working concentrations. Like earlier, we got 249 nanogram per microliter. So we can make either 25 nanogram concentration, 20, whatever we require for our, PCR reaction. Then buffers along with magnesium chloride or without magnesium chloride, they are supplied by the company from which you are buying your tag DNA polymerase, which is the enzyme which is involved in this amplification. Then you need DNTPs and you need primers and water to make up your volume. We generally do it at 25 microliter. Depending upon if you have, even you can use 10 microliter or 15 microliter of reaction mixtures also to, perform your PCR reactions. So this is the basic things that you require. One, these reagents, then you have a PCR machine. Earlier we were having 48 well machines. Now we have 96 well machines. There are 384 well machines are also available. So depending upon your, sample size, you can use those and the availability with you. So these PCR machines, they carry out this particular reaction that I'm telling you, like denaturation, annealing, extension. So this is being carried out in a PCR machine. So what is generally like in RAPD, I'll just explain. We give an initial denaturation of 94 for 5 minutes, so that whole double stranded, they will get denatured into single strands. And then we give a cycle phase. They may be from 30 to 40 cycles, like denaturation at 94 degrees Celsius for 1 minute, annealing 37 for 1 minute, extension 72 for 2 minutes. And this is repeated for a number of cycles, maybe 20, 30, 40. Okay, that will amplify your specific, will give you specific PCR products. And so this is being done. And then finally, an extension step of 72 degree, maybe ranging from 5 to 10 minutes, that is given, so that all, whatever amplification is being done, that is completed. And then you can set your machines at 4 degrees, so that if you are not there, your sample is there at your refrigerated temperature. Next day, you can come and you can take out your PCR tubes, put, gel loading dye in that. And then because this gel loading dye, it will give you, a weight, because it consists of bromophenol blue xylene cyanol, then glycerol, so that you can load it in your gels. So that is being done. For every PCR reaction, we first go for PCR, then we run it on gels. And gels, like if we are using RAPD, ISSR, we run it on 1.4 to 1.8% gels. And if we are using SSRs, we run it on 2.5% to 3.5% metaphor agarose. And even we can use, if we do, because metaphor agarose is very costly, so we can use even 2.5% of regular agarose also for separating our PCR products, which we have got with SSR genotyping. So this is the profiles which look for, if we run RAPD, this is the SSR profiles that have been run on 3% metaphor agarose. And this particular profile is on sequencer ABI Prism 310, and this is the ISSR profile. So I'll just again come to the annealing temperature, because all the basic steps like denaturation, annealing, and extension, they are same. The only difference is at the annealing temperature, where in RAPD, we are using 37 degrees Celsius. For, SSR, we are running, depending upon the GC content of our primers, forward and reverse primers, we are using in SSR. Another difference is, like in RAPD and ISSR, we are using single primers. In RAPD, they are 10 mer, and in ISSR, they may be 15 to 24 base pairs. And here the annealing temperature ranges from 48 to 64 degrees Celsius. So the basic thing is, like, you have to standardize. Why we say we have to standardize the protocol, that is basically dealing with the standardization of your annealing temperature. And more or less the components, like we can take, if we are taking 25 microliter of our total volume, 50 to 100 nanogram template DNA is sufficient. Then 1.25 millimolar to 3 millimolar magnesium chloride is sufficient. Then we can put 0.2 millimolar of DNTP mix. All the 4 DNTPs will be there, and 1 unit of tag DNA polymerase is sufficient. So this much, you don't have to toggle much with this and component concentration. But yeah, this annealing temperature is very, very important. So it will all depend upon the length of your primers that you are taking. And one thing I'll tell you with my experience, like you can use a touchdown cycle, because that cycle gives us if you are getting more non-specific bands, if you are dealing with SSRs, so that will not be there. You will get very clear-cut gels. And because here this particular PCR step is divided into two. One is the you do some cycles at higher temperature, and then you fix it at particularly say 55 degree, and do at amplification at low temperature. So that will reduce the amplification of non-specific bands, and it will bind only, primers will bind only to the specific bands, and you will get clear-cut profiles in this. Particularly if I am talking about SSR, when you run SSR primers. So again, RAPD, you have 10-mer primers, SSRs, you have forward and reverse primers too. And in ISSR, you have single primer only. But here, ISSR primers, they are like you have heard about repeats, dinucleotide repeat, trinucleotide, tetranucleotide repeat. So here, the repeats itself like 80, 80, 80, this itself act as primers. And here in SSRs, we design primers from the flanking regions where the repeats are there. So we have say double. I'll tell you this in the next slides how we design the primers. So this is just the protocol for RAPD, SSR, then ISSR. Then we have used Scott markers also in our lab. So basically, if we talk about Scott markers, these are functional markers or gene-based markers. In that, 18 primers are used, and they have been designed from ATG start codon flanking regions. And the beauty of this is like this also, give you beautiful bands. And they can be run on normal agarose gel. You can use 1.4 to 1.6% agarose gel. And the primers is like they have fixed positions for certain nucleotides like ATG at +1, +2, +3 position, G at +4, C +5, and A, C, and Z at positions +7, +8, and +9. The difference between the primers is at 3' position by at least one nucleotide. So within the last 3 or 4 nucleotides, the primers will be different. So in our lab, we have, I think, 80 Scott primers are available. So we have synthesized those primers, and then they are being used in different crops. We have used in medicinal cucurbit crops. We have used in millets also. They are working well. And here the difference is like annealing temperature here is 50 degrees. So depending upon the primer length, it has been standardized. And same, here 1 micromolar of Scott primer is being used. It's a single primer that we are using. First we do 94 to and denaturation at 3 minutes, then 1 minute the cyclic step for 94, 50 degrees, 72, then extension step, and then we have run it on 1.6% agarose gel. So this is the gel picture from the Morinda. Morinda is a medicinal plant on which we have worked. So this have been run with different primers, and you are getting different patterns. And then we have SRAP markers. This is sequence-related amplified polymorphism. And these are also functional markers. And here in this, we used, forward and reverse primers. Like in SSRs, we use forward and reverse primers. But these SRAP, Scott, RAPD, SSR, this we can, use it in any of the crop. You need not, because they are random. But SSRs, you have to synthesize it from a particular crop, and then you can use it. Though you can transfer it into, related species also. So SRAP markers are open reading frame markers and are known as functional markers. So in this, a unique combination of SRAP, forward and reverse primers are there. So, and it involves a 2-step amplification. So the one is like starting at the 5', the first 10 or 11 bases are non-specific. They are called filler. And in the forward primer, there is CC, GG. And in the reverse primer, there is AATT. So this is the composition of the primer. SREP primers. So they comes with the name of ME and EM. So those, we can try different combinations, and then we can see like wherever we are getting, these kinds of, clear cut, unique bands that can be scored. So that is called the standardization, because you take, say there are more than, I think I have seen in the publication, more than 200 such kind of primers are available. So different combinations can be tried. So whichever is, giving amplification, so that is the set for you, and then you can use it in, large number of genotypes to characterize them. So here is the first step for denaturation, that is there, 94 for 5 minutes and 1 cycle. This is first, cyclic phases for 5 cycles, but that is done at 35 degree. And the second one is done as 50 degree for 35, then 72 for 5 minutes. So here, this low 35, it will give you a partial, because at most of the places, because at low temperature, wherever you are getting partial matches also, the primers will bind to that particular site. And then when you are running it on higher temperatures only, specific one, they will go ahead and you will get your amplified product. So this particular, profile is SREP primer combination, EM1-ME8, 1.6% agarose gel is used, and this has been run on codomimetic sessions. So this slide is just to show like what kind of picture gels we get. Like this is for SSR, how we can score it. This is for RAPT profile. You can size it. Always whenever we run our gels, like for SSRs, we are using metaphor agarose. It has been run on 3% agarose. And then you have to, along with the gel, you have to put, your markers also. For gel, like if I am saying 1.6% or 3%, you have to weigh 3 gram and put it in 100 ml of the TBE or TAE buffer that we usually do. For smaller fragments, and if we want like our, that electrophoresis buffer, we can use it for longer times. We generally use trisborate EDTA buffer because it doesn't get exhausted easily. You can use it for longer time. And so that buffer is used in the electrophoresis tank. You make a gel, say if it's a 3%, you weigh 3 gram for 100 ml. And if your gel, if it is smaller one, you need 100 ml. If you're bigger gels, then you need maybe 400 ml. So accordingly, you will multiply 3:4, 12 gram will be there, and you will put it in 400 ml. So we put little extra like 410 when dissolving, because while we dissolve it and we warm it and heat it in a microwave, so during that some of the buffer will get evaporated. So to make the concentration appropriate, so we put it, say 410 ml, and then we use it. And generally for regular agarose, we need not have to follow a specific procedure. Simply you have to wait, put it in the tea buffer, and then you heat it, and then you put on a dye. We are using ethidium bromide dye, and then you mix it, and you have set your plate. A plate is there in which different wells are there, and in that we put a comb so that different wells are created, and then we pour that gel. So that gel should be, while loading, that gel should be at the temperature of 60 degrees, so that it should not be too cool or too warm, so that your wells are properly made. And after that, those PCR products that we have run in our PCR machine, we put loading dye so that we can track our gels. So that is put in the wells, and we run it for a particular time period. Say initially we give it 150 volt for half an hour, and then we run it at 110 or 100 volt for 4 to 5 hours, depending upon, because once initially we run it at a higher volt, because so that immediately that your PCR products along with the dye, they'll come out of the gel. Otherwise it will it may come out, or your sample will lose. So immediately they'll go to the inside the gel, and then we run it at lower volts, so that your PCR products, the bands which are not visible to you, they will be, properly, they will get separated depending upon the size. Higher one above, and the lower one will get in the lower size, the low molecular weight markers. And so that is the thing, you go for when you go for gel running of. After that, that gels can be visualized on a UV transilluminator. because ethidium bromide dye is there, it intercalates the double stranded DNA, and then you can see under UV. And because now we have gel documentation systems with us. Earlier, we used to, when I joined this institute, we used to have Polaroid cameras. Now we have gel documentation systems. So that is, camera is directly attached to your transluminar unit, then it is attached to a printer. So we take gel photographs, and these are the photographs that have been taken using those systems. And after that, we go for, once we have all the gels with us, all the photographs with us, we can score those gels. And if we have SSR data, we can size them as base pair, like in what is the size of that particular allele, or even in this also, we can score it by the presence or absence of band. So if I'll take this particular gel, so I see here we have 11 samples, and this is the molecular weight markers, 50, 100, 150, 200, 250. These are the sizes. And from here, we can size our fragments. And you can see like this particular band is that we have gotten 10 and 11. It is around 150 base pair, or we can say 148 base pair. And then for this particular band, it will be around 130 base pair. So while making our sheets, we can just record it as like this particular band. If we are taking presence or absence, so it will be up to 6, it will be present, then in 7 it is 0, 8, 9 present, and then 0, 0. So that way we make, these kinds of Excel sheets. If we are scoring it size wise, then in sample 1, what is the size that we put? In sample 7, what is the size we put? So this way we get, different scoring sheets, and then we can use different programs. Like we have Antisys, we have freely available systems like Darwin, etc. We can use to calculate similarity or dissimilarity matrix. We have power marker. Earlier it was there. Now I think it is not available. But if you have to run, if it is with you, or you can change the date in the computer, and then we can run it. So it was a very good program where you get gene diversity, you get heterozygosity, PIC, all those values. So finally, so these kinds of parameters, they can be calculated, and we can generate such phylogenetic trees. And different statistical tools that we coefficients are there, like Jaccard coefficient, Dice, Lee coefficient. Different coefficients are there that we are using. We particularly use Jaccard coefficient, and this is the formulas for all those coefficients. I'll give because all these calculations are being done in the software itself, but we should know like how we are calculating it, how like if we have a number of samples, how individual between two pair the similarity coefficients are calculated, how then they get an average value. So if this is individual A, this is individual B, and we can see like if it is 1, 1 A, like if it is 0, 1 B. So that way you can see and put your numbers like A, B, and C. Here you can see like 1, 1 individual, the band is there in both the individuals, individual A as well as individual B. So such kind of things are there in this particular sample here and here. So the value of A is 3. Now B we see it is 0, 1. 0, 1, it is there in here and here. So it is there in 2 samples. Now with C, 1, 0. So it is here in 1, 2, and 3. So the value of C is 3. So we put these values in this particular A divided by A plus B plus C, and we can get the value of Jaccard coefficient. Similarly, we can put values in for Dice coefficient. And in Dice and Knee, we get little higher values, but depending upon, because here you are more concentrated towards, in Jaccard, we are saying more similarity kind of thing, that is we are considering in between the two individuals as compared to Jaccard coefficient. So that's why, but further we use this Jaccard coefficient values if we are using Antisys for making, we do for sand clustering, and then we generate UPGMA kind of dendrograms. I'll show Darwin program and population structure, how we run them to get different kind of graphic presentations, so that I'll take later on. But before that, other coefficients that we can use are expected heterozygosity. where the formula is 1 minus summation pi square, where pi is the frequency of the allele. So whatever, first of all, you have to calculate the frequency of a particular allele. And then suppose with the particular loci, you are getting different alleles. So for each allele, you will see the frequency in different individuals, and you will calculate pi, and then you will square it. And for all the alleles, you will sum it and minus it from 1. So that will give you gene diversity. Similarly, we calculate PIC value for codominant markers, SSR markers also. But for calculating PIC value for dominant markers, there is a different formula that I'll show in the next slide. Another parameter that we calculate is resolving power. Here it is equal to summation IB, that is the sum of band informativeness of all the bands produced by a primer. And how we calculate band informativeness, it is 1 minus the bracket 2 into, this is absolute value for 0.5 minus P, where P is the proportion of genotypes containing that band. Because every time we are considering one band or an allele and seeing its presence or absence or its frequency, so that way we can calculate resolving power. Higher the resolving power, the more informative the marker will be. Now we can calculate marker index, which is the product of expected heterozygosity, gene diversity, PIC, and effective multiplex ratio. And for effective multiplex ratio, the formula is n into beta, where effective multiplier ratio is defined as the product of total number of fragments per primer and the fraction of polymorphic fragments. So that way we will get effective multiplex ratio. These are the different parameters that will tell you how informative a particular marker is. If higher the PIC value for codominant markers, it generally ranges between 0 to 1. So higher the value, higher is the informative is the primer, and that can be used for different analysis. And so that way their values will tell about the informativeness of that particular primer that we are using it. And when we talk about dominant marker, the formula that we use is 2Fi to 1 in minus Fi, where I is the marker fragment that we are considering. Fi is the frequency of the marker fragment which were present. Because I have shown you the gels in some, if we are considering a row of band, so in some of them it may be absent. So the two terms that we use is monomorphic, polymorphic. So what does monomorphic? If we are considering a particular band and it is there in all the genotypes or accessions we are using, it is there in all the bands, so it is monomorphic. If it is absent in any one of the sample, we say it is polymorphic. So that way, frequency of the fragments which are present, and 1 minus F is the frequency of the marker fragment which were absent. So we calculate the PIC value for dominant markers like this, and its value ranges from 0 to 0.5. Like for codominant, the value ranges from 0 to 1, but here it ranges from 0 to 0.5. Now you can, other parameter, it's a very simple one, proportion of polymorphic loci. It is equal to number of polymorphic loci divided the total number of loci that you have calculated, then average number of alleles per locus, sum of the alleles divided by total number of loci. Then mean diversity is calculated by averaging gene diversity values over all the loci. That we have told earlier also, because you will calculate it, loci wise. Marker wise, like 2 primer pairs for SSRs, we say SRAP, 2 primer combinations, we say. So we say it as SSR locus or loci, also loci when we are using a plural term. So that way. So these are different free statistical softwares, and these are very good ones which we can use for genetic data analysis, like GenLX, PopGene, Harlequin, Structure, Darwin, Antisys. Antisys is paid one. So they are freely available and can be used and easy to perform also. So now I will tell you about, because for, you are using SSRs, you need SSR primers. So you have to design them. So earlier what we were doing, we were generating genomic libraries by enrichment method, and that was very tedious, time consuming. You have to do cloning and all that. But now in the era of genomics, you have huge number of sequence data. You can sequence it depending upon your funds availability. You can sequence your particular crop DNA, at low coverage, and also to generate primers. And that finally, whatever data you are getting it in this particular FASTA format. So that FASTA formats, they are used for generating SSR primers, forward and reverse primers. So there are different programs available which will find SSRs in these regions, and then they will design forward and reverse primers from the flanking regions. Those SSRs may be di, tri, tetra repeats, they may be perfect, imperfect. So these are some of the programs where they have the combination of both like one is the SSR finder and another one is the primer design. Generally we use primer 3 or primer 3 plus. So full SSR, Gemata, SSR locator, SAT, crate, and polymorph predict. These are different, primer designing tools available online. And then the primers generated, you can validate using just a profile to show like whatever primer. We run it on a set of 4 or 5 lines in a particular crop from where we have designed. So we run it at a particular temperature, depending upon the annealing temperature, TM of your primers that you have designed. When you design it, it will come out in that particular sheet itself. Now, you know what kind of size you are getting, what kind of, what is the TM, and accordingly, you can set your annealing temperatures and run your profile. So you can see like here, you are getting, we have taken 3 samples. It is same in that, but here you see that it is one allele is of this size, another at this size. So this is a polymorphic. So we will select these kinds of ones which are giving us polymorphic patterns. This is also polymorphic. This is polymorphic. This is polymorphic. These are monomorphic. So we want polymorphic primers so that we can use it for characterization, and we can find it the diversity in the particular crop samples that we are going to use. So this way we validate the design primers, and once we get such kind of primers, then we use it in a larger set to characterize that. I'll just take the example of one program, Polymorph Predict, Dr. Rakesh Singh, to which you have heard in the morning. So he is one of the contributor to this particular program. Polymorph Predict. It is available online. So just what you have to, this is the interface for that, and you can see options for uploading files. And here you have to give your email ID. Then only you would be able to see, the output. You will make a file of FASTA format from which you want to generate. I have just made a file of 6 samples for you to show like how it will come out. So there are 6 samples. It is just 2 in this panel I am showing. This 6 sample FASTA format. I've uploaded it in the text file here in the upload template genome. And then you can set your minimum repeats for mono, di, tri, tetra, and penta. And then just you have to, after uploading, after uploading, and you will click and you will get the results. So you will see, this kind of file. You can download your primers by clicking it. So here you will get results like this. So from the 6 sequences I have uploaded, only Trinity 3. This one is showing, repeats like CTC, GCG, GTG, TG. These kinds of repeats were there. This can be shown in this particular output file. Like what was the composition of those repeats, what were the type of repeats, di or tri, and these were the primers that were designed, and this is the product size that you will get, and these are the annealing temperatures also. So accordingly, you can design your this sequence. We give to different companies who get it synthesized for us. So we use those synthesized primers that I have shown you for running PCR products, and then we can validate. We'll say we have synthesized, 100 primer sets, so we go for PCR, and out of that we are getting 25-30 primers, which will give you result. Here you can see no amplification is being done. No. We can standardize it also further, but in the first go, what you should do, say you have synthesized 200 primer pairs, just run it in first go, and wherever you are getting, at that particular temperature, generally we do it at touchdown cycle, so that at maximum of the primers, they will get amplified at that particular temperature, and we we can select our first set. Further, if we want more primers from that, we see like these are not sufficient one, we can again set our annealing temperature conditions. We can see like whether they are amplifying it at another temperature, and then again we can see the left of the primers and use them if we get the result. Otherwise, we leave such primers and we go for the polymorphic primers. So I'll just show you the results of some of the work that we have done in our lab. This particular work has been done by Dr. Manjusha. She is working on cucurbitaceous crops. She has developed, draft genome from this Pusanutan rich code variety using Illumina and nanopore data. And then she has used a MESA program to find SSR repeats. And we can see like total number of identified SSRs are 89,000. And this is the distribution of different kind of repeats that she got in from the MESA output. And then primers can be designed, and these primers are being used for the characterization of ridge-goat accessions, which are available in our gene bank. This particular slide also, here the in this one we have used our draft genome sequence data, but here again she has used bottle gourd rat sequence data that was publicly available. So that data was used for generating for identifying microsatellites, and then they have been primers have been designed using primer 3, and then those markers, they have been also available at NCBI, and this is the composition of different kind of repeats, percentage of trinucleotide, dinucleotide repeats that are there in this particular microsatellite data set. This is the thing that we have designed markers from genomic sequences, whole genome data, or from red sequence data. We can also generate the markers from transcriptome data also. This particular slide is for finger millet. Like in finger millet, we have generated leaf transcriptome from 2 of the varieties, GPU28 and PR202. That sequence is sub-project is available at NCBI, and from there we have generated markers. Now, earlier markers, I say, because they have been generated from DNA, so we call it as genomic SSR markers, and here they have been developed from transcriptome, so we say it as genic SSRs, and because they are from the expressed regions, so we call it as EST SSR markers also. So in that we have designed around 3,883 genic SSRs, and from 56,404 unigenes, and those from that we have synthesized 839 and tested it in a smaller set, like 3 or 4 varieties, different varieties, diverse varieties, and then did amplification, and 679 could be amplified. Further from that, we have taken 231 and tested it in a 20 diverse genotypes, so that we can identify the polymorphic ones that can be utilized in larger set, and you can see the amplification profiles. Further, the markers generated in such way, we can transfer them into the other crops also. We tested it in proso millet, foxtail millet. This is just one profile to show like how this finger millet markers, they have been transferred to proso millet, foxtail millet, and sorghum also. This is just to see the cross transferability of these markers. And otherwise, in these crops like foxtail, sorghum, enough markers are there, but for proso millet we have not that much. So Further, like because the markers that we are using, whether RAPD, ISSR, AFLP, SSRs, we can use them for, first here, it is the development of markers. Then this slide will show like how we can use those markers for diversity analysis. You can use it for, any set of genotypes available with you. This we have done in finger millet mini core, which was 110 accessions, and we use different SSR markers. We could get 114 alleles, and you can see the type of pattern we are getting. And you can see like the variability of alleles is there, number of alleles are there, but not like much, because it's a self-pollinated crop. That much variation is not there. And in this particular mini core set, we have accessions from Africa, we have accessions from India, from Sri Lanka and Japan. So when we use such kind of molecular techniques, they give you information also how close these genotypes are, how diverse they are, and what is the relatedness between the genotypes which we have taken from different geographical regions. So if we see this particular dendrogram that we have got using anti-cis program, UPGMA clustering, and in this we majorly got this particular and this two major clusters, and this particular cluster was pertaining to genotypes or accessions which were from Indian origin, and here African, most of the African accession with some exceptions were there in this particular cluster. And Sri Lanka and Japan accessions, they were grouped with Indian and Ugandan accessions. So this kind of grouping, they will tell you how related particular genotypes are with genotypes from one geographical region to other region also, and what is the level of diversity as we see from this particular scale. A moderate level of diversity we could get in this particular set. So this was like, if we want to go for region-wise specific, we have collected it from a particular region and want to see the diversity level from different districts or from different villages that we collected the germplasm. This particular study we have done in foxtail millet. And here, we used genome-wide SSR markers. 25 markers were used, and the genotypes, they were particularly from, Uttarakhand region. And here we can see like with this clustering, we could see like the accessions from Champavat were clustered here. This is from Rudraprayag, Chamoli and Almora, then Tehri. And from this cluster, if we'll see, Pithoragarh, Rudraprayag and Champavat. From all these three regions, we have accessions. So from this we can interpret the genotypes which were closely related from that particular region. They are grouped together. And even from Champavat, this particular accession will be diverse one. That's why it grouped separately in a different cluster. So that way we can interpret our results, whether we are getting any, geographical kind of patterning, whether they are clustering depending upon their, area of origin, or if we have there any phenotypic or biochemical profiling data, whether that kind of groupings we are getting. So we can correlate our results with phenotypic data also. And one of our study that we have done, student worked on land races and varieties of finger millet. And if your accessions, your material, they have different biological groups. So that also you can see like how those biological groups, diversity levels are there. Like from here we can see most of the land races, they were grouped separately, and varieties got grouped separately. But some exceptions are there. Maybe those might have been used in the development of these varieties, because land races, they have different genetic structure, more heterogeneous as compared to varieties. So they are grouped in a separate cluster as compared to varieties. But we see in case of finger millet, if we see, because in this particular, most of the varieties, they have been developed from Karnataka region, and the land races are also from there. Though we are seeing like land races are more diverse than varieties, not much diverse, but yeah, they are more diverse than varieties. That may be due to because they have been taken from a particular a small area coverage was there from which these varieties were developed or land races were collected. So this is the way we can interpret our results. So this is just a population structure. I'll show how we can run this population structures. So this is also, telling us varieties are grouped differently in different group and land races are grouped separately. Then we can use this our this 0-1 data or our allele score data for population structure analysis also that we have done in CodoMillet. We have the accessions from 12 different states, and we have used ISSR, SCOT, SREP markers, different marker systems like neutral and functional, both were used. And we analyzed the data separately and together also. And we could find like maximum of the accessions, they were grouped in one cluster, and 18 of the accessions were there, which were diverse. They were 1, 2, 3, 4. These were the 4 different clusters in which, those 18 diverse accessions were scattered. So what I want to tell from this picture is that one, you can see this particular graph that is at 4. Delta K value is at 4. I'll show you in the next slide how we get that delta K value. So when we run this structure program, and results are loaded on to another structure harvester that will tell you like whatever germplasm that you have analyzed using different markers, then you scored it, then you have run it on a program structure. So that will tell you the distribution of, this is the model-based clustering, and that will tell you how your genotypes can be grouped in different populations. So depending upon their genetic makeup, they will get clustered, because here we can see most of the lines, they may be genetically related. They are not having much variation, so they are grouped here. And these 18 lines with both the marker system like ISSR as well as this with this profile is with the SRAP and SCOT combined. So in both the cases, we are getting those 18 accessions which are quite diverse from these rest of the accessions. So that's why they are grouped separately. So that way, we can identify the accessions which are diverse, and depending upon their phenotypic traits or other traits, we can use them in our breeding programs. Now, we can use the markers. These markers for hybrid purity testing. This is one of the work that we have done quite years back in sorghum, where we have used capillary-based sequences, ABI 310, for running our PCR products. Earlier gels, earlier pictures I have shown you, they have been run on agarose gel or metaphor agarose gel, but this particular picture is from a capillary sequencer, ABI 310. That time that was available, on that we have run. And we can, how we can use it, this particular profile, these are called as electropherograms. How we can use it when we run this profile and we score our profiles and see like this is a particular hybrid CH13 and its 2 parents are RS29 and 296. So when we run different markers, we see their banding pattern, we see their sizes, so we see their peaks and see if in the parents we are getting the polymorphic alleles, that marker we use for hybrid purity testing. So here you can see CH13 is the hybrid, RS29 is one parent showing 196 base pair band, and this another parent is showing 182 base pair band. And you can see clear-cut profiles, like in CH13 you are getting bands, both the bands which are contributed from both the parents. So such kind of markers, polymorphic markers, can be used for hybrid purity testing, and you can make a diagnostic set that can be used for testing, because otherwise if you have to go for purity testing, you have to go plant and it will take time to go do the grow out test. But here, just if you have seed, you extract your DNA directly from seed, or you can raise the seedling for 6 to 7 days, and then you can use it for DNA extraction and use your diagnostic set of markers for seeing the purity level. So if you have taken randomly, you have taken 50 seeds and taken your DNA and run it with those particular markers, and if say out of 50 you are getting 2 of the band profiles in which that hybrid is not showing the alleles which were there in both the parents, so that way you can identify the off types and say the percentage of impurity in those hybrids. So that is another thing that we can use our molecular markers, and we have used it in sorghum. So because in NPPGR, we are providing DNA fingerprinting services also. I'm working on millet group. So I am doing for that. And Madam Dr. Bansha, she is working on cucurbitaceous crops. So accordingly, she is providing those services. And so in this case, like for DNA fingerprinting, for the crops in which we have SSR markers available with us, we are using SSR markers only. But in the crops, wherever we don't have SSR markers, we are using RAPD, ISSR, SREP or SCOT markers also for DNA fingerprinting services. So the technique is same. You have to isolate the DNA. You have to, you should have primers with you. Then you have to go for PCR. You have to run the gels, and then you have to interpret your results, score your bands, and make a report. And that, we are why this DNA fingerprinting services is being done, because for the release of varieties, CVRC committee that need, this DNA profiles as a supplementary proof. So that's why these are required, and we give services to private and public sectors both. So, but the basic technique is same. The only difference is that, like for fingerprinting, whatever sample they come to us, we isolate the DNA, purify it, and then depending upon the markers available in that particular crop, say if we have SSR markers, we run those markers with reference varieties. So reference varieties are the varieties that we have selected in a particular crop, which are they are the national checks, or which have, depending upon our previous DNA profiling data, we have selected the varieties which are diverse. So those varieties we take as a reference, and we always run it with the sample that have been given to us for DNA profiling. So this is just a picture to show. This has been this is the sample that we got for profiling, and these are the reference varieties of barley that we have used. This is the picture from sorghum. Here we got hybrid also along with their parents, and a variety also for fingerprinting. So here are the reference varieties that we run along with the samples to be profiled. These are the profiles of sorghum that if we run it on a sequencer, but because nowadays it becomes costly affair also to do it always every time doing it on a sequencer. So we run it on metaphor agarose or 2.5% agarose, and with a set of primers that we have already identified, like these are polymorphic, these are having high PIC values. So for DNA fingerprinting, in each crop, wherever markers are not available, you have to first, take a diverse set of varieties. You have to profile them. You have to standardize your primers, which will give you a polymorphic pattern, more the high PIC value primers, and that we use it for fingerprinting. If with that, particular set that we have already standardized in different crops, we are unable to, DNA profile that particular variety. Not profile means we are getting similar patterns. We are not getting any, different alleles, different kind of alleles, which are the alleles which are not there in those samples. Particularly like the one we are getting, we always try to find like we get a unique allele or a combination of allele for that particular sample. So we run more samples also. Otherwise, we do it with 15 SSR primers, if any of the sample we get from public or private sector. So we can use this markers for species identification also. This particular study, we did it on Morinda crop. Morinda, it is a medicinal plant. It's a very useful, and it's juices, and they are also they are rich in vitamins and minerals. So this particular work has been done under WNRF project, which was run from Chennai. Chennai was the center for that particular, and we did it this work with Dr. Veena Gupta, and she collected material from different places. And here I will just show you how the ISSR markers, they can be used for species identification also. We were having two species, Morinda tomentosa, Morinda citrifolia, and Morinda tomentosa we got from Gujarat, Rajasthan, and Dharampuri region. This is from Tamil Nadu. And Morinda citrifolia samples were from Kerala and from Jodhpur. So we analyzed with the set of markers. Here I am telling you again and again, these are SSR markers. You can use it on, these are publicly available. You can use it on any of the crop and you will get such beautiful patterns. Only just you have to standardize your temperature profiles, and this we have done in Morinda. We have used these primers in different cucurbitaceous crops also, millets also, where at that time SSR markers are not available. And from this profile you can see like this particular band, it is there in all the Morinda tomentosa. These were from Gujarat and Rajasthan, and this particular one was from Morinda tomentosa from Dharampuri. So you can see this particular band is there only in Morinda tomentosa accession. It is not there in citrifolia. Similarly, we can see this other bands also. This particular band, it is there in these samples, not there in citrifolia. So these are species specific markers, and with this particular marker also you can see. So then we combine all the markers, we have run it using NTSys program, we have generated such profiles, phylogenetic tree, and then we have used structural program also, and we could get clear distinction of those genotypes which were collected from which belong to two different species. And here further, like this is the way like you can interpret your results. You use for each data set, you are using PCR, you are running gels, you are scoring gels, you are analyzing it, but you are getting phylogenetic trees or structure. The next step is like how you analyze your data. So when we see that after this clustering, we can see like this particular cluster just belonged to Gujarat. So accessions which were from Gujarat, these ecotypes, they are separated in a separate cluster. From Rajasthan also, here we were having genotypes from Bundi and Kota regions. These two were from Kota or Bundi. So they were separated, and all other were from the same place. Some of the accession, one of the accession from Gujarat, Kopatpura region, this was separately. This was not clustered here. So it might be the very diverse accession from these 5 1. That's why it is separated. And this is Dharampuri accession, which is not from this Gujarat or Rajasthan region, quite far from these regions. It is there in Tamil Nadu, and it is separated as an outlier, but it is there in Morinda tomentosa, cluster. And then we here also we got the accession from Kerala, and accessions which we got, Jodhpur region. While analyzing this population structure, you can see like in Morinda tomentosa, there were no admixtures. There were no admixtures, but in citrifolia, we can see these admixtures. Okay, so for like, because these citrifolia is there in the coastal regions, so some of accessions might have migrated or they have been introduced to these Rajasthan region, Jodhpur. These are from Jodhpur, and there might have been not self-pollinated, but maybe due to heterostyle is there. So we have just searched literatures like why this kind of admixture we are getting in the Jodhpur line. So we could find like there is a heterostyle in this, and because of that, cross-pollination might be there. Here ants and bees, they are the pollinators, so they might have cross pollination might have occurred, and this particular citrifolia population got diverged. So this way they are now they are adopted to a different region, and so such kind of accessions we should collect, because they have adapted to a different region, and due to crossing over diversity is generated in this particular set, which is different from the accessions which were there in Kerala. So such kind of inference we get from our results. If we see them thoroughly, properly, and correlate with our earlier available information, or we have to think it how to interpret the results. Now I will just a minute, I'll just take water. Hello? Hello? Hello?
Yes,
ममता?
बोलिए मैम.
Yes, ma'am.
ओके, ऐसे लगता है हम सब सो गए.
No,
तो
no,
और
ma'am.
मैं भी बात करते करते थोड़ा पीछे हो गई, तो मुझे ध्यान आया कहीं मेरी आवाज आप लोगों तक पहुंच भी रही है या नहीं,
नहीं, मैम सुन
because
रहे हैं
it is
मेरे
the
ख्याल से.
difficulty when we speak online.
No, everybody is listening, ma'am. I was just on a call. That's why I delayed
Okay,
response.
so
also.
now I'm coming towards the end of my presentation. I'll just tell you how we use Darwin program, which is there, freely available online. So, here in the rows, we have entered our genotypes, and here we have entered our data. This is the 0-1 data. So primer by primer you can add, and then you can club all the primers. And then you have to save your files as text tab delimited. So this is how it will look like. So this will act as an input file if you have 0-1 data. And this way you can put your, SSR data, and this way you can put your sequence data. So all these data can be used for, generating phylogenetic trees in the Darwin program. So I have just run it on a small set for you. So this is how the input file looked like for all the 3 marker systems. Then this is the interface, and we go to the file and import it. So here you will select the particular file that I have shown you, which has been saved as text tab delimited. Then if we have 0-1 data, we have to select the single data. If you have allelic data, you can select that one. And if you have missing data, in this data, I have put 9 for the missing data. So where we couldn't get any band. So, and then we save it as codo.var file. After that, what we do, like it will appear like that. And here, this var file is used for calculating dissimilarity. So this is the input file. We save it as dis file. And here another option is there, missing data. So you can set it, and you will see, like if we click it, we see complete unit deletion, complete variable deletion. We don't want that, because otherwise units are removed or one particular variable is removed. So we selected pairwise variable deletion and set the value to minimum. And missing data code is 9. So whatever putting there in the Excel sheet, so that you have to put there. If you are putting 99, 999, so put it, that same should be there in there. And here in this particular sheet, it was 74 were the genotypes or accessions that we used for analysis, and 188 were the total bands. Okay. Then we have the option for different coefficient, dice, Jacquard. So we generally go for dice. So we generally go for Jacquard coefficient. So we can select that. So if we select that, so now after that, you will see this missing data option that we have selected. It will show units, variables. And then you have to save dissimilarity matrix as. You will just click it. It will come out as codo.dis. It will be saved there. And this particular we are doing for 0-1. 0 is for absence of the band, and 1 is for presence of the band. Then you have to say okay. And after that, we have to go for tree construction. And here the options are there, weighted, unweighted, and we have selected unweighted neighbor joining. And draw tree, when done, we have clicked it. We can click identifiers also. Identifiers means, like here, otherwise you will get numbers 1 to 74. But if you have put accession names, so if you select that identifiers, and the DON file is created, so you will get in the next slide, you will see, you will get the IC numbers also. So then you have to say, okay, it will be saved, tree will be saved as codo.arb, and when we see okay, draw a tree when done, so you will see such kind of tree after the analysis is complete. Now, if you want to put names to these numbers, so what were the IC numbers? You can go to this particular identifier tool. Here you can click on identifier, and it will select the DON file where from where it will take identifiers. That is the name of your accessions, or in whatever way you have put. So next you will get, so this is the tool. From here we have picked. So you will get, you have to just click on the identifiers, and these will appear on the screen. So here you can see like those 24 accessions, they were from Codomiller. They were separated into 2 major clusters, and there were then smaller clusters. So you can just see your data because you might be having that passport data from where which particular regions those have been collected. Then you can correlate, that data with this clustering pattern, or you can, if you have phenotypic data, you have their protein content measured, or you have their days to 50 flowering you have done, or you have calculated their 1,000 grain weight. So any phenotypic data or their genetic origin data is there, you can correlate it with your clustering. So what kind of grouping you are getting, and how you can correlate it your other phenotypic or geographic data, and you can interpret your results. So this is where it is very simple to run Darwin. So it is freely available. And here in that I have used neighbor joining. You can use, hierarchical clustering, UPGMA also. In that you select UPGMA and you will get such kind of clustering, clear cut too. And here the same data was used, and you can see like earlier you said this small one and the bigger one, two major cluster, and then we have outlier kind of things. So these are those. So this is just a, different kind of representation, and in most of the cases you will get similar groupings. And whatever you want to select, depending upon how you want to interpret your results, how it is correlating with your other phenotypic data, you can use that particular clustering pattern. And this is the dissimilarity matrix that look like. And now I will tell how we can run the structure program. So this is the icon of structure 2.3.4. This is the interface. And here also, we can save our data as text tab delimited. This is how that file look like after we save it as text tab delimited. And then you have to go to file, and here you will run a new project, and you will see this particular window. There you have to name the project. This I have done on Codo Millet, so I have just given name Codo Pop. And then you will, because you have stored your text tab limited file in a particular folder, so you'll make a folder. So you will browse and keep that open that folder, and from there you will select that particular text file. Then you will go to click on next. And then you will see this particular window. Here you will fill the numbers. Number of individuals were 74. Ploidy, because you were taking 0, 1, put ploidy as if you have deployed. If you have taken allelic data, you can put it as 2 deployed. For tetraploid, accordingly you can put the number. Then it is the number of loci, and it is the code for missing data. We have put it as 9. And then we will again click on next. So it will show these windows. So you have to click on row of marker names and individual ID for each individual. So after that, you will, get this window. You will click on proceed. So then after that, you have to set your parameter set. When you will click, you will get length of, you have to fill length of Bunn period and number of MC rep. So just for experiment test file, I have given 1,000 for Barnen and 10,000 for MCMC. But when you are actually running your experiment, at least keep Barnen period 10,000 and MCMC value replication, you run it for 1 lakh. And then say okay, and you have to give the parameter name also. Again, okay, you can give the same name. Then after doing all these parameter setting, we will go to the project and start the project. So you will get now one more window, like where you have to select this file, and you can set your K values. K values like how, your total accessions, they will get structured in different populations. So we have given 1 to 10, and then number of iterations, we are giving it 3, like it will repeat that 3 times. And then again, we will click on the start. As soon as we click on the start, so this program will start running. You can see this in black window, some numbers are coming. So they will, they will be in a scrolling mode. So that's how structure program runs, and it will take time depending upon how many, runs you have given, 1 to 10, how many iterations you have given, and main thing like how many value for Bunn period or MCMC reps you have given. So that's why after that it will, When the run is complete, project is complete, you can go to the file and save it. And you will see results in, such kind of files. You will see plot data, result. So these are the files. So what we can do now, because we have run the program, but we want to see our result, like what we got after this analysis. So we have to take this file, result file, and we have to compress it, because there are different files for K1, K2, K3, and that also repeated. So we have to make a zip file, and then you can go to the online, you can put structure harvester. So this kind of, screen you will get. And from here, you have to choose the file result zip that we got from population structure. So it will be uploaded, and within a few minutes, you will get the result. And how the results look like, a particular sheet you will get. And from there, you will get, this kind of peaks, delta K value. So, and this particular, like for 1 to 10, what are the different delta K values you are getting. So wherever you are getting a maximum delta K value that is set for your population. So that means for your this particular 74 accessions that we have taken, we can group it into 2 populations. So that will give you a exact partitioning of you will get the actual structure that is there in those genotypes, depending upon their relatedness or dissimilarity. So then again we can go to structure. And here we will click on the results. You will get these kinds of different runs, because from delta K, we could define like our genotypes could be arranged in 2 groups. So we will just check the take K2 value and click it on, and we will get this window. And from here, we can sort by Q, or we can plot in a single lane or plot in multiple lines. So this kind of, plot we will see, and you can see these particular accessions. When we do it in line, so all, line, it will come in line, just that I have shown for Codomillet also. So you are getting lines, and their numbers will be there, that I forgot to add that particular slide. So those numbers will be pertaining to the genotypes that you have analyzed during population structure program. And from there, you can find out what are the genotypes which are lying there in this particular, and what are there. So these might be different. That's why they are partitioning in different groups. So depending upon the structure of your population, you will get number of groups in which your population is structured. So that's what in this particular time I could manage to convey my message for molecular characterization of plant genetic resources that we are doing here, and you people also can do at your stations, at your center. So thank you all for your patient listening, and I want to thank you, program director, our director, ICR NPPGR, Dr. GP Singh, for giving me chance to interact with you all. And also I sincerely thank conveners and co-conveners of the programs, and Dr. Mamta for introducing us. I thank all the participants. Thanks, Manjusha, also for working with me. Thank you so much for patience listening. Thank you. Thank you so much. Now I'm unsharing my slide.
Yes, ma'am, sure you can unshare. And thank you so much for consistently speaking for long 2 hours without taking much gap or breaks. So, and within such a limited time, that was an exhaustive lecture that you gave. And I hope that because you started right from very basic to the some of the analysis, statistical analysis part that is actually required for most of the work to be actually interpreted in molecular works. So I hope that this explanation will definitely help our audience. So in this continuation, I can see that there are some of the questions. I think already Madam Manjusha has answered one of the questions. So for you, I will read that. So yeah. So the question was how to make good, it is from Dr. Vinod Kumar from JNKV. So how to make good breeding program molecular phenotyping conventional or conventional, which is, I think he wants to ask that which one is good, conventional or molecular phenotyping, which one is more effective. So Manjusha ma'am has replied to that question. A comprehensive phenotyping and molecular profiling is optimal. If one has funds, both are opted. In NBPGR, we do both, and we also have projects where molecular cores are being put for germplasm evaluation. Ma'am, anything else you want to add up?
Yeah, it is good because it all depend upon, nothing is like we can't adopt any particular technique. Like if we are doing phenotyping, that is sufficient, or we are doing genotyping, that is sufficient, because a comprehensive approach will give you a more reliable data, and you can complete your program in a faster, time period. So both the molecular as well as your, without phenotyping also, the molecular data is of no use. And without molecular data, like, yes, with phenotypic data and biochemical data, you are doing it for years, you are doing it at multi locations, you can identify your diverse germplasms and use it in breeding program. Yeah, but to make the process faster, if you use molecular marker techniques, you go for sequencing and use it for finding more diverse or trait specific germplasm. So that will certainly hasten it will speed up the process of your breeding programs.
Okay,
and you can correlate with your marker traits with your phenotypic data also. You can find out marker trait associations, you can find different novel alleles also, and further you can use this genotypic data for a number of purposes. So certainly if you have option to go for both, you should go for both.
Okay, ma'am. Now the next question is from Dr. Vikas. He is asking, what are the charges of DNA fingerprinting at NBPGR for hybrid and its parents?
Actually, for it is done at per sample basis. For private sector, it is 10,000 per sample for 15 SSR markers and 5,000 per sample for public domain. So it is per sample. If we
it's
do
under revision also right now. It is under revision also right now, so it will be updated on the NBPGR portal.
Yes.
Any other questions from participants side, please? I hope everybody listened to ma'am. It was a very nice presentation, at least for clearing the concepts and to move forward their work, because an analysis part is actually very important, ma'am. Where most of us feel quite difficulty. So related to that, if any question from participants, and if no, then
you can all the participants, you can interact with us anytime on phone also. You can put a mail also, and all of our scientists here, they are helpful. And if you have any difficulty running any, even wet lab experiments or running, your statistical programs, you can interact with us, and we are always there to help you all.
Okay, ma'am, thank you. And I would like to thank Dr. Manjusha ma'am also for being available for this long. Still, one more time, I would like to ask participants if there is any question, so we can put it in front of our speakers. Otherwise, we will end up this session. So I can't see any question or any question coming up from the participants. So ma'am, should I end this session here? And thank you so much. Maybe the lecture was too long, but it was informative.
Because it was a practical session, so I thought to include, whatever basic or, techniques and their protocols or statistical analysis, so that it may help the participant. I try to include those, but if you have to actually do the practical when we are, physically present there at a particular place, so that will make make it more useful. But I have tried my best. Thank you so much, all of you.
It was very nice.
Thank you, madam.
Thank you.
Thank you, ma'am. I hope that will be
Thank you, ma'am. Thank you.
beneficial.
Thank you. Thank you. Namaskar.
Thank you, madam.
Thank you very much.
Ma'am, if you wish, you can. Yeah.
So we are leaving now. Okay.
No, actually, we have one more session to be started from 4:30, ma'am.
Okay.
So, if you wish, you also can continue. You can
Okay.
attend, ma'am. Yeah.
Who is the next speaker? Lecture?
Dr. Dubey, sir.
Okay.
Actually, Dubey, sir. So, we'll start from 4:30, ma'am.
Okay.
Still time is there. So,
uh-huh.
anything else if you
7-minute
want to
break
say?
is there. We can they can take a break.
Yeah, they can
Because
take
they were
a-
also sitting along.
yeah, they have to prepare
Okay.
themselves for next
So
session.
I would like to say thank you once again to the project director of this program, Dr. GP Singh, Dr. Jyoti, Dr. Tripathi, Dr. Mamta, and all of your team who is organizing this particular training program. And thanks to all the participants for listening us, hearing us, and interacting with us. Thank you, Manjusha, for being my co-partner. Thank you so much. I will take your leave. Thank you so much. Bye bye.
मैम एक मिनट एक बार सभी अपना वीडियो ऑन कर लें फॉर अटेंडेंस। आप सभी से अनुरोध है कि आप लोग अपना कैमरा ऑन कर लें प्लीज।
Participants, please switch on your cameras.
ऐसे सही आएगा। नमस्ते।
थैंक यू वेरी मच। थैंक यू।
Thank you, sir.
Thank you, Dr.
Thank you so
Mandal.
much.
Thank you for coordinating this program. Thank you so much.
Okay, so this is for the participants. We still have 5-6 minutes, so 5-6 minute break is there. You can refresh yourself for the next session. We'll be starting with the Dr. H.C. Dubey's lecture from 4:30. So I request all of you to please be back sharp on time. So we'll meet at 4:30.
हाँ मैडम।
हाँ सर, मैं आऊँ क्या नीचे?
नहीं, यही बता दो, आप बताते जाओ, मैं करता जाऊंगा। स्क्रीन शेयर तो अभी कर ही सकते हैं ना, उनका जो बायोडाटा है।
हाँ कर सकते हैं।
हाँ तो बताते जाइए, आप वो ऑनलाइन बोलते रहिए। मैंने उनका पीपीटी खोल रखा, मैंने उनका। स्क्रीन शेयर करूं?
स्क्रीन शेयर कीजिए।
ठीक है, फिर ये हाँ बायोडाटा दिख रहा है, मैं उसको क्लिक करूं? हाँ ठीक है, हो गया। हाँ हो गया। हाँ ठीक है, ये तो हो गया।
स्लाइड शेयर में आया नहीं है सर अभी।
हमें तो दिख रहा है ये स्लाइड शेयर होके। यू आर स्क्रीन शेयरिंग। स्टॉप करो।
स्लाइड जो आती है फुल
हाँ मेरे में फुल कर दिया मैंने अभी।
प्रेजेंटेशन मोड में नहीं है अभी।
फिर ये क्यों नहीं आ रहा? जस्ट ए मिनट, मैं पहले उसको वहीं कर दूं क्या? जस्ट ए मिनट।
नहीं, वहां करेंगे तो नहीं हो पाएगा, फिर यहीं से कर दीजिए।
हाँ जस्ट ए मिनट, मैं वैसे ही कर रहा हूँ। जस्ट ए मिनट, स्क्रीन शेयर। हाँ डॉक्टर साहब एक मिनट ना मैं खुद ही सीख रहा हूँ ना। हाँ ये आ गया, अब ठीक है, अब ये हो गया।
एक मिनट से मैं चेक करती हूँ।
देखना मेरे में तो हो गया है।
नहीं, अभी भी नहीं है।
कुलदीप आ गए मेरे पास हेल्प करने के लिए।
अच्छा, आगे चलिए।
आ गए हैं।
ठीक है सर।
ये मैंने ऐसे ही किया था। हाँ क्यों नहीं दिख रहा लोगों को? हाँ ये स्टॉप हो गया, ये मैंने पीपीटी खोल रखा है। हाँ
हो गया था अभी थोड़ी देर पहले, लास्ट टाइम।
अच्छा हो गया था।
हाँ।
दोबारा करोगे? दोबारा स्क्रीन शेयर। हाँ हो गया।
नहीं, एक मिनट वेट। हाँ हो गया।
हाँ इसका मतलब हम लोगों को पहले स्लाइड मोड में पहले स्लाइड में कर लिया। हाँ वो हम ठीक है। ओके थैंक यू।
गुड मॉर्निंग डॉक्टर साहब।
गुड आफ्टरनून सर।
अरे सॉरी, गुड आफ्टरनून।
हाँ कोई नहीं, मॉर्निंग ज्यादा अच्छा है। वी आर फ्रेश, वी आर फीलिंग फ्रेश।
हाँ वो यहाँ दिन भर काम में मॉर्निंग दिखता है, 6 बज जाता है तभी।
गुड आफ्टरनून सर।
हाँ नहीं कोई नहीं, कई लोग तो ऐसा है वो मॉर्निंग ही बोलते हैं हमेशा, किसी टाइम भी मिल जाए।
जी।
तो सर कैन वी स्टार्ट? इट्स ऑलरेडी 4:30.
Yes, you can start.
Dear participants, are you listening me properly?
हाँ सर आ रही है सर, अच्छी आवाज आ रही है।
Okay. Before we start, I would like to introduce today's renowned speaker, Dr. Sunil Chandra Dubey. He is presently Assistant Director General Plant Protection and Biosafety, ICAR, New Delhi. Before that, he had headed the Division of Plant Quarantine, ICAR and BVGR, New Delhi. And earlier, he was Senior Scientist Principal Scientist in the Division of Plant Pathology, IRI. And earlier also, he was Assistant Professor at Birsa Agriculture University. He has immensely contributed to the field of plant pathology. In this one, he has contributed significantly in characterization, diversity analysis, DNA barcoding, diagnostic, host pathogen interaction, race profiling for fungal pathogens. Not only that, he has developed bioagents based integrated management technology for many crops, and he has also developed one novel bioformulation, PUSA 5SD, which has got the patent also, and it is based on Trichoderma hergianum. And he has monitored quarantine processing of plant genetic resources, including transgenics, for research purposes, and provided technical and policy inputs on phytosanitary issues related to biosecurity and quarantine to various stakeholders. And he is credited with release of 15 varieties in rice, chickpea, mung bean, garden pea, which is having a resistance in the background. And he has registered the germplasm of chickpea, urd bean, cowpea, lentil. Not only that, he has reported about 600 partial fungal gene sequences to NCBI Gene Bank. And he is recognized by different honors and awards. He has been recipient of Hello, kindly mute yourself. Yes, can I go ahead? Hello.
Continue, sir.
Yeah. He has been recipient of Dastur Memorial Lecture Award and Professor KPV Menon Award. And he also been bestowed with INSA Fellowship and Fellows of National Academy of Agricultural Sciences, New Delhi. And he is also recipient of National Academy of Biological Sciences, Chennai, IS, MPP, Udaipur, and IPS, New Delhi. He is editorial member and office bureau of various professional societies and journals. He has guided 21 students, and he has been actively involved in the teaching program of IERI and Birsa Agriculture University. And he has published 225 research papers with H index of 27, I-10 index of 79. Not only that, he has presented 160 papers in national and international symposia, contributed to 33 book chapters, 45 chapters in practical manuals, 3 books authored, 10 books edited, and 41 popular articles, 7 technical bulletins. Here participants, I was the senior scientist at Almoda, and he was senior scientist in the division of plant pathology. First course which I attended on host plant resistance, I attended and learned many things from him. I used to refer his IPM practices, technologies, papers. So he has immensely contributed to the science of plant pathology. I again welcome Dr. Sunil Chandra Dubey to this fora, and I am very thankful that he has spared some time and even adjusted time for this fora. And I request him to speak on plant biosecurity in India, emerging issues and challenges. Very new topic, and what are the needs, what are the gaps in biosecurity system legally. Still now India has to do, and he is such a position, he can influence the policy of the government. And I think he is trying, and he will elaborate all those things related to biosecurity in India. Thank you, sir.
Thank you.
Welcome, sir. And
Thank you, Dr. Hodaji, for very nice and elaborated introductions. And thanks to the UN organizers for providing me opportunity to be here. And it is always pleasure for me to be associated with any activities in NBPGR and IERI, both the institutes. Let me share my screen.
In the slide mode, sir.
Yes,
all right.
It is going slide mode. Okay.
Yeah.
So the topic which has been assigned to me to speak on the biosecurity issues and challenges in Indian context. And this is the very, very emerging topic. Always it is evergreen topic, rather we can say for biological science, this is very, very important. And as far as the definition of biosecurity is concerned, the different persons, different organizations defined in different way. Some of the definitions I would like to mention over here. In agriculture sciences, normally it has been defined at the encompass policy framework to analyze and manage the risk of food safety. And what are the risks? That means when we are talking the manage the food risk safety, later on I will show in one of the slide what the international regulations come under this. Animal life and health, plant life and health, and environments. So this 4 component has to be holistically managed for securing the biosecurity of any country. In WHO definition, if you can see, because they are mostly concerned with the human and animal diseases, so they are defining as a institutional and professional security measures designed to prevent the loss, theft, misuse, diversion, and intentionally release of the pathogens and toxins. Because the toxins, hazardous chemicals are also have the same concern at the pathogens there. So whether they are in a deliberating use or their misuse has to come in the eyes of the WHO as a biosecurity concern. In plant production concern, if you narrow down it is to protect the countries against alien pests. Only it has to be narrowed down. So even if you see the animal sciences, it is the one of the defense against the biological weapons has to be considered. So different way it has to be defined, but the main objective has to be protect the environments, human, plant, and food security, and all this comes into the biosecurity. So ultimately it will leads to the, if stringent biosecurity measures has to be implemented, it leads to the sustainable agriculture productions, safe food production. Safe food means there is no harm, though residual toxicity, no toxic, all these concern comes under biosecurity. Environmental production, it is because simultaneously we have to the biodiversity concern. Conservation of biodiversity is one of the very major component of the biosecurity. This will be directly related with the introduction of any alien species which are danger for a diversity available. I think this is the another important component where enemy PGR is dealing. If you look the basic elements, what are the area has to be covered? If introduction of exotic pests and diseases. Sometimes what happened, pest has already been introduced or present in our country, but maybe there is a abnormal change in the virulence pattern. It becomes a new race, and pathotypes or biotype that also has to be considered, because it may be the economically important or to becomes for a indigenous crop productions. Deliberated misuse of pest and pathogens, maybe they are importing some things or maybe modifying COVID. We have seen there already. Biosafety issues arising to MLOs, because in our country MLOs are considered as a pest, where it has not been approved by the government of India for the cultivation or use. So that's why the biosafety concerns related to MLOs also come in the biosecurity. And we are all knowing why we are discussing today at this topic, because due to the open border between the country, the bioterrorism has been increased nowadays. It becomes a global concern, and this is basically 2-3 reasons by which it has becomes mandatory for all the countries to protect the interest of the environments and human and plants being of their own, because the technology, as the advancement of technology is going on, we are bound to do the new research, we are bound to use the new technology, and there is the always chance for the development of the new pathotypes, genotypes, biotypes, whatever may be the super race or some things you can say. And simultaneously there is during the experimentation, there is also chance of the increasingly use of the new hazardous compound development and use of hazardous compounds. So these to protect the country, these two bad effect of or we can say this dangerous effect of these two, the country has to be secure. If you look what we have in definition we have discussed earlier, this biosecurity, we have the 4 sectors normally: human life and health, including food safety has come under here; animal life and health, including fishes also comes under this; environmental protection and plant life and health, including forest. So this 4 sector we have to formulate a regulation, we have to take the steps, we have to do all the processes by which we can protect all these 3. That means the entire environments, entire surrounding has to be protected. And this cannot be achieved individually. This cannot be achieved by a single organization working alone. So that's why the internationally long back 2006 in FAO had designed very robust procedure that is called an integrated biosecurity approach, where all these stakeholders, farmers, institutions, private companies, NGOs, other governmental organizations, everyone, those who are dealing with everything on the earth. You can see here the animal, plant, environments, everything. So all the organizations, all the sectors, those who are involved with the activities of these 4 elements comes together, join the hands and develop a regulations or whatever the regulation in their own ministry are there, harmonize them in such a way that any security lapses respect to the biological material should not be there in the country. Then we can achieve sustainable biosecurity. It is very difficult, alone anyone cannot be achieved. When we sectorial approach we can achieve, then what happens? Public health will be improved, enhance international trade, that is very important. We can protect our environment, improve the agriculture productions, along with the conservation of biodiversity, which is very important concerns nowadays. And if you look internationally, and different biosector, that means for the human, animal, environments, different international organizations, those are working. For examples, you can see the food safety, we have the Codex Alimentarius Commissions. For UNES and animal health, we have the World Organization for Animal Health, OIC normally it is called as. And for plant health and plant health, including quarantine, we have the IPPC, International Plant Production Convention. And they are the standard setting bodies for the plant health. And biosafety related to plant and animals, the same LMOs, we have the Cartagena protocol on biosafety. This will also relate to the food safety, where the chances of LMOs and all these there. And CVD is the invasive alien species. So these are the certain, you might have been taught about these organizations and setups. I'm not discussing, we are not going to detail on these organizations, but in all sectors take care of, this is the international organizations. And little bit being a students of the plant productions, I would like to mention over here, these are the 3 standards, international standard setting bodies, which will take care majority of the sector in the biosecurity. They are called as a plant, 3 sisters. For animals health, as a previous slide OIC I have shown you, the plant health IPPC is there, food safety codex is there, and they all together, they are called as the standard setting organizations, because the international standards has been set by these 3 organizations in respect of their areas. And these each country, those who are the partner or signatory of these international organizations, they have to develop their own national standards to implement the international standards in a harmonized way to protect the country. That's why they are the very, very important. And IPPC letter, we all are knowing, being those who are involved in the NBPGR or plant productions. This is the multinational treaty where the presently 148 countries, India is the signatory, the initial signatory of this IPPC. And presently we have the 47 standards of the different actions, different, just like for the movement of the, even the movement of the vehicles, assessment of the losses, import of any living materials, all these things related to the biosecurity and plant productions in general we have. And its aim, if you look the IPPC aim, it has to prevent the introduction and spread of the pest, promote fair and safe trade, and protect the plant life. Why I am mentioning here, in some of the cases people have thinking or have the misinterpretation that this IPPC, the standards are prohibiting the international trade, but they are not prohibiting, they are facilitating the safe and secure trade in agriculture. Certain factors, those who are influencing, always we are bound to face these factors. That's why the need stringent biosecurity regulations required in our country. Globalization is one of the factors, we all know this. New agriculture production and food processing technology, when as much as we are advancing using the different technology, I mean GM and others, manipulation at the genetic level, definitely there will be always threat. And this COVID, everybody is knowing, this was the experimentally, the organism has been changed and it has been released into the environments. Increased trade in food and agriculture, it always chance of introduction of something new into the country. Legal obligations, what we have to do, increase in travel and movements, advance in communications, greater public attention to the biodiversity and environment and impact in agriculture. Shift from the country independence to country interdependent for the effective biosecurity. So most of the country, that means always it is not essential that all the countries should have their very robust biosecurity. So sometimes they are dependent. So suppose the quarantine, if it is required at the place of crop grown, or you have to simply believe on the documents which are attached with the consignment or whatever the agreements, you don't have the stringent mechanisms to take observation at the point of origin and some things like that, then definitely there is always risk. Scarcity of technical and operational resources, this is almost all the country including India is also facing. Food imports, this dependency on the food imports, as much as import is going on, definitely you need a very robust system for examination and inspection. If you don't have the, there is always chance, increased chance of the introduction of some things which is very dangerous for the country. Food means not always animal food, plant food. This is animal food and any kind of the food. If you look the national legislations, we have the Environment Protection Act 1986, Biological Diversity Act 2002, Indian Wildlife Protection Act 1972, the Forest Conservation Act, and Plant Quarantine Import Order 2000. Other different seed related orders and different orders are there. Almost all the orders and regulations which are related to the animal health, plant health, environments, all together, even sometimes human being health also, all has the very strong component of the biosecurity. But unfortunately, very stringent coordination, very effective coordination among these regulations not there. Presently, the governments try to make a coordination. Subsequently, in the few of the last slide, I would like to speak on this. You can see, since I am plant pathology, so biosecurity has to be much more discussed today in relation to the plant pathogens and plant pests. This is the exotic pest, very long list of exotic. We have all the regulations which have been shown over there, especially plant protection quarantine regulation. But even then we are intercepting, our countries witnessed several pests which are introduced from the different part of the world in different years. And this is the main reason. This is the very common slide. All the plant production scientists, especially those who are working in the NBPGR, is showing this pathway of introduction. This is the legal pathway. Beside the deliberated use is something else. But this is the legal pathway. There is also very, very high risk of introduction of the any kind of the pest which has the biosecurity concern. So we either we can import in bulk consignment for consumptions, where the Directorate of Plant Production, Quarantine and Storage is regulating and monitoring, and small samples including the transgenics, where this is a small sample of germplasm where the NBPGR is. In both the cases, we have the very, very high risk. In bulk consignment, normally is supposed to don't have the risk, but definitely since here is the material is very precise, very diverse, that's why the risk is there. And the material is huge. That's why maybe the low level of infection, but even then the risk is there because the quantity is very high. And this is the setup. Maybe in quarantine, scientists when have delivered the lecture, I would not like to speak much more on this. But the entire country we have in respect of the plant, we have the very robust system. Whereas this is called as a plant quarantine system. And we have this is the Apex Ministry of Agriculture and Farmer, where Apex bodies, where the Department of Agriculture and Farmer is looking. We have the 66 plant quarantine stations across 7 regional stations. Red color, they are the regional stations. And we have IPM Center, Locust Control, Central Insect Lab, Regional Pesticide Testing Lab, and headquarters in Faridabad. Besides the ICR institutions and SAUs, those who are providing the technical backup to these organizations, although they are also doing little bit research, but not their mandate are not very, very sophisticated research, but they have to intercept, they have to draw the sample, they have to examine the samples. So they have to equip with the basic infrastructures to protect our country, to avoid the introduction of any new exotic pest and diseases. But even then, entire national agriculture research system, including non-agriculture system, basic sciences, are also supporting them as per the need based. Our country has to threat with the two, 3 ways. If you can analyze the threat from the new plant pest, there are the certain pests which are new to our country. They are maybe because nowadays the fast movement, very, very, very strong network of the movement of the plant-based commodities across the world. So the distance is not a matter. But of course, if any pest is neighboring country and its mode of spread inject our air, definitely we have the very likely to be very high chance for introduction. That's why there's always threat of the new pest, and every year 1 or 2 new pest India is witnessed. Threat from resurgence and emergence of the plant pest, which is already over there, but they may become widely distributed and maybe epidemic form. Sometimes it become very serious, certain changes. And third category is that maybe the whatever the population is there, but they may be changed, genetically modified and more virulent. But no systematic study in our country. In majority of these cases, third category is being done. Simply we are saying this is introduced. I don't know whether it has been introduced or not introduced. Just like the banana wilt is one of the very classical examples, because long back our banana institute is saying that we have the race 1 which is becoming virulent and going to Cavendish. After 3-4 years they started saying that the race 4 has been introduced. Several times I asked with them, where has gone this population which we have published 3-4 years back? The race 1 is changing. So there is a very stringent and systematic studies required to prove whether our population in the country which is already exists has been changed to become virulent in a new form, or it has been introduced from abroad. So pathway analysis and all these scientific evidences are required, but the concern is in the eyes of the biosecurity, whatever the changes has been made, whether it has been introduced or it has been developed in our country, if it is threat to the flora and fauna, definitely our great concern is there. Certain laboratory biosecurity measures and generic mandate of biosecurity at national level has to be followed. In generic laboratory level, because there is a biosafety and biosecurity, both initially they are running parallelly. If you see the biosafety means these are the discipline which is addressing the safe handling of the contaminants and infectious microorganisms and hazardous chemicals. So in the laboratory, when the pathogen is inside the lab, biosafety concerns are there to protect the human being, those who are working inside the lab. But the moment which it comes out the lab, the biosecurity concerns apply to them, to protect the environment, to protect the population, so on. So there is the biorisk management, which is very important at laboratory level. And unfortunately, in most of the lab in our country, 99% we don't have the very robust biorisk management options. Regulatory updated inventories with the storage locations. Majority of the cases we are failing this. How many scientists is working, what they kept and where they kept. This inventory is not very, some things been come out, some things has been released into the environments. They may be, suppose maybe you have imported some of the seeds which are very fast growing, fast multiplication, large number of seed producing. They have the capability to dominate in the existing species, and by you don't have inventory, and anyhow it has been released into the environments. Due course of time, it will be threat to the biodiversity. So that's why always this has to be maintained into the lab. Identification and selection of personals with the SS. Who are the persons where it has to be assessed, such kind of the materials where the always risk is there, biosecurity and biosafety risk is there. Plant of use of microbiological resources. This is the also concern. Clearance and approval process. What would be if sharing anything? In this level we have some of the things, but majority of the cases we don't have. Documentation of international and external transfers within and between the facilities. Sometimes what happens, the facility is also being transferred. This maybe leads to the spread and introduction of the things from one areas to another area. And disposal, this is very concerns. Very sorry to say, we are also have gone in the same way. We are also not taking care of the disposal of materials. In 99.9, rather I can say, disposal mechanism is very poor in our country. This has to be although in the lab we have the different colors dustbin, but stringently we are not following even in the national institutions like IRI and BPGR and so many. We are disposing even our gel, which is stained with ethidium bromide very roughly here and there. So this has to be very great concern. We are talking only, but we are not doing any concrete for this. At the generic mandate of the biosecurity, protection of human health and consumers confidence, for this is for the agriculture food safety is required. Protection of agriculture, forestry and fisheries, this is a system based approach has to be taken. Protect the environment, including indigenous plant and animals. They will say the protection of environment simply not in climate protections. Whatever the indigenous plant is there and animal is there, that has to be also this way you can understand how much big canvas of the security is there. Advantage of trade opportunity and demonstration to importing countries to appropriate level of protection is there. Utilization limited resources and food safety element. That means we have to secure the food, animal and plant utilizing the limited resources what we have. Provide a cost effective and efficient government services to private sector producers and processors. This is very important. Very good coordination is there, should be there. Meet the obligations under international agreements. Protect against uncertainty associated with the new technology. This is especially for GMO and others technology. Whatever the uncertainty is there, what stringent procedures government or institute is taking care of to protect the environments and the biosecurity concern. That is also some of the generic has to be taken and some of the lab stage we have to take it. If you look the Indian perspectives, 3 ministries are involving in this: Health and Family Welfare Minister, Science and Technology, and Environment and Climate Change. And these are the institutes, ICMR, CSIR, DRDO is very stringently involved in this. Of course, and ICR is also in the fragmented way. In the plants, we are totally concerned, but we don't have any separate regulations. DPPQS is taking care of the plants, and the agriculture system is involved with this. But in 2004, we have the legally the guidelines has been prepared. This is national, Dr. Swaminathan, at that time he was the national farmer commission has proposed a proposal for establishment of national agriculture biosecurity program. And for this program, the biosecurity bill 2013 has been formulated, which is focusing all the sectors: animal health, plant health, marine organism, agriculture, important microbes. And 2018, when I was in NVPGR, 4 or 5 rounds meeting has been done. After 2018, no meeting has been done on this. And if you look the plant production quarantine order, which is, as earlier we have discussed, is taking care of the plant, the biosecurity risk associated with the plants. In our country, we have the different level of biosafety lab, starting from level 1 to level 4, and only very few lab are there in the level 4 categories, majority of 2 and 3 categories. India is also signatory of the Biological and Toxins Weapon Convention, where the confidence building measures are being taken regularly. NVPGR is very strongly taking part in this at the national level. And based on the what development has been done in the biosecurity bill, in 2018, 3, 4, 5 round meeting has been done at the secretariat level. I was attending that meeting at that time. So what government had decided, there should be National Agriculture Biosecurity Board instead of the creating a council. And under this board, there is a National Agriculture Biosecurity Council. And this all the system which is presently for the plants, animals, living organism, aquatic, and all these has to come under this council, and they will work. Whatever the regulation is there, they will work. Representative of these all the systems should be come under the biosecurity council and then biosecurity board. But even this system has not been approved at that time. Partially we are agreed with this, not totally we are agreed, being a NBPGR one of the leading center in the country. We are not agreed with this, but partially we are agreed. Let us see, because but the cabinet secretaries and the Prime Minister himself emphasized that the country must have a very robust and stringent biosecurity system. It was given in the cabinet note. The secretaries has to see whether the same system has to be improved to take care of biosecurity concern in the country, or whether we need a very separate and other system or organizations. Some of the few points, sides more is there. Some of the recent initiatives has been taken by the government of India. This is the very important and very, very significant for all of us, those who are working in agriculture sectors. For the regulatory mechanisms of microorganisms, including plants, the government has decided and made a portal. The DBT is the custodian of this portal. This is called as BioRep, Biological Research Regulatory Approval Portal. All things which are moving in the country and outside the country, whether coming or going, has to be mentioned in the bio portal. Each institute has to register there, and scientists concerned, those who are involved in the project, they have to register and upload all the information, and present approval system will work just like in the import of the microorganisms. NBPGR will give the import permit, everything, but finally it has to be, earlier it was decided that this is the only system portal where all the information is kept. Whenever it is required, it has to be assessed. But later on, it becomes approval authority based on the recommendations of the earlier system which is existing. And already the regulation has been passed. Even the council has also get the instructions that all the institute has to be involved to register them into the BioRep portal for the exchange of microorganism, including the plants. There is for non-governmental organization, government thought we must have a very stringent regulation rather than the BioRep portal. So International Collaborative Life Science Research Review Committee has been created. And this is for the non-governmental organization institutions. It was the co-chaired by the Secretary DBT and Secretary Health. Jointly, it has been co-chaired by, and all the movement for the non-governmental organization has to move through this. Recently, the National Security Council of Secretariat, this is the very confidential, we don't have to suppose to speak about this, but since this is the all of we are associated somehow with the biosecurity concern. So biosecurity and biosafety cell has been established, and several countries, almost all the ministries representative are participating over there, and every month there is meetings going on. Any biosecurity concern at the national level or international level, any threat has to be discussed over there very significantly, and directives to the each ministry is being issued. And we see that not only that, we for the exotic pest, lot of work the country's biosecurity concern is going. This is one of the example. In 2020 we have imported the parasitoids for the control of the cassava mealybug, and very nicely in the presence of the honorable MP of that particular area in Tamil Nadu, we have released this into the environments. So simultaneously, both way this is going on. And if you look the emerging challenges, this 4 T is very, very dangerous for the biosecurity. Trade, transport, tourism, and sorry, this is trade, transport, tourism, and this one is the last one. I think what is this? Also trade, transport, tourism, and this is also some movement, travel, trade, tourism, travel, transport, and trade. This 4 T is very, very dangerous for the. There are the certain challenges. Transboundary animal and plant diseases, movement of this is going on. Migratory bird and fishes and insects, they are carrying the pathogens, or they may be the itself, they may be the pathogens for that particular area. Weak capacity of some of the neighboring countries, they cannot, suppose in the Bangladesh something appears which is dangerous for India and Bangladesh not able to identify, this will gradually introduce in our country. Limited understanding and knowledge about the biosecurity, inconsistent approach system, fragmented accountability, insufficient use of available trained human resources, poor risk assessment, weak monitoring systems. And this is very, very important points. We at the institute level or national level, we don't have very robust monitoring system. Budgetary constraints, which is also very, very important. And what are the way forward? There are the certain policy issues where we need to be strengthen ourselves. Integration national biosecurity system. Biosecurity bill is not there, then even we are thinking to something has to come in the concrete, where all the ministry, all the department has to be jointly work together. Global initiatives, trans-international collaboration and cooperations. Safe trans-international collaboration and cooperation are achieving how we have the One Health program has been, CGR center has been started, where the concentrated efforts has been put on the biosecurity. Let us see how much they are successfully. Integrated surveillance program. This is very weak because every country has their own surveillance program. At the moment, if the regional level and international level, we have the surveillance, definitely we have the different categories of the pest in particular area. So the preparedness accordingly has to be done. Establishment of facilities, lab facilities, basal level facilities required. Prevention of transmission of the pest across inter or intra-country borders. The stringent procedure is required to the policy level, some procedure is required. E-reporting system has to be there. If something has to be intercepted anywhere in European country, very strong e-reporting system is there. Suppose in EU country, in out of 25 country, any border, anything had to be intercepted within a few seconds. All the countries, those who are the 25 countries, they know that from the Netherlands side or from the Germany side, something has been reported. But in our country, if something is reported in Kerala and Srinagar, we don't know. So this is e-reporting system, global level or regional level is required. Timely declaration of pest invasiveness. This is very, very important. Pest is there most of the time in our country, but we took the decision 3, 4, 5 years to declare that this is invasive pest and non-invasive pest. And what are the steps has to be taken to protect them? They are spreading very fast. National Diagnostic Laboratory Network is required. This is very, very, most of the time it is felt. Upgradation of quarantine facilities. Of course, it is, we don't have the, even in NVPGR, we don't have the international level of quarantine facilities. Lot of huge amount is required to upgrade the facilities. In national level, we have the good facility, but definitely there is the very large scope to develop and refine this facility to make them internationally. And national database, we don't have the database. What are the red list or different categories of the pest, like pest of the most significance, pest of the likely to be introduced, pest has been introduced, pest under the concern, whatever the database quarantine have there, and they are very old, they are upgrading gradually, but there should be the interactive database. Immediately one thing today intercepted, tomorrow it has been entered into the database. So that is required. So there are the researchable issues, lot of identification and classification, taxonomy required. Digitization of biosystematic keys, diagnostic protocols, surveillance of invasive pests, model for risk analysis. Simply we are searching the literature, but there should be a very computer-based model is there, because the risk analysis, simply we are intercepting something there, we are considering this very harmful. But whether it merely its presence, whether we have the options to mitigate them or not. If we introduce, what happens? If risk analysis clauses, you have seen the different step is there. So we are, whether it's how stringent we are following all these steps to analyze the risk, whether it has been allowed to introduce or not introduce. Epidemiology in relation to climate change is very, very important. Detailed study on alien pest. Almost all the basic experimentation is required on the alien space to define them and how much they are endangered for the environment and biodiversity. Eco-friendly salvaging techniques required. We don't have this, especially for the PGR. stimulated evaluation of mitigation options. This is very, very important. When any time, suppose COVID came, the vaccine was not there, and luckily it took time to develop. But in the plant pathogens, if you have the list of the categories of the pest likely to be introduced, depending on the their pathway of introduction, the international markets where the India is involved, we have to develop the mitigation options in an advance. We should not be, but what happens when the pest is introducing, problem is there, then we are searching what kind of options has to be. Human resource development, of course, which is going on presently, we are working on. Thank you very much. This is some of the points I would like to highlight, and we have the time is still 5:30. It is there, 20 minutes time is there. So we have the discussion, whatever 10 to 15 minutes. Any question, answer, any discussion. That's why I hurriedly I have finished 10 to 15 minutes before. Thank you very much. Whether I am audible or not audible?
Audible, sir. Audible, sir.
You are audible, sir.
Okay, मैं तो सोचा पूरा बोल गया, सुने नहीं.
नहीं sir, बहुत अच्छा जो है.
Because
Thank you,
lecture
thank
was
you.
so visionary, and where we are and where we have to go, you have shown the path.
So
Any questions from the participants? Kindly put in the chat box.
Chat box or speak, there is no problem.
Akshay, I want to update myself. You have mentioned somewhere agriculture biosecurity bill in 13 it was present in the parliament. But in 17 or 18, there was some drafting or some presentation in the parliament.
No, it was in parliament. 13 it was presented, and this it was the cabinet secretaries has discussed on this, and they have given one paragraph cabinet note. The exact language, it is very difficult to remember, but what is just note, because I have gone through this note with the joint, at that time joint secretary plant production was there. So the government has realized the importance of the biosecurity, and definitely it has mentioned that all steps must be taken to secure the country for in the angle of the biosecurity and biosafety. And whether the secretaries had to examine whether we have to establish a separate system organization, or the existing organization has to strengthen and coordinated mode, they have to work together. So that's why it was being discussed. So biosecurity, agriculture biosecurity bill, then they slightly modification, DPP said, did the modification in the drafting and all these things. Some of the points which has been discussed in the meeting with the JPP, Joint Secretary Plant Production, DPQS members from NBPGR, I was there, 2-3 meetings were there. Even Agriculture Secretary with at that time, Agrawal was there, Agriculture Secretary, discussion has been made. And they thought that there should not be a bill, because when the authority is created, the authority is independently working. They are not listening any department. They have told the NBA is one of the example, and FSSI is another example. Even they are not listening to the government Minister of Agriculture most of the time. So we have to create a such system under the Ministry of Agriculture, where all the system has to be strengthened, and proper participation of all the ministries should be there in a board and council will be there. And maybe the board is so big, so the other eminent persons from across the country from different sector is the member of honorary member of this board. So very 16-17 members, very big concept they have prepared. But I don't know, from 2018, I am not in touch with this. I discussed with the 1 month back, there was a meeting with the direct ministry. So I pointed out, and they thought that let us see, the new joint secretary PP has come, secretary has instructed him, let us see where this file is there and what development is done and in which it has to be implemented. So this is the only the.
So do you feel that we will be able to make a robust biosecurity system like Australia, New Zealand, Belize?
No. Unless we will not make a umbrella organization.
Okay.
That means single organizations, and all the department has to answerable to a single person, or that in the hierarchy base. Because if you have to horticulture commissioner, agriculture commissioner, and animal husbandry, all has to be there. All boss will be same. Then that they will work together. If there was in India, the people are listening only the boss. I'm not saying as a ADG plant production, but as a scientist, I am saying. So there is the always risk for implementation. But the government is thinking some things. Let us see what happens. B2 cell is nowadays. I am not discussing much more about this because every meeting they are saying don't say, because internationally people should not know what India have such kind of recognition. So they are the intelligence people, raw people and intelligence people. So they are inviting almost all the ministries. And we have to raise the point what national biosecurity concern is there. And they are searching the literature. You will surprise, they are updating me on agriculture most of the time. Something happened this. So some of the points they have pointed out and discussing if any regulation is required, and that they are also discussing. This is the initial phase, how this B2 has to function. Still they are not in the good shape. But of course, the government has the concern. Simultaneously, they are working on this. In plant sector, less attention is there, but animal sector and veterinary sector, even they are monitoring from where funds are coming, how many DAHAG projects are working on the very, very dangerous pathogens of human, animal, and plants, from where funding is coming, what kind of research, even who is going abroad for what. That's why BioRep portal has been made. If anything has come from abroad and who is handling, after handling, where this has gone, because in the northeast some outbreak has become, some diseases, animal disease and human disease outbreak was there while working into the laboratory conditions. So that's why the government is thinking and trying to make it as stringent as possible in the existing system.
डॉक्टर साहब, हमें तो एक चीज लगती है, अगर हमारा बायोसिक्योरिटी सिस्टम स्ट्रॉन्ग होता, कोविड से इतनी मोर्टेलिटी नहीं होती, इतना कोऑर्डिनेशन डिफरेंट मिनिस्ट्रीज में होता, तो शायद ये ज़ूनोसिस जितने भी आए हैं along with this one trade. There are small countries like Belize, South American country, small fishery based economy is there. He has also passed the biosecurity system in compliance with the FAO, and he is selling his fishes that they are free of any disease. So we are lagging far behind, and people like Dr. Shashibun Sharma, this alumni of IARI, he was director of biosecurity in Australia. They are voluntarily ready to help, and they have given presentation to ministries. But problem is that once they come, and next time they come, there is a reshuffling of the persons to whom they have interacted.
नहीं, वो प्रॉब्लम है ना कि प्रेजेंटेशन हमने जैसे प्रेजेंटेशन दे दिया, इसका क्या एडवांटेज है? अगर आप कल आप जेल चलाएंगे ऐसे फेंक देंगे, कोई पौधा लाएंगे ऐसे ही आप कहीं नहीं करेंगे. दिस इज द माइंड सेट इज रिक्वायर्ड. Two aspect is there. Legal framework is required, where all the stakeholders concerned to biosecurity come together in single umbrella. And second is the mindset has to be changed. People's mindset had to change. What they are doing, this is not good for us, not good for others, not good for us, including those who are handling this. So then it's become easy. Provision is there in our country, fragmented regulation is there, but this in fragmented regulation also very strong provision is there. For example, institutional biosecurity committee is there, the disposal committee is there, but how many people are following? आप ऐसे सीड को कहीं डस्टे गंदे में फेंक देंगे, कल्चर को कहीं फेंक दिया, अरे हमने भी किया. What I can blame to others? मैंने मोस्ट ऑफ द टाइम हम स्लाइड नहीं किया, वैसे कल्चर ले जाकर के ग्लास हाउस के पीछे रख के धो दिया. So but this is very, very important. Some of things before discarding the culture, you have to autoclave. Nobody is doing this. Either somebody wants to do, they don't have the space, autoclave, money, and all these things. So many factors there. But this definitely, if everybody will think, I have to follow stringently the biosecurity regulations, whatever existing is there, and it has to be implemented very strongly at the institute level, society level, state level, district level. Definitely there will be a change is there. भाई कोविड में हमें कोई कहा नहीं ना, सब लोग थूकते नहीं थे, पहले पुलिस डंडा से मारी जब सड़क पर थूकते थे, बाद में बंद कर दिया, आजकल फिर थूकते हैं हम.
नहीं, मेट्रो यू सी, मेट्रो एवरीथिंग फॉलो.
वही ना, आप पहले लोग कहीं कुल्ला नहीं करते थे, और थूकना नहीं हो रहा है, कुछ नहीं हो रहा है, सब लोग मास्क लगाए रह रहे हैं. लेकिन अभी जब कोविड ढीला हुआ, सब कुछ ऐसे ही हो गया. सड़क पर देखिए, अभी पहले कोई यूरिनल नहीं करता था, अभी मैं आरएमएल से वहां तक जाता हूँ, कम से कम 5-6 लोग कार रोक के सड़क किनारे दिखते हैं. तो ये इनबिल्ट होना चाहिए. और थोड़ा यह है कि बायो सिक्योरिटी बिल जो पास हो जाता तो उसमें प्रोविजन था पनिशमेंट का, लाइफ टाइम इम्प्रिजमेंट है, मैंने 3-3, 5-5 लाख रुपए का इम्प्रिजमेंट है. तो दिस whatever presently in biosecurity, any regulations, take the example of plant quarantine regulations, environmental protection act, and all these regulations, there is not a very strong punishment provision, because we are democratic country. तो वो जब तक punishment अपने यहाँ, तो जब तक डर नहीं, डंडे का पैसे का डर नहीं होता, हम सुनते ही नहीं है. That is required.
Yes,
तो
sir.
अगर जो एग्जिस्टिंग स्ट्रक्चर है उसी में सब लोग अगर सोच लें कि वी हैव टू डू वेरी स्ट्रिंजेंटली, ऑनेस्टली, लीगली, so 50% problem will be solved. आखिर में जो बाहर से some pest is coming, we are bringing the seed material. I will tell you, every month, every intelligence bureau is submitting the report to me through the DG. Every month, what happening in the import and export of the agriculture commodities. For example, fall armyworm, they have given a very exhaustive report and pointed out why not this is deliberating introduction of the fall armyworm. They have given the plot number, farmer's name, where this seed has been grown and from where this has been purchased.
Oh my God.
And later on, the company who has given the seed, they have get the registration of the pesticide for sale. And when I inquire with the ICR institutions and SAUs, we cannot validate this.
Yes, sir.
So you can see how much they are concerned, and being a plant scientist, how much we are. Even small things, you say, you will surprise. Last month, 2 months back, I got a letter. Every month agenda is coming, and on that we are discussing in B2. They have told us that there are the resistant herbicide resistant rice varieties going, whether this is good for the environment or not, and is there any conflict between TNU and IRI? I wrote to the director IRI and TNU separately and got there is no conflict, and discuss with them everything. So that means the country is vigilant nowadays. Maybe the person, those who are involved in this intelligence and raw, they don't have the expertise in this area, but even then they are vigilant, and this is the indication from the government side. We should also be vigilant and aware.
Yes.
To make the country, the government don't want to be introduced such a very big problem like COVID in the plant system or animal system. This is the one animal disease. Immediately you see this ICR Institute had developed the vaccine for this. Next month it will be the Bharat Biotech will be selling this vaccine for lumpy disease. So ICR is very vibrant, big organizations, a lot of good pupils are there, only motivation and then refinement in the system and is required, how we have to adopt this.
Yes, sir.
I mean, NBPGR, if you take the example in quarantine from last 1978, nothing, not a single pest has been escaped from the quarantine processing. But such kind of the system, if everywhere is there, then there is no problem. Of course, there is the problem in the bulk import. A lot of consignment is coming. We have seen at the seaport how much it is dumped over there and how the sample has been withdrawn. You are talking Australia. They have very robust system at each and every step. We don't have manpower. We don't have man. All our quarantine stations, they don't, only microscope they have, and maybe 2-3 months they are not being used. Maybe in 1 year, 2-3 times they are using, because 2-3% in each station, they are in entire day they are doing file business. So practically, when we visited and see, practical problem is there. If we can, whatever the infrastructure and policy makers make in our country, what system is there, if it has been supported by human beings, supported by the infrastructure, supported by manpower, so system can do up to any extent.
Yes. So any question from the participants? Before that, I conclude the lecture. Any question regarding biosecurity?
And one question has been asked there, is there any new law for formal seed system, sir? Formal seed system, I don't know what formal seed system is saying, but there is a new regulation for seed. Even the seed bill is being revised. So new bill is coming for the seed. Already, I think 1998 has been revised. 2001, I don't remember, but still it is some revision is going on.
So any question from the participants, please? If not, dear participants, I am happy to share that Dr. Dubey has given an enlightening lecture on biosecurity. He has started from the elementary thing. What is
that's one person has got difference between the biosecurity and biosafety. As I have told, very, very thin line is there.
Yes,
but this is quite different, very significant difference. We should not, this two term cannot be used as synonyms. If you see the definition of biosafety, the discipline which is addressing the safe handling and containment. That means two action you have to take, handling and containing them. Kisko karenge? Infectious organism and hazardous materials. That means infectious organisms and hazardous material, how safely you are handling them, containing them, not to release into the environments, not cause any harmful effect to the human being. This comes under the biosafety. And and to regulate this biosafety, we have the WHO practical guidelines. अब nutshell में आप कह सकते हैं, when suppose any organism which is very dangerous, take the plant pathogens, which is suppose if you are any quarantine pest, you can take the tropical race 4 banana wilt, you can take. When you are handling into the lab, you take all the precaution that either if it is pathogenic to human being, it should not cause harm to the pathogens. All the safety gloves पहनना है, apron पहनना है, coat पहनना है, sleeper पहनना है. We are not doing anything. हम ऐसे उसी से इनोकुलेट करते हैं, ऐसे नाक पोंछते रहते हैं, मुंह में लार भी चू गया, पोंछ लिया, something we are doing. So all safety precautions we have to take into the lab, that the organisms which we are handling are chemicals, toxic chemicals or substances which we are handling should not cause harms to the human being, and how we have to contain them and handle them and contain them. रखें कैसे अच्छे से और कैसे उसको harm न करें. This is the, but when it's release environments, suppose हमें infection हुआ, that means lapses of the biosecurity. When the COVID got infection to a single person into the environment in the China, Wuhan, that means there is a lapses in the biosafety concern. And when the person has gone outside and spread in the population, then the biosecurity concerns involved. That means the other country will not allow to take the passengers from the China. All the movement has been stopped. Even the particular districts are block or taluka get infected. We have take all the precautions, biosecurity concern that it should not be spread in the others. So this is the difference. in the nutshell, very precise way or simplest way, the biosecurity and biosafety.
Very much, sir. Thank you. And you have aspect of biosecurity, and you have given the visionary lecture what we should do for a robust biosecurity system in India, like other countries. Thank you very much. Keep in touch, and we will also keep updating from you in future also. Thank you, sir, for sparing time.
Thank you very much once again. Thanks to the
Thank you, sir.
organizers for giving me the opportunity to be here with you all, and thanks to all the participants. And thank you very much.
Kindly give a big hand to.
Thank you, sir. Namaskar, sir.
Namaste, namaste. Thank you,
Sir,
madam.
namaskar, sir.
Thank you, namaste.
So, Mamta, can we wind up?
Yes, sir. For today's session, the schedule we can wind up.
Good evening to everybody. Thank you very much.
Thank you, everybody. And tomorrow we will meet. We will be updating the schedule of tomorrow in the group. So I think we should close this meeting now.
Thank you. Thank you, Mamta, for taking every care. Thank you very much.
Thank you, sir.
Okay.
मैम क्लोज कर दूं मीटिंग?
I think close कर सकते हैं, sir.
Okay.
Okay. Thank you so much, sir.